Last row, please come forward and be seated in the first or second row.
Rule of third থাকে ন মানে সাইডে রাখো
Hello. Good morning all. I hope सबकी नींद बहुत अच्छे से हो रही है. सुबह सुबह का लेक्चर है, काफ़ी बोरिंग होता है, थोड़ा टेक्निकल है, थोड़ा सा आपको कोई क्वेश्चन है you can ask. ऐसा नहीं, बीच में also you can ask, later also you can ask. So myself Abhijit, working as an application specialist in Agro, and अगर my education is like I've completed my post graduation in bioanalytical sciences from University of Mumbai. यहाँ पे I'll be talking about a chromatographic technique, a separation technique. Yesterday's lectures were mostly on what kind of therapeutic effect we can have from using different kind of herbal drugs, combinations, formulations. Now, how to test those formulation? What are the guidelines? How can you quantify them? How can you standardize them? So this is one of the technique. I guess most of you must have somewhere or the other perform thin layer chromatography, normal TLC. Now take a plate using a capillary, apply a sample, go for the development and check for the separation. So first basic, chromatography was invented by Michael Sweat. So separation of pigments were performed. Then in the 60s, it was basic TLC. Then slowly it developed to instrumental TLC, where instruments were used for application of the sample. So, slowly human intervention was decreased. And currently it's like the fully automated, just, put your stationary phase, put your sample, load your standard, keep the mobile phase and go for the analysis. So this, I guess everyone must have done. Thin layer chromatography, take a plate, apply and go for separation. But now currently it's the high performance thin layer chromatography using all of instruments and nothing manual. So you can see the major difference is like the application, the type of application. On the right hand side, it is the spot application. And on the left hand side, you can see that is the application in the form of a band, thin layer or band, or a very sharp band using a spray on technique. So even while the sample is being applied, it is not touching your surface. It is just above the surface and spraying on it. So you can see here, majorly the surface area is playing a vital role. Higher the surface area, higher will be the diffusion, the mixing and improper resolution of the bands. So again, this is on the right hand side, HPTLC, on the left hand side TLC. You can see the major difference is like merging of the bands, which I just told you, mixing of the bands, improper resolution. So basically what I'm doing is nothing but miniaturizing the complete step. Instead of developing it to a farther distance, I'm reducing the development distance as well as the sample application quantity. So this is the whole setup of the HPTLC instrumentation. So there are different steps. Whereas I prepare the sample, I apply the sample, I go for the development, I go for the photo documentation or visual documentation, that is using a UV cabinet, just say 254, 366 white light. So this again is automated. So first you can see that is the applicator. So that there are different instruments for each of the steps for performing a particular HPTLC analysis. So first is the applicator, then is the developer or the development chamber. Then we have the visualizer, that is a photo documentation. Then we have the scanner, that is nothing but your detector, which works in the wavelength range of 190 to 900 nanometers. UV, visible as well as fluorescence. So deuterium lamp, mercury vapor lamp and tungsten lamp. Then we have the MS interface. That is nothing but a hyphenated technique where you can combine two different techniques and get the result. So if there are any particular unknowns or any impurities which are present in your sample, they can easily be detected using this mass spectrometer. And then we have the derivatizer. All of these instruments are in the end controlled by a single software, that is the VisionCAD software. So you just have to connect and you can use the instrument. Now why HPTLC? You will say, yaar, hamara kaam to ho raha hai isme. But why this particular technique? Basically, you can go ahead with the quantification part, where you can find out the unknown mein se kitna that particular component is present in my sample. So if I have a standard, I can plot a standard calibration curve graph, and then everything is software controlled. You have very high accuracy, precision. Why again? Because we have proper validated methods. So validated method means reproducible results each and every time. You can go for multiple detection techniques. That is like once you have done with the analysis, you can analyze it under UV. You can use visible light. You can use fluorescent light, or if you want, you can hyphenate it with mass spectrometer, NMR, IR, or something called as effect directed analysis. That is nothing but whatever microbiological assays which you perform, so that you can directly perform onto the plate. How to do that? I'll be explaining you. So this is the basic thing which we always perform when we are working with any herbal samples. That is the fingerprinting, because each and every plant or I'll say species have a specific fingerprint, as we have like every one of us has in the room which is present here. So if I put my fingerprint, that will give my identity. So basically I can just profile a particular plant, how many number of components are present, what components are present, what is the concentration of the active component which is there, what is the geographical variation which is making them separate. So basically what I do is I prepare the sample, I develop a fingerprint and I match it with a botanical reference material, that is a certified raw material ki yes, this is belonging to this particular specific species collected from here and all of the other classification, taxonomical classification. So that is how I can identify just by applying the sample. From the marketed samples also I can find out whether it is adulterated or it is the pure sample which I am working on. This is one example for the same thing. That is each one has a unique and separate fingerprint. All are piper, but different species, nigrum, cubeba. So you can see there will be some bands which are common for all of them, but some which are specific to the specific species. So this is how you can develop a specific fingerprint and you can identify the species differences or say geographical variations or say at what time it was particularly harvested. Is it of the particular species or is it some other species which was given under that name or it has been adulterated? So how to do that? How to perform HPTLC? So first thing, basic thing is your sample preparation. Here we don't use any complicated kind of technique for sample preparation. We try to keep it as simple as possible. Why as simple as possible? So that it will give you reproducible results. Or if you are using a specific technique or a unique technique which you have developed, you have to make sure that you have to validate that process so that each and every time you do it, you get the same results. Or you do it here in Assam and I try to do it in my Mumbai lab. So it should give me the same result. So I have to use the same particular method and technique which was used. Then I apply onto the plate, that is sample application onto the stationary phase. Then I go for the development. After development, since this is a visual technique, I go for the visual detection, that is taking the photographs. Then the densitometric scanning, that is trying to find out how much is exactly present onto the plate, what all components are there. And if needed, as I said, I can hyphenate the same technique here with MS, IR, NMR. So what I can do is I can simply collect the sample in a vial and then I can proceed with either MS, that is GCMS. If you're performing LCMS directly, you can hyphenate it with the technique, the instrument. Then if you want to perform IR or NMR, liquid IR or NMR, you can collect it in a vial and you can proceed, or you can go ahead with the effect directed analysis or bioactivity. Now we come to the regulatory acceptances, because when we go for any standardization or validating a method or developing a method, everything comes to the regulatory aspect. Like, is this method certified, or valid, or if you want say it is given in a particular monograph, what are the guidelines which you are following? So here there are different chapters, USP chapters, European Pharmacopoeia chapters, which are mentioned, which state specific parameters, which will help in determining what steps to follow, what in what way you should proceed with the quality control. So USP chapter, which is official from 2018, HPTLC methodology and fingerprint identification of materials of botanical origin. Then USP chapter 621, 202, even if you want to work with oils, because there are different oils which are used in the Indian system of medicine. So those also you can identify, you can characterize them, that which is used, what is the particular oil which was used, what is the mixture. So those mixtures you can separate properly, you can identify them, you can quantify them. USP chapter 2251. Then European Pharmacopoeia chapter, that is 2.8.25. So it's similar, but as the countries change, that is US FDA, European chapters, then the rest of the world and everything. So that way some of the changes are made, but otherwise the basic is almost the same. So you have to ensure that whatever standardization or whatever you are doing, you have to follow specific chapters and guidelines which will tell you how to proceed. Now, what does this chapter say? Essential requirements for QC, that is quality control. So there should be fixed sample application position and band length. So what should be the length of the band? How much should be the quantity which should be applied? What should be the dimensions of the stationary phase? Should it be a big plate? Should it be a small plate? Or you can select it as per your requirement. Then what should be the humidity? Because humidity plays a vital role in your separation. Higher humidity will result in lower RF values, whereas lower humidity will result in slightly increased RF values. So that you have to make sure that it is maintained at a specific percentage. Then reproducible drying of developed plates. So each and every time when you dry the plates, so it should be in a specific manner, it should be in a specific pattern, it should have a specific method. ऐसा नहीं कि directly you just keep it in the sunlight and you dry. No, you have to dry it properly. You can dry it using cold air, or you can dry it using your hair dryer, or you can heat the plate. So there are specific methods by which you do. Then photo documentation as well as, and when we come to the instrumentation part, it is very important that whatever instrument you are using, it should pass, or it should have IQ, OQ and PQ compliance, where installation qualification, operational qualification and performance qualification. That is, Is the instrument doing what it is intended to do? Then is it giving the required results for which you have purchased the instrument? So these are some of the parameters which are very important when it comes to any analytical instrument. And you have to make sure that whatever instrument you have or you are using, that has passed all of these parameters. Otherwise, your results cannot be valid, because once they are passing all of these parameters, it is letting you know that the instrument has been properly calibrated. It is or it can be used for the intended purpose. So these are some of the parameters which the USP chapter mentions. That is, what should be the size of the plate, what should be the distance at which the sample should be applied from the bottom of the plate, then what should be the distance which should be left from the x-axis, that is from the left-hand side, then how much should be the development distance, should it be applied in the form of spot or in the form of a band, and what should be the band length, what should be the development distance, and how will the saturation and humidity affect. So saturation, in chamber saturation, what we simply do is we pour in the mobile phase or the solvent of our interest in the chamber, keep the chamber closed for a certain amount of time. So what happens is the liquid will slowly turn into gaseous phase. So that gaseous phase will be saturated in the complete chamber. So this will ensure that whenever you're dipping the plate inside, the liquid mobile phase as well as the gaseous phase both will play an important part in the separation. Now, as I said, in 2019 this system was launched, that is CAMAG HPTLC Pro. This is the fully automated system, and currently in the whole of Asia it's only present in our lab in Mumbai. So that's a fully automated system. A small video. I have two videos here. So this is the instrumentation. This again has multiple instruments for different steps. What does this video tells us is, like in simple or layman's language, it's kind of a relay race where it starts and goes on in the front direction, as well as nothing is manual. All of the things are taking place in the instrument. So one, I have completed with the application, automatically my plate will be passed on to the module for development, next module for development through the conveyor belt, and then so on. So this is how first, second, third, the plate will be going for application. The sample will be applied onto the plate. Then it will go for development. And again, once it is finished, the development is finished, it will again come back and get stored into the plate storage module. So this will keep on happening one after the other. So you can just program the sample applications and everything, and you can continue with your work. And why, basically you'll be thinking why I'm telling you that this is the most useful system, is simply because you need very less sample preparation. Directly you can just macerate or cold extraction, hot extraction, whatever you do, just apply, go for development. Plus you can go for multiple sample applications, like more than 15 samples, say basically 18, 19 or even 20 samples you can apply on one single plate, go for the development, as well as you can go for quantifications. So then this is the next part, that is the regulatory compliance. So there were some chapters which mentioned why HPTLC is needed or what are the parameters you need to follow. Then again we come to the next regulatory compliance part, that is the 21 CFR Part 11 compliance. Code of Federal Regulation, which states for the electronic signature, which also complies with the current good lab practices as well as CGMP manufacturing practices. Then IQ, OQ, PQ compliance. All data is stored so you can anytime go back, check your data, digital signatures, user access controls. So basically you have the full audit log, audit trails, whatever you do, everything has data which is stored, log which is stored, so you can just go back and check what was done, and it complies with the latest regulations. So as I said, generally we prefer to keep the sample preparation as simple as possible. So how do we prepare the sample? If it's a powder, I just take the powder, weigh 100 grams per 1 mg of methanol, because most of your components will be dissolving in methanol. If not, you can go for hydroalcoholic extracts also, which will give you good extractions. Then sonicate it, centrifuge it, and use the supernatant for application. So I have completed my sample preparation. Now I come to the application part. So there are different kinds of applicators, the semi-automatic and the fully automated ones. Only difference is the sample throughput. The automated will have higher sample throughput, whereas the semi-automated will have a little less sample throughput, but the precision and accuracy will be the same in case of both of them. And the last one which you can see are the robotic applicator that is completely free from human interventions. Just prepare your sample, load it in the vials and give the command from the software. So now what happens is we have different kinds of matrix, like it can, and mostly plant matrix is also very complex. If you're working with biological samples, even then the sample matrix is very complex. So cleanup of sample sometimes might become difficult. So even there are technique after the application or just preparation, and you can do that. That is your in situ cleanup. So on the plate itself, like after the sample has been applied, you can use petroleum ether and you can clean up the sample which has remaining fats or fatty material which is present and which can interfere with the separation of your sample. Then you can go for micropreparative isolation, that is nothing but isolation and purification of your compound of interest. Then you have quantitative and the superimposing. So after the application has been completed, I need to go ahead with the development part. So again, you have different ones. So the auto development, gradient and robotic are again fully automated, where you can maintain the humidity control. Even the drying of the plate is also done automatically. This gradient system here, you can go for some R&D, because since there are many components which are present, it becomes difficult to separate it in one single go. So what you can do is from time to time or step to step, you can just go on changing the composition of the mobile phase. That is nothing but you can change the polarity and you can go for separation of multiple components on one single plate. So this is the same thing which I was telling you, that any combination can be used for separation. Your stationary phase, mobile phase, there is no specific limit or thing that no, only these kind of solvents can be used, these cannot be used. So this, as I said, USP compliant, so it has compliance of the regulatory body. You have automatic development, humidity control. So whatever humidity percentage you need, you can just select the salt, which will give you a saturated salt solution of which will give you the exact percentage of humidity, and then you can proceed. So automatically it will maintain the humidity during the development. Then as I said, if you have multiple components which you need to separate, so based on polarity, you can keep on changing the polarity, either from polar to non-polar or non-polar to polar. And this is how your components will be getting separated. So first, if you take polar, so you're working with non-polar component, so you can slowly go on reducing the polarity. If it's the other way around, you can just change from non-polar to polar. Also lipids and fats also you can separate or work on specifically on this technique. So if you have a mixture of phospholipids or lipids, cholesterol, you can also easily separate them. So now once after the separation has been completed, I need to detect what is present onto my plate. So I go for the photo documentation. So white light, 254 nanometers and 366 nanometers, that is my fluorescent wavelength, short UV and long UV. So in short UV, my plate will show fluorescence and the compounds which are absorbing UV light will be shown as dark black color bands. Whereas in fluorescence, it will be the other way around. The plate will be completely black, whereas the compounds will show specific fluorescence. Now this is comparison of multiple samples. Okay, so if you're working on a research project, you generally generate large amounts of data, and compiling the data and viewing it all together becomes different. So in the software itself, what you can do is you can select the tracks or say peaks or spectra of your interest. You can just electronically take them, combine them, and you can together view it on your laptop or your computer screen. Then sometimes what happens is if you're working on a specific component, it is neither visible in the visible region, the UV region or the fluorescent region. So in that case, what you need to do is you need to derivatize that component. So basically what I'm doing is I'm adding a chromophore to it, which is imparting a specific color and then giving a specific colored band for that particular component. So after that, I will just heat it and get the result. So you know there are different classes of compounds like alkaloids, glycosides, sterols, phenols, phenolic compounds, flavonoids. So how can you detect as to which class of compound you are working with? You have specific derivatizing reagents which will impart specific colors, as I said. So you need to derivatize the plate and then you can get this kind of result. So the first year is your alkaloids. We're using Dragendorf reagent. So that will give you the presence of alkaloids and so on. There are different reagents which can be used to detect specific or different classes of compounds. Now we come to the hyphenated part. So now once the separation is finished, fine, everything is done. But if there are any unknown components or something which is new to you, how to find out the structure or how to find out which particular component you are working on. So what you can simply do is you can hyphenate HPTLC with mass spectrometer. So once my separation has been completed, I will hyphenate it with the MS. So the plate itself, I will connect it using the HPTLC MS interface between your existing liquid chromatography and mass spectrometer. What will the solvent from the LC system will do is it will come onto the plate, dissolve whatever is there onto the plate and directly send it to the mass spectrometer. So this is how it works. So specific band is selected of my interest. The piston will be lowered down. The solvent will come. It will dissolve whatever is there onto the plate and then it will send it to the mass spectrometer, which will give me the specific M by Z ratio. So this is how I can at least find out the basic mass, or if I want to be more specific, I want to elucidate the structure, I can combine it with IR, NMR, and then I can elucidate the complete structure of that particular component. So this is a common technique nowadays because since our Indian system of medicines like Ayurveda, Siddha, Unani, everything, like what happens is it gives somewhat better response as compared to, it has a holistic approach. So majorly people are turning towards those, and to make out a profit, these can be easily adulterated. So they say ki ha isme, this is a painkiller or this is something else which is giving you the required effect. But if you check, it has been actually adulterated with a corticosteroid or say whatever is a synthetic API. So this we had done for the Maharashtra FDA lab. So it was added, tadalafil was added in the Ayurvedic sample. So we quantified it using the HPTLC system, as well as we use HPTLC MS and went for the confirmation that gave the mass to charge ratio. So it has tadalafil has your mass as 388. So on the positive mode, it showed 390 and sorry, it has 389. So on the positive mode, it showed 390 and on negative, it showed 388. So the same thing was done with the standard component as well as the band which was obtained from the sample and it was confirmed. Then we have something known as bioactivity. So generally when we are working, clinical trials and this part forms a later stage. But to find out a particular component which actually has the value which you can use for your further studies. So this is one of the simplest technique where you can do. So the microbiological assays and all directly you can perform onto the plate, or say if you want to find out some toxic agents or toxicity producing components. So you can use this technique. What you need to do is you need to work out on the separation part of the sample. So that plate, you have to coat it with a bacteria known as VIBGYOR fishery. So that has bioluminescence, which is not visible to your eyes, but the instrument, through the instrument, you can get a result kind of this. So the black patches which are shown or the black bands which are shown indicates the death of the bacteria, whereas the bright background will show the bacterial colony which is still living and giving out the bioluminescence. So this is a kind of toxicity testing. So toxicity profiling of water, your foods, toxicity from foods, your herbals. So method development and validation. So whatever method you're using, it should be properly developed as well as it should be validated. So there are different guidelines which you can use. So the common which we use is your Q2R2 guidelines, ICH guidelines. There are different parameters which we need to follow. Now I won't go into the details because it will take a lot of time. Then we come to the application areas. Different books are available. So basic, we come to this one. So yesterday, if you remember, we were speaking about the Negundo or Nirgudi, which we say Vitex Negundo. So it can be detected, or the fingerprinting can be done, and it can be found out that is the sample which is being commercially used is actually Vitex Negundo or just some, adulterated sample which has been given. You can find out the proper profiling. Also, you can quantify the active ingredients which are present and which is giving the therapeutic effect. Then this is your kokum or simply Garcinia indica. So garcinol. So this was a formulation. We had done this recently. It was done for your drug testing lab in Raipur. So they also published a paper and got a prize for it. So this is a mixture of total 8 different plants. We developed 8 different methods for separation as well as quantification of those 8 different components of those particular raw materials. So here is just one example of gallic acid. So that was quantified and as well as it was confirmed using the spectrum that this is the gallic acid which we are working on. Then this is ginkgolides, A, B and C, ginkgo biloba plant. This is beta cytosterol. So quantification was done. This is again one more example of your nimesulide and diclofenac adulteration. Again for FDA Maharashtra we had done this. It was detected, quantified as well as confirmed using the spectrum. So diclofenac standard spectrum, nimesulide standard spectrum, it was overlapped with the samples and it was confirmed. Now when I say adulteration, so one species is substituted with the other. So normally in our general use, whichever curcumin we used, so that is nothing but your curcuma longa, which has 3 curcuminoids, curcumin, that is the topmost one. The middle one is D-methoxycurcumin, and the bottom one is the bis D-methoxycurcumin, which has the lowest RH value. So if I simply change the species from Aromatica to Xanthorrhiza, so you see the amount of bis D-methoxycurcumin is going on decreasing if I increase the percentage of Aromatica or Xanthorrhiza. But there is one specific band which is getting detected. So this will tell you the difference in the species to species variation, and you can find out what exactly you are using or working on. Then again, one most important part for allopathic as well as Ayurvedic medicines also, that is the stability, which is very important. So that if you're preparing a specific formulation, it's not that you can just keep it for years and then later use it. You need to find out the stability also, the degradation profilings also. Because if you store it, since mostly those are your secondary metabolites which tend to go on degrading. So you need to check out that are they staying stable, or if in case they are getting degraded, to what extent, and is it present in the required range when I'm taking it for the intended therapeutic use. So this was again done in our lab. This is an old study. So one was done in 2008. The same samples were analyzed in 2009. So curcuminoid, gallic acid, boswellic acid, and gingerol. So you can see the change in the percent weight by weight. So this is also very important and easily you can do. So multiple samples can be done together. Then pharmaceutical applications. So again, quantification or label claim. So if they are claiming that this much mg or this is the strength of your tablet or solution or whatever it is, you can actually confirm it here. So quantification. This was done for Zydus Cadila Ahmedabad. So this was actually a very difficult thing for us because this miltefosine is generally used in leishmaniasis. So what happens is this is not getting detected easily. So after derivatizing it only, it was visible, and that is how we had to detect, quantify it, and we also validated the method for them. So again, degradation profiling, azithromycin, cefixime. Then quantification of simultaneous detection and quantification. So this also can be done easily on the same plate. This is your content uniformity. As I said that yes, the label claim they say कि हाँ इतना percentage है उसमें या this much quantity is present. But how will you verify it? So you prepare solutions which are 80, 100, 120%, and then you go for the quantification and you find out that exactly that much is present. If yes, then okay. If not, then what is the range or the deviation in which it is present? Then detection of artificial sweeteners, sucralose and fructose. So this was also quantified. Common one we had taken in, purchase it from some local vendor on the street in Mumbai, and that we, compared it with actual your reference material for your paper. So we could easily find out the adulteration there. So yes, and this is a small lab in Mumbai. So we have all the instruments there which we use. So right. Does anyone has any questions, or we can proceed? No questions? Yes. No, it's okay. Yes.
Of large quantity of samples.
Yes, you can do it. Yes. This if you see यहाँ पे I have mentioned. There are slides where I have mentioned the cost also. corner, left corner में. Yeah. So 800 for the complete analysis, but this is also slightly on the higher side. So Abhi, this is the total cost which I am considering in case of the use of stationary phase, the use of solvent, the use of your electricity, whatever is used, the wear and tear of the equipment, as well as all the other consumables. Yes, that can be done. हाँ ये दिस इज जस्ट द कॉस्ट कंपेयर टू द अदर. यहाँ पे कैसे किया था हम लोग ने कि इफ वी गो फॉर एचपीएलसी, तो एचपीएलसी में क्या होता है, आपका जो मोबाइल फेज विच यू आर यूजिंग दैट इज नॉट इन ml, दैट इज इन लीटर्स. तो वहां पे क्या होता है कि यू हैव टू क्लीन द कॉलम, इक्विलिब्रेट इट, देन डू दैट. सो वो कॉस्ट के बेसिस पे दिस इज मच मोर चीपर, प्लस यू कैन डू मल्टीपल सैंपल्स टुगेदर. So in that case, I have mentioned it. But if you want, yes, you can send your samples there also. We can do that way. Cost of the instrument. See, basic agar if you see plan to take this whole instrument, like for quantification, everything, and then so that will come around 50 to 60 lakhs. And running cost is very low because consumables hai but less hai. But 50 to 60 lakhs will be around 60 lakhs pakad lijiye for the whole system. Yes, it is affordable. See, extreme large quantities, this is not, I won't say this is feasible. For that, you can go for LC, preparative liquid chromatography. But if you're working with some active components which are required in less quantities, so this is the best and the cheapest one. So what kind exactly are you speaking? Like how much quantity? Rough estimate. If I say caffeine, okay, I want to purify it. So what will be the rough quantity which you're planning to? No, the actual quantity which you're trying to get out from it. Curcuminide. So yes, that you can separate because curcuminoids, yes, 3 fractions. So basically what happens is these are available in the sample in very high dosage. So that you can easily separate in milligrams also. Yes, definitely. Yes. So here what I can suggest is, so what generally we do is we apply small bands. So instead of a small band, what I'll do is I'll increase the concentration by around 5 to 10 times of the same sample. Okay, and instead of one small band, I will apply one big band onto the plate. So what I am doing is higher quantity as well as higher concentration. So this will result in the quantity of the extractants also will be increased.
But in that case, we'll have to pick it up from the band and then solubilize it in the solvent.
Yes.
That's fine.
Because if ekdam high quantities hai, to I will prefer that then this won't be feasible because it will be very cumbersome. But small quantities may definitely yes, this is feasible.
For research purpose,
It's I'd say it's the best thing because most of the time क्या होता है कि also now generally ऐसा होता है कि research के लिए you need small quantities and if you go to purchase those are very high. If you go for USP and European pharmacopoeia standards, so those are pretty high in cost. So it becomes easy if you just purify it at your lab itself because most of the students which they use, I know most of them who just purify the samples in their lab and use it. So and easily you can get around I'd say 90-95% of pure samples, which is more than enough for them.
Thank you so much.
Okay. Any more questions?
I'd like to understand regarding the gradient illusion that development and further. So in our lab, I am using the not the automatic one for the development sample. So we need to saturate the sample before we can develop our whatever the state is there. So in case of your gradient illusion, so how do you manage that? Because you are saying that we will change the solvent time to time.
Okay.
So how to?
this basically works on a different principle altogether. So here we don't use any kind of saturation. This works on vacuum. So what happens is, instead of saturation, there is complete vacuum which is created in the chamber inside, and based on that vacuum the separation is there. So what happens is, if this is my plate, so if I am working say on this particular plate, I have multiple samples. So what I do is, I give stepwise grading. So say for the first 10 millimeters, I use a certain mixture or component for the separation, certain concentration. Okay. Then I will program that this is the composition of mobile phase which has to be used. So I will program it. So that pre-programmed mixture will come into the chamber below it, get mixed, come into the chamber. Till 10 millimeters, that particular solvent will be used. As soon as it reaches the 10 millimeter mark, it will be sensed by the charge couple device. The solvent will be evacuated from the chamber. It will be dried using the vacuum, and then it will go on to the next step of development, where the next programmed mixture will be coming into the chamber. So this is how the development will happen when you are using the gradient. And there is no saturation, it works on vacuum.
Thank you.
Any more questions? Or we can proceed. So if you feel it, or you want, you can just visit our lab in Mumbai anytime. It's free for all. You can visit it. So, okay. Okay, thank you very much.
Thank you so much, sir, for the interesting detailed presentation on HPTLC, where I'm sure which will be useful for all of us in future. Coming on to the next dignitary, we have Professor Alka Sharma ma'am, who has been working in the domain of food and nutrition since 1996. Her contribution towards the field has been enormous, and she has published numerous research papers and journals.
I think I'm also not very tall, but I'll try to manage without this. Thank you so much. Very good morning to one and all present here. To start with that, we thanking the organizers for giving this opportunity to interact with all, and of course to you for being present here and listening to me. So I'll try to do as much good as possible, and I'll try to make it more interactive if I'm permitted to. Questions can be in between also, I won't mind that. Because most of you, I believe, are engineers here, but some of you might be from medical sciences or biological sciences background. Because this particular topic, today we will be speaking on more of nutraceuticals with specific reference to flaxseed. And probably many of you must have heard, and maybe you are consuming also in your daily diet, or maybe your parents may be consuming it. It is considered one of the wonder crop also. So before that, she has already introduced me. I'm serving for Guru Jambeshwar University of Science and Technology, and it's a state-owned university. And we have very courses which are job-oriented also, and more of what you call it, like applied subjects are there, other than the basic subjects. So my department, food technology, like yesterday I completed my 26 years of experience. This I would like to add, because it was my job anniversary yesterday. Thank you so much. And I'm happy to be here with all of you, celebrating that particular special day of my life. So we prefer, rather I'll say that we are trying to shift our area of interest, and we want to shift from allopathy towards more of Ayurvedic sciences. But that doesn't mean that I'm against allopathy, because when the situations are very grave, you cannot wait for the results to come. You have to go. I respect all the lines of treatment and all the systems of medicine.
But still we can shift, like we can try to avoid that problem which can occur in future. Like prevention is better than cure, you must have heard that. And like I'm a believer in our traditional sciences also, and I feel there is enormous potential in so many plants we have around, and every plant has value. Maybe we are not aware of that, or maybe we're not able to exploit that, and ho sakta hai future mein we are able to do that. Nature has created such a huge amount of biodiversity. And in this particular area, for the last 2 days, I have been observing there are so many medicinal plants also planted on the campus itself. And otherwise also in the city, I could find those. And every place has its own flora and fauna, which should be respected, and we should try to use it instead of going for other. And that is fine going for us like for some time, but not permanently. So today, this is the slide which I'll be talking about flaxseed. But we are working on other nutraceutical crops also. Like my scholar who did a PhD on this particular topic, I'm lucky to have her here, Dr. Priyanka, with me today. And we can continue the discussion even after this session, because 30 minutes or 45 minutes is not sufficient to discuss everything about this particular crop. We can continue it later on also. So like we are working on moringa, then a number of fruits also, like wood apple, then garden cress, aula also. A number of crops are being worked. So let's come back to flax seeds. आपने सुना भी होगा, like linseed, flaxseed, or ulseed has so many names. Linum usitacissimum is the Latin name, and this name has a relevance also. This name has been given because it is very useful crop. So that is why it has been termed as wonder crop also. May I know how much time is available with me so that I can move accordingly? Like, okay, maximum 30 minutes. Okay, thank you so much. So I won't be discussing about how it looks like, because you must have seen it. But this I would surely want to tell that it is a very rich source of omega-3 fatty acids, which are getting popular in newspaper articles, in day-to-day life also, in many food packages also you must have seen it. Earlier fish oils were used because of the icosapentaenoic acid and docosahexaenoic acid. But those people who are not comfortable with consuming non-vegetarian sources for these fatty acids, which have enormous medicinal potential, and they are very good for a normal person also from the point of view of health, like be it your memory, be it your heart health or your cardiovascular system, and in general, like the dietary fiber, the probiotic effect. So many advantages we'll be discussing shortly in the next slides. So ALA is the most active component present in flax seeds. This is alpha-linolenic acid. I need not mention about what are omega-3 and omega-6. I believe you have already studied that. But this is one of the richest source of ALA, which is an omega-3 fatty acid. Whether it is 3 or 6 will depend upon how the structure has been numbered, which system has been followed. But point of interest here is that they cannot be synthesized by the human body. That is why we have to take them from outside. Like you might be knowing, we have so many organs in our body, and liver is also a very important organ. Every organ has a function to perform, but not every species is comfortable in synthesizing all the components. So we need to seek help from some other source. That is why we consume a mixed diet and a balanced diet, of course. Like one example, I would surely want to expand. In the meantime, you can have a look at the composition. I won't be reading it out because of the lack of time. But yes, our diets have become such that the ratio between omega-3 and 6 has been so much disturbed that in those people who consume more of processed food or more of omega-6 fatty acids, it can be as worse as 1:40. Like 40 you are taking omega-6 and just 1 is omega-3. Whereas what WHO says that the ratio should be somewhere between, I'm talking about 3:6 ratio. Omega-3 is to be taken in very short quantities, and the ratio in the day's diet or the whole diet should be 1:4, or at the most it can be up to 1:10. But 1:40 is surely not a good ratio, and it can disturb your whole physiology. And please be careful about consuming how much amount is consumed, because excess of everything or excess of anything is bad. And every component, be it the nutrients, if they are in excess, they do have very bad effects on our body. So balance in anything in your life, that is very important, and it has to be taken very cautiously. So this is about the composition. Of course, it is going to be rich in calories because it has so much amount of fat. Total lipids are around 45%, and it has been considered an oil seed also, because oil is to be extracted and 45% has to be there. Generally 28 to 45% is a good amount of fat, and the fat can be extracted also. It can be consumed as such also. Very good source of many minerals, dietary fiber also. Then the quality of lipid, most of the lipids, more than 50%, about 55% of them are the alpha linolenic acid, which is one reason of making it special. About vitamin E, vitamin A, it's a good source. And calcium, magnesium, phosphorus, it is a very good source, but be cautious about how much amount you are consuming. Because yesterday also, when the experts were discussing about the iron deficiency in this area, I was just thinking whether flaxseed can be used as a source of iron for those women, because this has a good potential. But then you have to be careful about this thing, that you cannot exceed the amount. Generally 1 tablespoon, like 15 gram, that is sufficient, this which covers the whole population. We have to see the body weight also of the person. So in that case, you cannot give much, because when you are giving flaxseed as whole, or you are giving the oil in the purified form, a huge amount of these components like lignans, which have these estrogenic properties, they also get extracted along with that. Similarly, if you're giving it as a source of iron, the other components, they are going to come along. So if any of you who are planning to do research in this area, it's a good topic of research. Like if you're able to fractionate the components of flax seeds in such a manner that some of the fractions, they can be used for those populations which need iron, and the fatty portion and the lignan portion or the other fraction can be used for the other population, so that no part of the seed is wasted. Though this particular crop has an application, commercial application from textile industry point of view, because it's fiber, they are used in textile industry also, but we'll be concentrating ourselves on the food aspect only. So it's a functional food. And if the purified components, like you have the theme area nutraceuticals also, when it is being consumed as a whole, you call it functional food. And if the purified components, they are taken, they are more of a like medicine or a drug, and you call them nutraceuticals. It belongs to that category of foods also. So this SDG, which is a lignan, this is also the omega-3 fatty acids and the lignan. They are the ones which are being targeted and which are being exploited for various applications in the human physiology because of their useful or health benefits. So coming to this history, I won't go into that, but more than 50 countries are producing. And I'll be happy to share that we are amongst the top 5 countries who contribute to the global production of flaxseed. But there is a sad situation about this thing also, that farmers are not very comfortable with consuming it, though it is very environment friendly also. Like it can be grown before harvesting of the paddy crop in the waterlogged portion where there is a huge quantity of water. So it can be utilized from that water conservation point of view also, and it is an annual crop. Basically, it is a winter crop. But the thing is that the some support which is given to the major cash crops, that is not yet available to the farmers who go for flaxseed cultivation. But this can always happen if anybody from the administration, if anybody from the government side, and maybe in future this can be done, because I'm of the view that instead of growing from the farmer's point of view, you will get more earning when you are going into some like a progressive farmer can do that, something which is not very usual, the one which is different. So that can fetch you more money, but of course buyers have to be there. And luckily the urban population or the doctors or the Ayurvedic people, they have a lot of interest. So sale, I don't think it will be a problem for the farmers. But yes, those policies like the support prices, that is always required. That is always helpful for the farmers. And we basically are a country where agriculture is a very important sector. So I hope something happens in that area also. Canada is the largest producer. And if we come to the state-wise production in India, the contribution to the global is around 11%. It fluctuates from year to year. Maybe now it has increased or slight decrease is also possible. But in this area also, and in a number of states, like including Assam, West Bengal, Jharkhand, Orissa, they are also producing it. But Madhya Pradesh ranks higher in production. Like even our samples, they came from Madhya Pradesh, right, Dr. Priyanka? So this is not an unusual thing to happen in this area. It can always be grown, and most of the North India also, South India also, it can be grown easily. It's a very humble crop. Does not require much attention or much help. It can be easily grown. So awareness needs to be created in the population also. So these are the application, a lot of application, but you can just have a look at the water soluble emulsifying agents and thickeners. These are the additives which are purified from the flaxseed because of the soluble fiber content and the mucilaginous substance which is produced in it. It can be used as an emulsification action, as a thickener. A number of others are also being used, and this can be used in if anybody has interest in that area, like packaging materials, like edible coatings, or maybe films can be prepared. This has a research potential also. But so many products are available, but be careful about how you consume it because of the high amount of unsaturated fatty acids present into it. More is the unsaturation, you know that it is more liable to oxidation also. Either the packaging has to be such that no oxygen is available to spoil it, or the other thing is you can always consume, grind it or mill it fresh when it is to be consumed, or it can be packaged in such a manner that oxidation does not happen. So a number of products can be incorporated into a number of products. The traditional recipes or the modern baked products or any product like steamed or the shallow fried or roasted, it can be incorporated very nicely because of the taste, which is not very intense. It is a nutty taste, and it is very much acceptable. Personally, I like it, and I'm sure you will also like it. So it can be incorporated, but you need to calculate that per person recipe, how much amount will go into the body of that person. And gender wise also, you have to be careful about such crops which may contain estrogenic substances. Like if you ignore that part, it can happen that imbalances can occur not only in the pregnant and lactating mothers whose hormonal status is different from the normal women, but in males also it can happen. It can create imbalances. So please be careful about that. But yes, while we are using it as an additive, as a thickener, you will be getting double benefits of the crop also. Plus, you will not be using any other costlier alternatives for that additive. So composition we have already discussed. This point I would like to stress on. This active principle, the active lignin, that is SDG, it is converted in the colon of humans. The mammalians, we are mammals, and the other mammals also. It is converted into the metabolites, and these ones also have very good properties in the gut. I'm not sure how many of you are aware of the concept of, because most of you are the younger population. Those from biological or food sciences, you must have heard about the gut motility and the gastric transit time and the satiety value of the food because of the presence of these fiber. Two types of fibers, either soluble or the insoluble type. Both are present. More is insoluble, but more than 10% is soluble type also. They have different roles to perform in our guts. Whenever a food goes into our gut, and after how much time you feel hungry. Suppose had we been given a flaxseed based meal in the morning, we'll feel hungrier later. Maybe during lunch, we don't feel like having more because it has a satiety value. And when it enters into the body, both of the fibers, like one is having the satiety value because it will stay in your stomach for longer period of time. it moves slowly into the intestinal tract. The other part, which is the insoluble part, of course, that is going to protect you in other ways, also helping the probiotic bacteria to grow and multiply in your gut, and which are very important. Let me tell you, you have to be very careful about what kind of flora you are carrying in your body. They are very useful. They can help you also, and they can make you sick also. So be careful what you eat. So this can be a good thing to consume daily on in the morning, preferably along with the breakfast, or if otherwise also it can be consumed. It is going to give you good results. One more thing I want to stress about this is regarding the proteins, the quality of proteins which is present in flaxseed. Of course, you won't be taking much amount, but it will help in balancing the other proteins which you are consuming, because it is going well with cereals as well as pulses, and mutually it will be supplementing the amino acid makeup of those proteins, so it can be helpful. And the amount of globulins and albumins, basically 4 fractions are there according to the solubility classification, and these are the ones which have a better amino acid makeup in comparison to prolamins and glutenins. So these are present in more quantities. Otherwise, in other crops like rice, wheat, you have more of those types of proteins than even in pulses. But this has a good profile from protein quality point of view also. This has already been explained, but it has been reported to have antifungal properties also, and reducing the chances of having tumor in our body. They have all of them, they have a mechanism in the body, how it works. But of course, that's going to take a lot of time. But yes, we can discuss it that way, that insoluble fibers are also useful, and your soluble ones up to 7 to 10%, they are also useful from the point of view which we just discussed. Coming to the non-nutrients, yes, these are the ones which have the nutraceutical properties. Cyanogenic glycosides, they are also present. Instead of calling them, earlier we used to call them anti-nutrients, but the concept of calling anything anti-nutrient is not considered good, because nothing in the nature can be anti. If it has been produced by the nature, maybe it has one role or the other role to perform. So now people are shifting from calling it as anti-nutrient to non-nutrients, because maybe in future they can be our life savers. Cyanogenic glycosides, they can, of course, they can contribute to problems like goiter, because of the chelating action. They have a tendency to bind the mineral elements. So iodine deficiency can be created, but you're not going to consume it raw. That's the best part. It has to be processed, and processing eliminates most of these non-nutrients. So there's no need to discuss each of them individually. But yes, they are heat labile, or some of them like phytic acid. This also binds minerals. It is a chelator like calcium, zinc, especially the divalent cations. But if you soak it, if you sprout it and germinate it, then the enzyme phytase that takes care of all the this, and this phosphorus is made available because of these processing treatments. The point here is that whatever non-nutrients are present, they can be easily taken care of by the processing treatments. And this is what we have done in our project, which was completed by one of my scholar, that various processing treatments, they can easily eliminate these antagonists and non-nutrients. So one by one, we can come to alpha-linolenic acid. These things, they are considered good from heart health point of view also, because of the ratio of the fatty acids which are present. This is the structure if you just want to have a look. 18 carbon fatty acid with the double bonds at 3rd, 6th and 9th carbon atom. This is what is special about that. And the number of enzymes like saturases and elongases which occur in the body, they are contributing to increase in the chain of fatty acid or creating the double bond for unsaturation. But if we take this flaxseed, we can start with some intermediary product, and further processing is possible in human body. That is why we have to take them in small quantity. Rest our body is capable of doing that. Human body and any other body, we are wonderful survivors. We have so many defenses in our body, and we have so much potential to save our body, because nature has made us such that we try to survive. So if we are careful and if we go for the signals which our body gives, we can always survive and have a good life. So this is about how the conversion happens in the body. I need not go into details, but it is available easily if you want to study that. Lignans are the ones which have phytoestrogenic properties. They behave like the female hormones, estrogens. So they are present in other crops also, but this is a richer source from that point of view. How it is metabolized in the body, how the demethylation, dehydroxylation in the body, in the human bodies, they convert the flax lignans into mammalian lignans, and they help us. Like these microbes in our body, they can be of great help to us from this conversion point of view also. So these microbes, they help in the demethylation process and the changes from one form to other, and to scavenge the hydroxy radicals also. This point may interest you, because nowadays what is happening is every food is being told as this has antioxidant properties, and many of the foods, they are being labeled as this is anti-carcinogenic. It has become so popular, calling every food ki isme ye carcinogenic, cancer nahi hoga, but it is not as easy as it is said so commonly. So, but research studies, they have demonstrated that this lignan, this takes care of these hydroxy radicals which are present in the body. And not only in the body, these oxidation happens in the food outside the body also. And the same things, free radicals are produced in our body also, because we are surviving because of the oxidation. Oxidation has to happen. That is why we are getting energy for various metabolic processes, for the BMR also. So they are very potent antioxidant after they enter into the human system, and otherwise also when they are used in the food, they have antioxidant properties, and the shelf life of the products can be elevated, it can be enhanced. Such kinds of treatments, they were given in our project, like germination, roasting, steaming. Roasting is when the dry heat is applied. Steaming, of course, you understand steaming, autoclaving, soaking, and combination of these treatments, they were done. And defatting is also one of the treatment. This is actually separating the components, because when you defat it, the fat will take along all the fat soluble components, and the other components will be staying with the portion out of which it has been defatted. So, and both of them, they have different applications, so that it can have wide application, it can be utilized. So according to our studies, there was a reduction in most of the non-nutrients, like the polyphenolic content also. And the amount which is required, that is sufficient to take care of our human need of antioxidants. And our body also has the natural antioxidant mechanism also. Then coming to how much amount can be supplemented, like up to 10% level was fine according to our studies, but it will depend upon what product you are manufacturing. Maybe if you're manufacturing a spread type of product, you may have to consume more than that. But the point is, per serving, how much is available, that you have to take care, and you have to be you plan accordingly. But it can be well incorporated, like sensory properties point of view, where you go for finding out the taste, the smell, the texture, all of them, they give good results. So like products like idli, chutney, porridge, they were tried, and it can be, of course, incorporated into baked products and a number of other products also. These are some of the images. And the digestibility, of course, it improves with the treatments which we give. Steaming is a very effective treatment with less harms. Steaming and boiling along with the same water which is being used for boiling. That is one way. But because here we'll be taking in ground form in idli preparations, so that way it can be taken as such, and then it can be steamed. Texture was also found to be very good. The sensory properties were really good. About the recommendations, which we have already discussed, that if you're taking it as a whole, not in raw form, but as a whole in roasted form, up to 15 grams can be consumed. But if you are taking the oil form, because it concentrates the lignins and other components which are fat soluble, it should not be consumed more than 7 grams, because 1 tablespoon of oil will provide you 7 grams of ALA, because it has more than 50% alpha-linolenic acid. And 1 tablespoon, usually a standard tablespoon is considered to be of 15 ml. So around 7 ml is sufficient, not more than that. So safe doses for the purified form of ALA is 3 grams per day, and this covers all the population. But age-wise, it has to be taken differently. Oil forms and these powdered form are unstable, but yes, we can take care of this spoilage or the degradation because of the what you call it, the packaging material which you are using. It can be made vacuum packaged. So intakes we have already discussed. Cautious. We have to be very cautious if you are giving it to pregnant and lactating women. So doctor's prescription is recommended for that case. But what overdose can do is like breathlessness, rapid breathing, weakness, difficult walking, and paralysis and seizures like dora padna. This can also happen, but this usually does not happen. If you want, even then you cannot consume very huge quantity of these things, because it gives you that kind of satisfaction. It swells a lot. It has those properties that which give satiety, so you won't be consuming more amount naturally also. So be careful and respect your food. Don't waste food. And I'll like to have the queries also if you have any. Thank you. It has a good potential. Still, so much work can be done in this area. We have done a small portion. Much work can be done. Blending of the oils from flaxseed, that is one research area. Like instead of using one type of oil, because we have to be careful about the ratio of omega-3 and 6 which we are consuming daily in our homes. अगर हम सिर्फ mustard oil is good from that point of view, but some oils, they have a huge quantity of omega-6 and very less quantity of omega-3. So blending is one concept which is happening also. Some of the companies, they are blending, but you need to calculate and formulate how much amount of which type of oil should be blended so that the total ratio comes about to be the one which is best for you. And one more thing is important from flaxseed oil consumption point of view. If you are blending it with some oil, you need to recommend the usage also, because when you are heating it, you will be destroying the medicinal properties and the nutraceutical properties. In that case, you can grade your oil into different fractions, and you can suggest that which one can be used for deep frying and which one is good for consuming as raw, as a salad dressing, or in some of the traditional preparations also, like litti chokha, you must have heard about. This mustard seed oil, it is blended in the end. Salad dressings, they also have raw oil added into that. People use olive oil, so that is good, and that can also be done. In that case, the raw one is okay. And one more thing, when you store the oils in your homes, you take care of the headspace which is left in the bottle. Gradually, what happens is you will be consuming oil, and the space is keeping on increasing. Every time you open the bottle, oxygen will enter the bottle. So you have to be careful about that headspace area also, so that it can be protected. Thank you. No questions?
We were talking about this conversion. The
conversion in the gut?
Conversion in the gut.
Okay.
So is there any study done on when we consider it as oil? I'm not talking about individual component when it is applied in vitro. I'm talking about in vivo studies in gut. Is there any study done?
We have mentioned the reference in that also. Let me check whether it was the gut research was on the oil or it was on the bone flaxseed. I believe that was on the bone flaxseed. Let me check it once. May I go to the previous slide? Priyanka, if you remember, you that case. So oil will not be producing any effect because effect is coming because of the lignin and it will not be having any influence. So in that, but studies might be there, maybe not in my knowledge, but we completed this project I think 5-7 years back. 16. But yes, other students are also doing work on this crop, but not on this aspect like the gut health. But this has potential. Because microbiological studies doing that and that too in vivo is not an easy job. Because getting the sample, getting the subjects who are ready. And for animal studies also we need to take so many permissions and because animal rights are also rights. So, thank you.
Thank you so much, ma'am, for opening the treasure of knowledge and showing us a new direction in the field of food technology. Till now, we have come across the two informative lectures. Now, I request Dr. Siddharth sir to take over the session.
Thank you. Thank you all for joining this second day session. We are sincerely apologizing. We are making some changes in the lecture schedule. Like we are supposed to have a lecture on bioprospecting through chromatographic techniques that we are not going to have here. We are going to have that in Guwahati Biotech Park. So those who have registered for that bioprospecting workshop, that's a separate event will be going on simultaneously. They can go for the high tea and contact our registration desk. They will arrange for the transport to GBP. There, that entire session will be carried out. And the rest of the participants, they can stay back here. After the high tea, we will resume with the oral presentation session, where again we will be hearing some of the very interesting, research highlights we are supposed to, experience. And in the post-lunch session, we again have a very specific session on herbal products, particularly herbal nutraceuticals or herbal food supplements. There we are trying to cover the entire aspect of lab to field transition. So people who are from industry, from startups, they would probably get a lot more insight that how we change lab discovery to an actual commercial product. And eventually we will shift to a valedictory session where a lot of way forward we want to have, where all of you probably take some part. So thank you all again. Please proceed for the high tea. And as I said, now we will be breaking for two different sessions. One is for this bioprospecting workshop, and the other one is this research presentation will be happening here. So after 15 minutes, we will come back here again, those who have not registered for bioprospecting workshop. Thank you. Thank you, everyone.
Among them, 49,000 plant species are there, which are claimed to be medicinally useful. And among them, 1,700 species are there that are available to our northeastern India. Among them, 740 plants have been described in Charak Samhita and Sushruta Samhita of Ayurveda that are claiming to be having Arbut Nasa. Arbut is cancer in Ayurveda. So I have selected some of the products as because natural products have played a significant role in the discovery of cancer drugs over the years. As you know, you can see these two drugs, these four drugs. The first drug in front of you, it is known to you or Nayantara. That is Catharsis roseus. The second is Campotacin. The third one is Taxus baccata. Some of you will be from Meghalaya. This is the plant taken from Meghalaya, Taxus baccata. And the fourth one is Campotacin. So from this, Vincristine, Brainstorming are products that have been extracted from these plants and now being used widely in plant research law. And to say happily that these Vincristine and Brainstorming are not synthesizable in lab. Even if it is synthesized, they are not going to work in that manner when it's extracted from plants. So my materials and methods I am going to study in vivo, that is in experimental animals, that is white oyster rats. The fifth one is evaluation of cytotoxic, that is cytotoxicity study or toxicity study in vitro, that is SRB diagrams. So one by one, if I come to this is my drug. This is a medium-sized tree. You can see this is Muchukunda in Ayurveda and Pterospharum acerifolia belonging to Sarcoidaceae family, and it is the flowers where importance of my use. The part use was flower, and for this collection after collection, I have authenticated the plant in the Department of Botany, Guwahati University, and herbarium samples were submitted with accession numbers 1722. And then it was re-validated by the Central Research Facility of KLE University, that is KLE Bangalore, nearby NMC. And the samples were deposited for future references so that it is reproducible. And during my pharmacognostic studies, I found that some of the trichomshare vascular vessels, etc., jelly vessels, as usual, were present. But in powder microscopy, I found some crystals. And this picture, this one is sending some tracheids, presence of some tracheids. Here, these are some tracheids, and this is a dry group. And here, powder microscopy, it is showing some crystal structures. Now, this is the preparation of the sample. First, when the drugs, then I went for soft cell extraction. That is a type of hot extraction. And that was done in ISST Boragam. Now, the second picture, it is showing the preparation of the extract solvent. That is 70 to 30% of ethanol and water. That is called hydroalcoholic acid. Then it is the Kimball preparation where you put the sample inside. And lastly, the soft cell extraction. It is successive extraction. I went from the non-polar, that is like acetone, benzene, hexane, to the high polar like butanol, ethanol, methanol, and water. So that I get all the materials whatever available, maybe high polar or maybe low polar in my solvent system. Now the first for standardization, here you can see it is fluorescence analysis. So that the drug can be standardized. I mix the drug with so many solvents and studied it under 254 and 360 nanometer. That is short UV and long UV. To just record. Here you can see there are some extracts formed, starting from the hexane, benzene, acetone to the ethanol, methanol, and water, from low polar to high polar. It is the end product of the extracts. Now coming to this is the study that is showing, that is a phytochemical study. There you can see a benzene ring, that steroid, the presence of steroid. The ring of brown color shows the presence of steroid. Here in the second, it is saponin test. Presence of saponins. These are nothing but flavonoids. As you know, phenolic contents and flavonoids are the points of importance in plants. Why we are looking towards plants is the presence of phenolic contents and flavonoids, especially the quercetin and luteolin. So coming to the third one, this is hemolysis. I use my own blood in this study so that I want to know whether the drug, if I consume it, whether it will do hemolysis or not. But to say happily, it didn't do any kind of hemolysis. So it is simple for my body. Now coming to the solvent extraction process, where I got maximum extracted solvent, extracted value in water, and next is ethanol. That means the compounds, bioactive compounds present in my drug were highly polar in nature. Coming to next, You can see the qualitative analysis with different extracts. Aqueous and ethanol I took. Phenolic compounds were present. Steroids were also present, as I have shown. Flavonoids were also present in this. This shows the presence of flavonoids, phenolic compounds, and steroids. Coming to the HPTLC and TLC of it, when doing TLC, I use the solvent system like ethyl acetate, which chloroform and methanol. Ethyl acetate, chloroform, and methanol in a ratio of 70, 20, and 10. Then I got the RF values. 2 spots were derived in TLC and 7 spots were derived in HPTLC. Somebody came from NPROM, as I know, in this seminar. NPROM, I used that software. NPROM and Chemec. I got RF values to 6 to 7. So it was showing that 6 to 7 important bioactive chemicals were present. Coming to this UV spectrophotometry, what I did is I used a sample of hydroalcoholic extract in that spectrophotometer. I got a pick in 546 nanometer. 546 nanometer, as the drug library suggests, it is quercetin. The presence of quercetin in the sample is confirmed. Next, the FDR spectra. The FDR spectra shows some peaks of 3689 and 2932. These two are important. The first one is isonitrile group. The second one is alkyl group. Next, I have come to the NMR analysis, that is carbon 13 NMR. I didn't perform the H1 NMR, it is carbon 13 NMR. And it has also shown the same presence of 4.1 and 4.07, that is isonitrile and alkyl group. Now, coming to the AAS, that is atomic absorption spectra. That is done because we are facing so many problems that herbal drugs are having heavy metals and all like that. The United States of America, they are resulting the samples like that. So I have done this study to show that the lead and nickel content of my drug is below the permissible level of parts per million as per FDA and Ayurvedic pharmacology as per drug uptake also. So it is usable and it is not having heavy metals in my case. Next, coming to the acute toxicity study. Toxicity study, everybody is telling that it should not be talking to me. So there's this acute toxicity study. I use the OECD 420 limit test guideline. OECD 420 limit test guideline. What I did is I took 10 female white albino rats weighing 100 to 150 grams and then divided into 2 groups. To the first group, what I did is I used the decoction of my terospermum acidicoleum. The decoction is nothing but in Ayurveda, it is called quartz. Quartz. How to prepare it? You just take the amount of powder, just add 8 times of water, boil it, reduce it to 1/4. That is the part or decoction. It is decoction. And the other part, what I did is hydroalcoholic extract of terospermum acidicoleum. I got the 2 samples and I used the both. To one group, the quartz or decoction, to other group, the extract in the dose of 5,000 milligram per kg. 5,000 milligram is higher than the 50 times higher than the usual oral dose. That is called limit test, LD50 study. Why I use this? If I found any kind of toxicity in these cases, then that process has to be rejected. Now, what I found, the animals, when it was fed to both the groups, we have seen the motility or any kind of toxicity developing in the animals. But surprisingly, after 14 days, there was no motility, there was no signs of toxicity, there was no toxicity at all. All were living. So I can say, if I consume my drug 50 times more than the oral dose, I can be safe. So this shows that the drug is safe to humans. That's why the oral toxicity, acute oral toxicity was done. Next, before doing any test with the animals, what we have to do is we have to take some permissions from the ethical committee. That is called Institutional Animal Ethical Committee. I got 2 permissions. The first one is from Cali University for acute toxicity, and the rest, the main care group of institutions, that is NIPA, under the Murthy side. I think you were present yesterday. I also. So this is the second one. I got the permissions to do. Now, coming to the second test, acute inflammation. That is inflammation of short duration. What I did, I took 24 animals and divided into 4 groups. Each group containing 6 animals. And the groups were named like control, standard, test 1 and test 2. The drug one, the control was distilled water. The second is standard drug, indomethacin. This is a quite good anti-inflammatory agent. To compare that, I used indomethacin. The third one is the decoction of the my drug, and the fourth one is hydroalcoholic extract of the drug. The dose is as per the classics. 20 ml, 2.25, that is as per the drug update. 4.32, it is calculated as per Pagatin-Barnes table, 1969. Pagatin-Barnes table, 1969. What is that? Our body surface area multiplied by the dose into 0.18. That is the body surface area of a rat. And accordingly, we have divided the dose, and I got 4.22 per ml per kg. Now coming to the 500 mg per kg of dose of the hydroalcoholic extract, because 5,000 I have used and there was no toxicity, I divided into 1 by 10. 10th part of the 5,000 is 500 milligrams. So I have decided the dose for oral test. It is 500 milligram per kg. Now the first slide showing the grouping of the animals. The yellow marks you can see, that is the picric acid, so that we can mark the animals. Which one the first, which is the second. Picric acid is given to the head, to the tail, to the back, to the right foot, to the leg foot, like that. Now coming to this is oral gavage. That is, we put the drug through a gavage catheter inside the animal. The third one is okay. So I have so many first. Anyway, the third is the carrageenan solution to induce the power edema, and the fourth and fifth is the calculation of the edema volume, so that it is showing some activity or no. Now coming to the power edema, we can see my drug is showing the activity from the 3rd hour onwards. Next, coming to the brass rod melanoma, that is chronic inflammation. What I did is, this is the weighing, grouping. I grouped into 3. This is brass rods. What we did is, we anesthetized the rats and then inserted with an incision to the border region of the flat. And after it, suture. Again, after 14 days, we collected the blood, sacrificed the animal, and got the pellets like this. We waited the weight and dry weights of the samples. Now coming, this is the histopathology I took, the kidneys, liver, pancreas, and brain, and the sectioning was done, and accordingly it is studied. The histopathology of the liver, histopathology of the kidney, histopathology of the pancreas, and histopathology of the brain shows no distinguishing characters between the control and the test. That is, there is no toxicity to the brain, not to the pancreas, not to the liver, or to the kidney. Now coming, after knowing that this is my drug is working, so I studied about the anti-cancer study. That is SRB, sorbohydrin B assay. Now what we did is, we took the sample, some cell lines, that is MCF7 is breast cancer, HeLa it is cervix cancer, and then lung cancer, and then prostate cancer. We took the cell lines from NCCS, that is Pune, National Center for Cell Service. So from Pune we got it, and it was kept in the minus 60 degree nitrogen cylinder. Then we collected from the cylinder, and what we do is, we put the medium. That is, medium was RPMI, which glutamine and penicillin and streptomycin. Then we seeded the plants. Seeded the plant means we extracted the cells to one different chamber. And after putting the cells into 96 well plate, that is a standard process. You have to put some plates. You can see the photographs at least. Yeah. So seeding the plants, and this is the 96 well tube where we put the sample. We put the culture media, and then we put our test drug. Test drug in certain concentrations, 10 milligram per ml, 30 milligram per ml, 50 milligram per ml, 70 milligram per ml, and 100 milligram per ml. Then we study it under ELISA. ELISA test is done for optic density. So how it is collected? Here you can see the methanol extract has shown PC, that is prostate cancer cells, 78% reduction. 65 and 68% with concentration of 70 and 100 with ethanol. Ethanol also has seen some effects. And then coming to the next, it is aqueous extract has shown 60% reduction with 70 microgram concentration per ml and 65% reduction with 100. The standard used was 5-fluorouracil to consider these cases to be significant. So coming to the discussion point of view, it is well known that our edema or inflammation is practiced. In the first place, histamine, serotonin will be released, so people will be sneezing like anything. And in the second phase, prostaglandins will be active. As because my drug was acting and after the second hour, so I can say that my drug, our promising drug is acting over the prostaglandins and serotonins. Now coming to the cytotoxic effect, to conclude it, my drug is very good anti-inflammatory agent, and my drug is a very good novel agent for prostate cancer. And to know. These are some journals we have published. As the journals we have published, and as I can say that the prostate cancer activity, anti-prostate cancer activity of any drug is known to be the propaganda. That is known to be the anti-cancer activity of any drug. So I can conclude that my drug is anti-cancerous. It is having good and potent anti-cancer activity, so it can be tried in clinical practice. Thank you so much. Thank you.
Very good work, sir. Thank you.
Thank you, sir.
Now I'm interested about the cost and all these issues.
Yeah, cost and all.
Yes. Prepare, I mean, even the laboratory scale also. How much will be the approximate cost of your thing? I'm not saying, large scale, small scale.
Sir, I'd be happy to say that the IIM Jammu, Triple IIM Jammu, Dr. A.K. Saxena sir is doing it free. If you approach him, we have certain letter and MOU has to be done with the two institutes.
No. I'm not talking about that, sir.
Okay.
That's okay. He's doing it
That's
free.
fine. Okay.
But in general, if you want it to be widely applied, you cannot say that, we are doing charity. It's not possible, right? There is a minimum input cost and all that. So approximately, you have any idea?
And I can say if the setup is already in full length or in its own capacity, then it will be costing around 2 to 3 lakhs rupees. Our experiment. If
No, I'm not taking experiment. I'm taking your pill. Ultimately, you are saying drug.
my drug, sir, if it is given in 500 milligram per capsule, then it will be costing only 150 rupees.
Okay,
150 rupees for 30 capsules.
okay, 5 rupees per capsule. Fine.
500 milligram of hydroalcoholic extract of terrestrial mesophilium will cost you 150 rupees. It is so less. Because it is available in Khasi Hills. You can extract it. The extraction cost will be.
I agree with sir. Actually, sir, what sir was asking about, what in your experimental setup also, there must have been some cost.
Yeah.
So what is the actual cost that per capsule or per serving of that your drug is going to have?
Including your experimental industry.
Yeah, everything.
It will be difficult.
Overall, your experiment costed you 10 lakhs?
Maybe, sir, I don't know. That's because I am from a government sector, working in a government college, and I was consultant.
And how many of these rats were killed in due course of
It
bleaching?
was around 78. I got segregation around for 6. It was 78 in total. Because
The rest all are living fine now.
no, sir. After 1 month, I have reused it. There's a refreshment period. After toxicity days, there is a refreshment period for 28 days. After that, you can reuse it. If it is not having any kind of toxicity or anything else. It is moving freely and all.
That is fine. After 28 days, they're moving.
That is, it can be used for next experiment also, a different experiment.
That's okay, but what is the status right now?
Now, we are going to, and I think I have claimed the Form D. Form D is one form where you can claim the animal
Can
life.
you do any of these experiments without killing them?
If I read about that histopathology part, I can say it. If I read the histopathology part,
There are no advanced techniques like tracer techniques or anything for the histopathology kind of. You have to kill. Just like, human being, and they also have life.
yes, sir. I got the permission to kill 6 animals. All good. 1 animal is good. Freedom, sir, allowed me. Freedom. So I killed 6 animals and 2 died. That 2 died, I reported in Form D to Menaka Gandhi. Is that good?
Okay.
No questions. First, congratulate for your good work. 2 questions are, as Sir was also rightly pointing out, supposing if we have carried out the research and we have got the observations. Now, with this observation and these testings, whether your drugs or your formulation can be, first of all, can be administered to the public or the people as a drug, number 1. If it is so, then you tell how it is. And for that, whether you need any IPR or other things. Please, why I am saying all this, because there are a lot of
B2B kind of things are there, a lot of platforms are there, with CCAP and all, you may be knowing or you would have approach also, I do not know. So whether that kind of a high level, because industry has to be there when you are talking about the supply, whether it is Ayurveda related or something, so those efforts have been done or not. If it is so, then please share. And when you are saying about the cost, vis-a-vis, you should say the current practices cost also. Now, when you are talking that 150 rupees, you buy your formulations. That means the duration, how long person has to take, and overall cost, if you can give us the picture, that would be helpful. Thank you.
Okay, so I'll be happy to say that up to the in vitro SRTSA, after that, in vitro, that was done in the lab, that doesn't need information. That was done in IITG.
Those are today, I don't come to direct point.
Okay,
Human applications, human
Whatever done, let's see.
Human in vivo has to be done.
Yeah, madam. In vivo means clinical change each stage of life. First stage, we 100 people, second stage, 500 people, third stage, 5,000 people, and the last one is.
Correct.
Well,
Phase 4, the phase
phase
1
one.
and phase 2.
So, those persons are there, that has not been taken care. That is all that. उसके लिए मैडम ह्यूमन एथिकल कॉम्पिटिशन का काउंसिल लेना पड़ता है. That is institutional animal committee that I call the council.
ये सब हो गया. I am also from this line only. Okay. So I understand all this. Now you tell me your good work. To escalate very fast. Time is nothing short of time. For all these things to reach out to the people. Let us know what you need to be done and how can we expedite. That is my another question. Because otherwise, क्या होता है हम इधर तक आके पेपर पब्लिश करके कुछ stop.
For some time.
And somebody else will take up your other things and then sell it to us. See, why I am saying, because I come from Sustainable Advisors Office and there we can, we are having, our government is also doing a lot of practicing skills. We have IP, we have other protection things also. So such things, if you want to expedite, we can just take there's no advice to take. So don't restrict yourself to this dermatology.
That you will have, I will go for the blessings.
Blessing is okay, but action is required. Madam is saying action is required.
Yeah.
हमें डायरेक्शन में करना है एंड फोकस में करना है.
अब 99% आपका काम हो गया, अभी 1% आप नहीं करेंगे तो ये रुक जाएगा. That's what मैडम is.
ये आप बता दीजिए क्या कर सकते हैं.
हो जाएगा. But ह्यूमन का क्यों होगा?
आपको सीएम के बारे में पता है. तो ये क्या
क्योंकि उसके बाद उसको लेके यहाँ लगाना पड़ेगा नहीं तो रिकॉर्डिंग.
Whether any possibility for connecting it or this kind of innovation, and always introduction pass it on to the application, where the innovations and all of this guidance. Because unless some good person is involved, it will be already other things.
I'll do that.
Thank you.
Thank you so much, Mr. निकालिए तो.
हेलो 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 हेलो.
Thank you so much, sir, for this fruitful information. Moving toward our next presentation, we have Mr. Lakshadip Nath, who is Assistant Professor of NIPS, and his topic includes a comparative study of leaves. I welcome you, sir, on the dais.
Can I see pointer? Pointer points. What is it? I can just go trying to.
Time is only 12 minutes for each presentation. It's monitored strictly.
Okay, thank you. A very good afternoon to the panelist member, Dr. Ram Gopal, Professor Ram Gopal sir, and Dr. Hemsudi sir. And I would like to thank School of Agro and Rural Technology for giving me the opportunity to present my oral presentation. Myself, Mr. Lakshajit Nath, working as an assistant professor at National Institute of Pharmaceutical Science, would like to present my study topic, that's a comparative study of the protective effect of the leaves of Murraya coengi and Centella asiatica in experimentally induced inflammatory bowel disease in animals. So actually this study is a comparative study between two commonly available plants in Assam. So these are the contents: introduction, conventional therapy that is used for the treatment of inflammatory bowel disease, alternative therapy that is being available, and objective materials and method, results, discussion, and last one is a reference. So let's come to the just introduction part. What is inflammatory bowel disease? Inflammatory bowel disease is defined as the group of disorders that causes inflammation in the intestine. It is one of the most a common disease that is prevalent in India, and around 3% of the population generally suffers from this disease. This disease mainly includes your Crohn's disease and ulcerative colitis, where the inflammation of the intestinal or the digestive tract or the lining of the digestive tract generally takes place here. But till now, the exact cause of this inflammatory bowel disease is unknown. But it is presumed that due to the depression of the immune system in the digestive system, this disease generally occurs. Okay, this immune system is mainly responsible for killing the viruses and bacteria that is present in the intestine, in the intestinal region. But when this IBD disease occurs, what happens here? The defensive mechanism, the immune system that is present in our digestive system, they are unable to work, and as a result, it causes your inflammation. So this is just a little introduction about the IBD. There are general normal therapies that are being generally available here. There are different drug therapy that is being used for the treatment of inflammatory bowel disease, such as your sulfasalazine, glucocorticoids, and thioguanins, and also like your methotrexate, cyclosporin, and also different biological agents. These are all some common allopathic medicines, but these all medicines have some serious side effect. Along with the treatment, they can cause different side effects, like your sulfasalazine, they are being reported to cause fatigue, nausea, abdominal pain, and also diarrhea. And along with it, the glucocorticoids, it can cause your peptic ulcers, upper gastrointestinal bleedings, and thioguanins, which is a methotrexate, which is a chemotherapeutic agent, they are also believed to cause gastrointestinal complaints, and along with it, hepatotoxicity and others. Like that, the other drugs like methotrexate and cyclosporin have also some serious side effects, and including different biological agents like infliximab, adalimumab. They have a very potent they're being used as a potent drug therapy for the treatment of IBD, but they are also reported to causes malignancies and also reported to causes opportunistic infection in the patients. So my area of interest is become forwarded to the alternative therapy that may be available for the treatment of this inflammatory bowel disease. So for this, I have selected these two, also known as your boar manimuni. These are the two common available plants in our household. And we have seen that in the last 2 days I have also gained a lot of information regarding the shifting of our current treatment therapy towards the Ayurvedic. So I have given Susan this topic. So these two plants, Murraya coengi, which is also known as curry leaf, is belongs to the family Rutaceae. It's a deciduous aromatic shrub with a strong smell, pungent with a strong smell, and it grows up to 3 to 5 meter tall, with a trunk of around 40 centimeter in diameter. And this boar manimuni, it's a very small creeper plant which grows in the places having altitude of around 1800 meter. So my objective of our work is to do a comparative study between these two plants, this Centella asiatica and Murraya coengi, to see the better efficacy in the treatment of your inflammatory bowel disease. So for this, I have collected these plants that are being available locally, and I have just collected it and dried it in a sunset dry and powdered it separately. These two plants are after being powdered, they are being extracted with the help of hydroalcoholic extraction with the help of using Soxhlet apparatus. And after that, the extract is being cooled up dried and concentrated, and they are being stored up. These extracts are being dissolved in the saline solution while administering in the animals. After this, I have not forwarded for the OECD experiment. I have not gone for the acute toxicity study because this plant can be generally regarded as safe plant, ZRS. These two plants can fall under the ZRS category, so I have not gone for the OECD guidelines because these two plants are commonly taken in our kitchens. So for this experiment, I have selected Swiss albino strain rats for this experiment, who weighs around 210 to 250 grams, of age around 5 to 8 weeks of either sex, and total number 45 numbers of animals are being taken and purchased from the certified vendors, and they are being housed in our animal house facility only at an ambient temperature of around 25 degree plus minus 2 degrees Celsius with continuous supply of the food and water. Then after the acclimatization of the animals for around 15 days, they are being divided for grouping. So the groupings is being done into 5 groups. First group, second group, third group, and fourth group, and fifth group. The fourth group and fifth group is being again further subdivided into 3 more groups: 4A, 4B, 4C, and 5A, 5B, and 5C. Then after that grouping of the animals, they are being caged separately, and after that we induce ulcerative colitis in all the groups of the animals. The ulcerative colitis was being induced by the help of using 4% acetic acid solution, and this 4% acetic acid solution is being given intrarectally. While giving this drug intrarectally, we have administered anesthesia, and before inducing the ulcerative colitis in the animal, we have kept the animal in a fasting condition so that the food components doesn't affect the acetic acid to induce the ulcerative colitis. We have given anesthesia to the animals so that the animals don't give out the acetic acid. This is the grouping of the animals. They have been divided into 5 groups: group 1, 2, 3, 4A, 4B, 4C, and 5A, 5B, and 5C. The group 1 is being considered as the control group. We have not induced ulcerative colitis in this animal. Group 2 is being induced ulcerative colitis, and they are being given treatment with only normal saline solution. Group 3, we have induced ulcerative colitis, and we have treated this group with the standard marketed drug, that is a sulfasalazine. We have considered sulfasalazine as a standard drug. Group 4, group 4A, 4B, and 4C are being treated with different concentrations of Centella asiatica at a concentration of 50, 100, and 200 mg per kg. Like that, similarly, group 5A, 5B, and 5C are being again given with the treatment of 3 different doses of 50, 100, and 200 mg per kg of your Murraya Koenge. This is the grouping of the animals. After that, after the ulcerative colitis is being induced in the animals, and after the treatment is also completed, we have assessed the first parameter for our inflammatory bowel disease. The first parameter is weight to length ratio of the colon. So for this purpose, the rats were euthanized on the day 8 by cervical dislocation with the help of using your anesthesia, pentobarbital. They are being dissected down, and their colon part is being taken out longitudinally, and after that, whatever the contents that are present in the colon part, they are being washed out under normal running tap water, and their weight to length ratio is being calculated. This is the first parameter. Second parameter is being done to assess the microscopic assessment of the colonic damage. After the colon were being taken out, they are being fixed in a 10% formaldehyde solution so that they don't get destroyed out, and they're being fixed up in a liquid paraffin block and are being cutted out by the help of a microtome, and they're being again stained up with the help of using hematoxylin and eosin, and they're being observed for the microscopical changes. Different microscopical changes like your necrosis depth, extent of necrosis, degree of inflammation, and extent of inflammation, and also the fibrosis parameters are being checked out for the changes in this different microscopic in these different tissues of the colon. The third parameter is the enzyme content estimation. That this parameter we have assessed the superoxide dismutase parameter. This is an antioxidant enzyme. This antioxidant enzyme is responsible for scavenging the free radical activity. So this activity was being again done on the colon. The colon part using ice cold trice HCl buffer with the help of your grinder, and the homogenate is being taken out and they are being assessed for the checking the parameter of your superoxide dismutase. This is the step for checking the parameter for performing the experiment of the superoxide dismutase, and after that the absorbance is being checked out. This is the calculation for checking the absorbance of the test sample, absorbance of the blank divided by the absorbance of test into 100. This parameter is being checked out, and the SOD parameter that is responsible for scavenging the free radical is being taken out, and all these three parameters are being taken togetherly to check the better efficacy of these two plants. This is the results of my experiment. First result, it denotes in the graph. So from this parameter, we can just assess the changes in the colon length ratio. So these are the groups that I have already described earlier. These are the 5 groups of animals. From this 5 group of animals, we can see that this group 2, this is the control colitis. It induces the ulcer, and that ulcer is being treated with the help of normal saline water. So you can just see the value is too high, that is around 0.8. The third group is being treated with the standard drug, that is a sulfasalazine. And fourth group and fifth group, you can just see the treatment that is being done with the help of the 2 different drugs. This is the colon length ratio. The second one is the histopathological scores. I've already told you that the histopathological parameters such as your necrosis, inflammation, and fibrosis parameters are being checked out, and their parameters are being plotted in the graph. The third parameter is the SOD, the percentage inhibition parameter, is being also plotted in the graph, and from these 3 parameters, we can conclude that the administration that these 3 parameters, that is your colon length ratio, your histopathological changes, and also your SOD parameter, there are different changes is being observed. The first parameter, it was observed that the administration of acetic acid by interrectal route significantly increased the colon length ratio. How it increased the colon length ratio? Because of the severe inflammatory cell infiltration and also vascular permeability and also the edema of the colon. These 3 parameters are mainly responsible for increasing the colon length ratio. The second parameter that is being checked out was the histopathological parameters. It reveals that the preserved entire colonic mucosa and inhibited inflammatory cell infiltration, ulceration, and erosions are mainly responsible for changing the histopathological parameters in the colon tissue. From this, we can conclude that we have seen that in these parameters, we have seen that the histopathological parameters are similar like the treatment group, that is just sulfasalazine. From there, we can conclude that there are very little changes that are being observed as compared to the standard drug. And the third parameter, that is your SOD. I have already told you this SOD, the superoxide dismutase. It's a free radical scavenger activity. It produces antioxidant, and this parameter also reveals that as compared to the sulfasalazine group, the 2 plant group 4A, 4B, 4C, and 5A, 5B, 5C have similar SOD activity, the similar percentage of inhibition. From there, we can conclude finally that the Centella asiatica is more effective in treating the inflammatory bowel disease than the Murraya coengi, and this result can be helpful for future clinical trial of Centella asiatica leaves or its bioactive components as a natural, safe, and effective treatment for the treatment of inflammatory bowel diseases. So these are the references of my presentation. Thank you. Sir,
Your groups, control groups.
This one, sir.
Yeah, so somewhere you're putting 100 mg, somewhere you're putting 200 mg, somewhere you're putting, all these different things. So what condition, what is the effect of concentration? What is the optimum concentration
Sir,
as per your observations?
Sir, actually, the first thing is that I have not done the acute toxicity study, sir, because these two plants are generally regarded as safe. So I have taken 3 different concentrations to check their activity, sir.
200 is the best?
Sir, maybe because
No, what is your histopathological scores are coming? You have a score there, right?
yes, sir. This 200 is optimum. We are getting the optimum result, sir.
Because it's the maximum.
Yes, sir.
Why you did go to 300? Who stopped you to stay?
Sir, because I have limited numbers of animals also, sir. From
That's okay, but why did you choose 50 to 200? Why you did not choose 50 to 500, 50 to 1000, 50 to 2000? Why you choose only 200? That's my question.
Sir, because the maximum dose that can be given to the rat is also fixed, no, sir.
How is it fixed?
Sir, we can't give 1000 mg because the animals may die, sir.
Okay.
Yes, sir. It is limited, sir.
So your 200 is maximum.
According to guidelines, sir.
Okay. Fine. That's all from my side.
There is one query, actually, for the last speaker also, I had the same query. So you, while preparing the sample, you wait for grinding the sample?
Yes, sir. I have just dry set the sample and after that grinded it, sir.
So what were the conditions? Like at what temperature, for how long did you do?
Sir, in a sun dry only. It means within the room only, sir. Within the room means not directly sun rays will fall it and under the room only. I have just yes, sir. Set drying only, sir.
So the temperature
it will be maximum, sir, up to 25 to 27 degrees Celsius, sir.
Okay. Thereafter you went for hot software extraction.
Yes, sir.
Why hot software extraction? Why did can't we go for some extraction where the temperature can be kept at a
And ambient means for percolate, like that means just dipping the plant in like that extraction.
Yeah, because in both the cases I have seen that you went for hot software. And what was the temperature for that?
Sir, it was maintained at around 30 degrees Celsius, like that only, sir.
So,
sir, for ethanol, sir, hydroalcoholic, sir. Hydroalcoholic, sir.
So why did you go for the soft slate? If you want to do at 30 degrees Celsius, what I feel is you can simply dip in some
solution and just
solution. Why did you go for the software apparatus?
Sir.
Okay.
Thank you, sir. Thank you.
Thank you. Thank you so much, sir, for the presentation. Now, for the next presentation, I would like to call Ms. Arunima Dhar, a PhD scholar at Fab Lab, IIT Guwahati.
Good afternoon, Uplori sir and Himjyoti sir, and all the participants and dignitaries present here. So I'm Arunima Dhar, a PhD scholar from School of Agro and Rural Technology. Today I will present a oral presentation on the topic on extractability of pectin as a potential therapeutics from citrus fruits. Now, in this two-day seminar, we had talked many about this pectin, the health benefits of pectin. So this basically, this is a soluble fiber, which is a heteropolysaccharides, and this is present in the primary cell wall of many fruits that we normally have in day-to-day life. And this citrus fruits are considered one of the essential source of pectin and being used in food industries for many years as a stabilizer, as a thickening agent, and also as a packaging material. And Northeast India, as we know, have a very favorable climate to cultivate this kind of citrus fruits. And this citrus lemon or Kaji limon this got the GI tag also from Assam. About that, the origin of this particular lemon is from the Assam. So I took up this for my further study. So objective of this study was to see the effect of drying temperature on the pectin yield from the citrus lemon, and also exploration of that application of that pectin as a nutraceutical or a raw material for the pharmaceutical industries. Now, since we know that the pectin, this have a very complex structure where galactolinic acid is present, and also there is different side chains that is there, and the extraction method of it affects a lot on the functionality of the pectin. As it is not extracted properly, it might cause the destruction from the favorable function that we want from that particular drug. So it is important to optimize the extraction method. So now, as of now, the extraction methods that is available to extract pectin are basically microwave extractions, conventional extraction methods using tray dryer or this heating method. Also, acid-based extraction is there. Other than that, water extract is also present, and enzymatic extraction is also done. So based on all the studies in citrus fruit, mostly they are using conventional heating and followed by an acid hydrolysis for the extraction. So for my method, I basically use this acid hydrolysis method. So I used to sample from the citrus lemon, and I used the peel of the citrus fruit, and along with that, the peel and the albedo. I basically extracted the juice, washed it properly, and the peel and the albedo was used for the further study. So these two samples I have dried in two temperature. One is 105 or till its bone dry mass, and one was till 60 degrees centigrade till its equilibrium moisture content. And finally, the acid hydrolysis was done with 0.01 normal HCl, and it was then kept to water bath at for 1.5 hours at a temperature of 80 to 90 degrees centigrade, and the pH was kept 2. Then for the precipitating agent, I have used 96% ethanol, and then the further filtration was done, and it was washed with 74% ethanol, and then drying was carried out at 50 degrees centigrade to extract the pectin. Now let's come to the research, the result of the study. So basically, as 105 degrees centigrade is a still higher temperature, so the drying was achieved in very faster rate. Around 2 hours time, we got the dried mass as a bone dry mass, and for the 60 degree temperature, it took around 12 hours to achieve the equilibrium moisture content. So basically, in the lower temperature, we are hitting it in a higher time, but the intended product at 60 degrees centigrade had very light color, which was more acceptable as a consumable product. So the next, as the idea behind to dry it till its bone dry mass is to see what changes is happening if the sample that we are using for further pectin extraction have some water present on it, or if it is only the mass of it, what is the changes or what is the impact of it in this like pectin extraction and the yield. So coming to the yield, it was seen that the temperature obviously have a very significant effect, giving a p-value of less than 0.05 for both the samples. If we compare between peel and peel with albedo, there was no such significant difference seen, and that is the way that in a lower temperature, when we are heating it and going for further extraction, the pectin yield is better. So now why we want to actually extract pectin in a very controlled manner, and what is the usability of it. Now the application of pectin is very high in food industries as a packaging material that I already mentioned, but in health and pharmaceuticals it has a high possibility. So that so for that we need to first know that how the digestion of the pectin that is digestion of the pectin happens in our digestive system. So first of all, there is no change that happens due to the maceration or mixing with the salivary alpha amylase that is present in our saliva, and also in the stomach there is no enzymatic change in the pH. No degradation happens of the pectin. Also in the small intestine, not much degradation happened, but yes, it do have some functional value in the digestion process. It sometimes acts as a agent which gives us satiety, and also some of the in the very hormonal effect, it actually changes due to high consumption of this soluble fibers, including pectin. Finally, it is got fermented in the colon by the probiotic bacteria that are present, and this bacteria are basically the gut bacteria that we know, and the gut health have a very high impact in our overall health. Like it is related to our cardiovascular health, even the brain health. So gut, making a healthy gut is a very important thing. So that is possible when we take a proper amount of prebiotics. So other than that, the application of it as a nutraceuticals can be possible, because pectin is considered as a grass food that is generally recognized as safe. So there is no proper doses that is defined for this pectin, but that has to be done, because if we are intended to use it for any therapeutic purpose, that how much pectin in what dose for how many days it is needed, that proper defining should be important. But other than that, there are different application or the benefits of the pectin as a nutraceutical itself. First of all, it's a natural prophylactic. That means it can help in preventing disease. It might not help in curing any disease, but it can prevent many disease. So as a preventive measure, we can use it. And other than that, as I already mentioned, as a prebiotic, and also it can, our gut, if it is in a good condition, can digest 90% of the pectin, so that this can also help in the treatment of high cholesterol, even high triglyceride as well, because when the gut bacteria that ferment pectin, there is a compound called short-chain fatty acid formation takes place, which actually can regulate our cholesterol mechanism and can decrease the whatever LDL or LDL cholesterol is there, which is not needed for our body, and increase the HDL cholesterol, which is important for our body. So that way it can balance the cholesterol mechanism. Now coming to a case study, as I already said that it can prevent many diseases. So it was done, basically it's an animal-based model that was studied, that a galactonin 3 is a serum galactonin 3 is a novel indicator for cancer. So if somebody have a high range of galactonin 3, it can be concluded that that person might have that malignancy or might any malignancy or any cancer, like they can have, it is a preventive measure can be taken for cancer, and if somebody having high this particular compound in a high amount. So basically in this study, 5 groups were prepared along with a negative control, which was given no treatment, and other groups were given treatment of different concentration of the citrus extract. And then finally it was then studied after 3 weeks of the treatment, and it was seen that the negative control which haven't received any kind of treatment have got 100% the highest that metastasis or the cancer, like the actually detected cancer in that particular mice. It was and then which the group that received almost the citrus treatment that got around 60, like the highest concentration got around 60%, lowest of this liver metastasis. And then finally that group with negative control, they have higher level of galactonin 3 as well. So it can be concluded that the citrus pectin is a potential inhibitor for liver cancer and can be used in a human system as well. For that, the proper studies are needed because animal digestive system and human digestive system has a huge difference. So in case of cancer treatment, proper clinical trial is needed to conclude that. So in case of not only as a nutraceuticals, it can also be used as a raw material for pharma industries. One can be a targeted drug delivery agent. That means if somebody wants to have a drug which might affect our intestine, but it is needed for our colon. If somebody having colon cancer and they have to have a chemotic drug, then that will be voted by the pectin, and that is if that is administered, that will only degrade it by the colon cells. It will not degrade it in the in our mouth, not in the small intestine or the large intestine. So that this particular drug can only affect the colon cells. In case of any antimicrobial drug also, we can administer it through the pectin coated film, so that the same effect is going to be there and we can have a targeted delivery of our drug. Now to conclude my study, that the pectin is a promising nutraceutical and it can become in case of preventing disease and also in as a raw material of pharma industries. But yes, further studies are needed to make the to define its proper dose, as dose response relationship also have a very higher impact in this dose detection and dose and that that exploration of doses is needed if we want to make any supplements or any treatment in that case. So that's about my presentation, and this is my references.
You are doing acid extraction or just conventional extraction?
Both.
So the values you reported are for what? Can you go to that slide? Yeah, this one, next one. Yeah, this one. These values that you are reporting.
Sir, it is first of all, it is dried convectionally, then acid hydrolysis is done.
And what acid you are using?
HCl 0.1 node. 0.01 node.
Okay. The second thing is that, see, any application requires purification. So, given the facilities that you have here in IIT Guwahati, going into the therapeutics might be what you plan then, because you are only reporting the literature data.
Yes.
So what is it that you intend to do?
Sir, basically, as of now, as a diet, I want to implement.
Yeah, but that type of experiments can you do here in the campus? Even if you say you're
Sir, if it is in human trial and
you can do human trials.
Like a clinical trial, that if
Can you do in vivo trials?
So basically,
Is the campus having that facility? So you should think about that. So another area that probably, because I'm also working in this field, I'm working with that Jamura, you know.
Yes, sir. Okay.
Yeah, so we are working with that. One of the questions we recently received were, what is the purity of the pectin that you produced? And how you can enable 100% pectin extraction, and how you can purify that raw pectin that you got. So that would be an interesting area to explore. And also this, you have mentioned all techniques, but you did not mention ultrasound. Can go back to your slide. We are actually trying ultrasound. So your microwave is there. And your ultrasound also you have ignored. Ultrasound also is, it's like, very good procedure. What we found is that you don't get a brown, very dark brown pectin after ultrasound. Whereas with your process, I think you'll get.
Yes, sir. 105 was very dark, but
yeah, but 60 also.
It was not that much good, but after the extract I got, that was okay. But after I dried it, it became dark. And whenever I storing it, it getting dark.
Yeah, so some things that you will have to explore again. I mean, another area that you can probably think about is a combination of, say, microwave and ultrasound. So microwave can do like very short time you can give microwave, and then you can do it. And another thing is that this drying entire thing you will get rid of it if you make it like a paste. Are you exploring that? Because you're drying and then your dry powder you're using for extraction, am I correct?
Yes.
So that's a lot of energy you're taking in. So instead, if you just make it like a paste or even a slice, and then do whatever extraction you want to. And if you want to dry, then shade drying probably. Okay. Or 40 degrees for 1 day. Something like that. Then think all these things, and more you can focus more into that purification part. Because if you want to apply it anywhere, I don't know about that case study, what is the purity of the pectin that they use, what you have shown. That, lemon or, citrus fruit extracted pectin you are saying.
So they
What is the purity?
modified citrus pectin.
Yeah, but the purity is important.
That was not mentioned.
So you have to look into these aspects, because unless it is not pure, they will not allow to even, do it for mice also. All the best.
Here is the degree of esterification is a quality parameter for pectin that we have to consider. And that also relates to the ripeness stage of the ripeness of the fruit.
Low methoxide pectin and high methoxide pectin, those things.
So those are clearly different qualities of pectin. Okay. So that we have to consider.
Characterization I have planned to do, but it is still.
And in the case study, you have mentioned modified pectin.
Yes.
Pectin, it can be modified by a number of techniques. Which modification technique are you talking?
Was not actually the mention.
Please go through properly. Okay.
One more question, Radhima. It's a good word. Pectin is very important. We need also. Now, do you think with fruit you have extracted? So, if I look at the economical kind of a thing, you can use in forget about this in anti-cancer also. Can you use in a cosmetic or so, and where the purity or something? Any other potentials are also there for the.
Yes, ma'am. For the cosmetics, it is being used as a gelling agent. Many cosmetics they are using, and as a filler also they use. So,
can you is it possible what you have got the yield here? What you have shown? I do not know much about this, but I am just asking for idea to make in a cosmetics. Okay, my idea is only that. So could you please share how economical it would be? Because so many farmers and also many fruits gets wasted or so. So if any standardized method is there, so can they earn something or can we use it for
this fear is thrown to the bin. So
yeah, that is what I feel. Such kind of a thing. See, we need to also look into that, and if the techniques are standardized, well studied, can we also promote that through for our SAGs also? That is why.
What is missing, madam, is the circular economy.
You said it, so.
So we do research, we give PhD degree, and we try to, make Arunima as Dr. Arunima. One of my students also working, Mr. Khalid, will become Dr. Khalid. What we require is a complete process by which you can produce that pectin and you can sell it in the market, like in an Amazon or some place, even, in a small scale also. Yeah, that type of, technological, completeness is required.
And since I haven't got much difference between the citrus waste that is with the albedo, I just extracted the juice and then I just use that part. So any citrus industry waste will have the same thing. We don't have to actually go and peel it properly. So that we can consider even, and I am working in this method development part actually, so maybe.
I just want to, like nothing related with the presentation, this presentation and the earlier presentation. Like, as far as the citrus fruit or lemon is concerned, in Ayurveda, especially in South, out of this, the special type of lemon, which is produced in last 20 years, we make in Ayurveda, we produce Madhifal Rasayan. So Madhi means basically, like the tonic for your complete GI tract. And see, like in our traditional practices, wherever there is a problem, like in our food practices, those things are used, like here, like lemon is used, So if we use, this is just for like general knowledge, like I'm talking about our day-to-day use, because suppose like we make a nutraceutical preparation and it comes to the market, how many people can afford this or not afford this? But these things which are ample here, and if we like, among community, we talk the benefit of it, and it is plenty available. Like, see, if we are using this lemon every day in our day-to-day use, after food or before, like if you go in West, lemon is there, like with every meal, they will have a sweet lime, lime is always there. Even if, like if you go in the restaurants or in the hotel, like when they put the water for common use, there are lemon slices. This I'm talking when we are going in community, we should talk about the benefit of that. Your scientific presentation and all those things should go on for the modern world and for industry and all those things. But this is just locate that. And before that, like in other presentation, the other gentleman was talking about that is like in Bengali we call it Thankuni, and in Ayurveda it is Brahmi or Mandupani. So in Bengali tradition, like every Bengali from childhood, every day they have to take that, mothers and Dadima and Nanima used to forcibly give them Thankuni every day. Okay, because you are taking Thankuni every day in the morning, and then you are having bhaja in breakfast, bhaja in lunch, and bhaja in dinner. So it's a balance. Like if you go in South, there are a lot of spices in their diet, all garam masalas, but then they will have, chaach and all those things. So if you go in Gujarat or Rajasthan, they will have a lot of farsan and papad and all those things. So they are balanced. Our ancestors have balanced everything. So that, we have to remind people like this. I'm talking because like, I think this department is connected with the rural population, working with the rural crowd. So when you go there, you are talking about technology or promoting technology, but just when you have gathering of people, remind them of their, like traditional practices. See, now we, like we go and we pay a very good money and we buy prebiotic and probiotic and all the other things. Okay. Kanji is a very, very cheap thing, like every household can make kanji, which we have forget. In South, like kanji vada. So one thing is fried, which is going to take care, like, like play around your GI tract, but then there is kanji with it, which is going to take care of it. So see, we have to go to the, like अपने हमारे traditions में हमको जाना है, लोगों को याद दिलाना है. See what is happening now. है ना, लोगों को बड़ो माइक्रोवेव निकाल दो, कुकर निकाल दो. ट्रेडिशनली और लोग आ रहे हैं, क्योंकि जब तक हम नेचर में वापस नहीं जाएंगे, जब तक आप नेचुरल हो ही नहीं सकते. ठीक है, लोगों को खूब पानी पीना है, पानी पीना नहीं चाहते हैं, और फिर वो कॉस्मेटिक्स लगा के सुंदर नहीं हो सकते, पानी पी के सुंदर हो सकते हैं. मैं आपको एक प्रैक्टिकल एक्सपीरियंस, आप सभी लोग यहाँ के हो, आसाम के हो, नॉर्थ ईस्ट के हो, आप सभी लोग ट्राई करिए. कुछ नहीं करना है, सुबह से शाम तक जो एक आपका जो गोल नींबू होता है, जिसको जो हम आयुर्वेद में उसको जमीनी नींबू बोलते हैं, like that is used in medicine. एक तो बड़ा वाला ये सेंटेड वाले की बात नहीं कर रहा हूँ, जो गोल वाला, गोल नींबू जो होता है, आप सिर्फ वो पूरे दिन में एक नींबू खत्म करिए, and you see your detoxification. यहाँ से यहाँ तक जितना कचरा है सब निकल जाएगा. So things are there, उसका हमको इम्पोर्टेंस समझना है और उसको डेली यूज में ले जाना है. कोई आपको इंडस्ट्री पे जरूरत नहीं पड़ेगा, कोई डॉक्टर के पास जाने की जरूरत नहीं पड़ेगी, कोई कैंसर नहीं होगा, कोई कुछ भी नहीं होगा. तो थोड़ा सा हम लोग अपने जो प्रैक्टिसेस थी, जो हमारी ट्रेडिशनल प्रैक्टिसेस थी, we have to remember that and लोगों को याद दिलाना है. ठीक है, now like this is all traditional things, like even our traditional alcohol and all those things are also getting very good market, like the liquor made out of mahua. What is the English name for that, for mahua? Acha. So like, let's see, our traditional things were pure and they were having medicinal values. So see, we'll work in nutraceuticals because we have to sell also, we have to make money and we have to, you know, like work with the world. So that should go on parallelly, but we should remain to our grassroots, because if I tell somebody to buy a gold limbu and use it every day, that's possible even for a, you know, villager, a poor man, a rickshaw puller, or anybody. But pectin, I think 5% or 10%, even if we process and we put it in the stock, 1 or 2% of population can afford. So let's, like, all these natural things, herbal things, Ayurveda is made for the masses. So let us remember that also, that, you know, we have to keep our communication in that way also. Thank you.
Thank you, Arunmati, for the wonderful information. Now for final presentation, I request Mr. Gaurav Bhatt, a PhD scholar of Applied Biodiversity Lab, to come forward for his presentation.
Good afternoon, respected DC members. Hello. Good afternoon, respected panelists and one and all present here. I'll be presenting my work on the compound which was isolated from Pongamia pinnata tree way back in 1925, that is a century ago, which is named Karanjan. So Karanjan, if we see the structure of Karanjan here, it is a flavonoid compound and it has a furan group attached to it. If you can see on the left hand side, there's a furan group attached to the flavonoid backbone. So Karanjan was isolated in 1925 and since then it has been used for various biological processes. If we look into the biological effect of Karanjan, it has both in vivo, it has shown both in vivo and in vitro studies various activities such as anti-inflammatory, anti-hyperglycemic, anti-ulcer activity in vivo, whereas in vitro it has shown anti-diabetic and anti-cancer activity. Recently, studies have reported the anti-cancer potential of Karanjan and it is marked as one of the potent anti-cancer molecule. A study reported by Roy et al. They have demonstrated the various IC50 value in various cancer cell lines. And here we can see that most of the cancer cell lines, the IC50 is very high. It is greater than 50 micromolar concentration. But it is still regarded as a potent anti-cancer potential. So there is a gap in the literature that this high concentration, that is 50 micromolar, can it be used in a cancer therapy or not? So to work on this gap, we wanted to know whether the effect of Karanjan what is the effect of Karanjan in breast cancer cells, first of all, because there is no study reported in the hormone responsive cells that are breast cancer cells, and also the genomic effect of Karanjan at the gene level. What effect does Karanjan have? That is never been studied. And we wanted to know the genomic level through the transcriptome profiling, so that we can know all the 20,000 genes that are being regulated by Karanjan itself in MCS7 breast cancer cells. To cater to our objective, which is where we wanted to know the genomic correlates of Karanjan at the cellular biology level. So we did RNA sequencing. And for RNA sequencing, firstly, we isolated a compound Karanjan from Pongamia pinnata seed oil. So we ran the oxide extractor, and ethyl acetate extract was, we got the ethyl acetate extract, and through column chromatography, we isolated Carangen crystals. These Carangen crystals were then characterized via mass spectrometry, NMR, and the purity was assessed via HPLC, and we got around 99% pure compound, as we can see through our spectral peak itself. After getting the purified compound, we wanted to know what concentration should we use for our RNA sequencing and the genomic effect that we wanted to know. So this is a cell viability assay. Here, if we can see, the red dots represent the 0-hour time point, whereas the blue dot represents 24-hour time point. And on the x-axis, we can see there is different doses of Carangen from 0 to 50 micromolar, and estrogen, which is a mitogen, is used as a positive control. So if you can see in this graph, at mostly all the concentrations with respect to control, we can see that all concentration has similar effect, which gives us to a hypothesis that all effect that we are getting is direct and free of any toxic or mitotic effect. There is no toxic effect of Carangen at any of the concentration at 24 hours. So we used 24-hour time point and 10 micromolar concentration, which is a physiological range, which is in this physiological range. We haven't taken 50 micromolar because 50 micromolar is far beyond the physiological range and cannot be used for therapeutic purposes. So we move ahead for our RNA sequencing study, where we treated MCF7 cells with 10 micromolar Carangen for 24 hours, and we did the RNA sequencing, and we found out that there were 736 genes which were modulated by Carangen. And among these 736 genes, 374 genes were upregulated, whereas 362 genes were downregulated. We also validated our RNA sequencing result via QRT PCR, and we found out that some of the genes which are highly upregulated, if you can see in the down panel, the CYP1A1, CYP1B1 was getting highly upregulated upon Carangen stimulation. Similarly, genes such as cystatin A, CST1 was getting down modulated upon Carangen stimulation. Also, we wanted to know what are the pathways that are being regulated by Carangen. So we found out that G2M checkpoint pathway, which is a hallmark gene set, and what we have found out that in the literature itself, Carangen is known to be a cancer prevent. It is known to prevent cancer, and it is known to arrest the G2M checkpoint. But what we have found is opposite. In MCF7 cells, there was a progression of G2M checkpoint genes, such as SENPF, CDC25B, which leads to the progression of cell cycle rather than cell cycle arrest. Similarly, another pathway was upregulated upon Carangen stimulation, which is estrogen responsive pathway. That is, estrogen receptor alpha was getting involved in the Carangen stimulation, which lead us to a conclusion that Carangen may mimic estrogen and can have proliferative effect rather than anti-cancer effect. So here, when we compared the data from our Carangen data along with the estrogen data, the RNA-seq data, where we have compared 736 genes that were from our RNA-seq data, high throughput data with estrogen-regulated genes, we found out there was a significant overlap. More than 420 genes were common in both Carangen-regulated genes and estrogen-regulated genes. Also, when we did the correlation analysis, we found out there was a positive correlation between estrogen-regulated genes and Carangen-regulated genes. So in the end, the key outcome from our RNA sequencing result was there was 736 genes which were modulated by Carangen stimulation. Also, G2M checkpoint genes represent cell cycle progression rather than cell cycle arrest, which was previously reported by various researchers and cell cycle arrest. Similarly, we also found out that another pathway which was enriched upon Carangen stimulation was enrichment of E2 response early pathway, where we found out that genes which were regulated by estrogen was also regulated by Carangen itself. Hence, we predict Carangen may be estrogen-like molecule itself. This is for the first time we are predicting that Carangen is not anti-cancerous in nature, but it might induce proliferative effects. Also, what we found out that there is a tamoxifen, which is the ER alpha modulator. So the genes that were regulated by tamoxifen was also regulated by Carangen, which gave us a hint that there is involvement of ER alpha in Carangen mediated effects. So to cater to that objective, that whether ER alpha is involved in Carangen mediated simulation or not, we did a long-term simulation of Carangen where we checked the cell viability in different cell lines. MCL7 and T47D are ER alpha containing cell lines, whereas MDMV is devoid of ER alpha receptor. So when we see the MCL7 cell lines, if we see at 10 micromolar concentration, there was an increase in the cell viability. Similarly, in T47D, at lower concentration, that is 1 nanomolar, there was an increase in the cell viability. But at higher concentration, that is 10 micromolar, 50 micromolar, there was a decrease in the cell viability. But this trend was not observed in MDMB231, which is devoid of ER alpha, estrogen receptor alpha receptor. So this further cemented our hypothesis that there is an involvement of ER alpha in Karanjin-mediated effects. So we propose an hypothesis where we predict, since Karanjin is semipolar in nature, it can diffuse to the plasma membrane, it can activate ER alpha and hence regulate different gene expression, which we have already seen in our first objective. Further, it is also known that flavonoids, which is, since Karanjin is a flavonoid, and flavonoids are known to bind to ER alpha and affect cell proliferation and gene expression in a concentration or cell-dependent manner. It is because flavonoids, because of the structure similarity to estrogen, can mimic estrogen itself. So if we compare the structure of Karanjin and estradiol, Karanjin also comprises of 5-ring structure and 6-ring structures similar to estrogen. Hence, we predict that Karanjin may be estrogen mimetic in nature. Further, it is known that these phytoestrogens, the compounds which mimic estrogen, could be used in hormone replacement therapy, where it can be used to treat postmenopausal women. It is also used to treat osteoporosis and cardiovascular disorders. So we wanted to know whether Karanjin binds to ER alpha or not. We did a molecular modeling study where firstly we found out that Karanjin binds in the same ligand binding pocket as estrogen. And also we did the molecular dynamic study where we ran our MD simulation for 275 nanoseconds. And we found out that both Karanjin structure and estrogen structure lies in the same ligand binding pocket, and it forms hydrogen bonding with histidine 524, similar to estrogen itself. Our in silico molecular document study led us to a prediction that yes, there is involvement of ER alpha in Karanjin mediated effects. Next, we wanted to know through our in vitro experiments that whether there is a involvement of ER alpha in Karanjin mediated effects or not. We did the Western blotting analysis where we found out that Karanjin, similar to estrogen, leads to the down modulation of ER alpha protein level. If you can see that at 36 hours, 48 hours and 72 hours, there was a reduction in the protein expression with Karanjin simulation, which was accompanied like estrogen itself. Then we removed the ER alpha from the system. We knocked down the ER alpha from the system, and we found out that certain genes, which are estrogen target genes such as TFF1, cystatin, and STC2, there was, if we can focus on this TFF1 gene expression, there was an upregulation of gene in normal scenario. But when ER alpha is removed from the system, this upregulation was abrogated. Similarly, cystatin A was getting down modulated upon Karanjin simulation, but when ER alpha was removed from the system, this regulation was suppressed. Further, we did a chip analysis where we wanted to know that whether ER alpha is physically getting engaged to the promoter region of certain genes or not. We did chip analysis where we found out that with respect to control, there was an induction in Karanjin. As you can see in the down panel, there was an induction in Karanjin panel of all the genes that are PS2 gene, FOXP2 gene, and cystatin A gene, which led us to a prediction that there is an involvement of ER alpha in Karanjin mediated effects. All these studies led us to an overall conclusion that firstly, there is a dose dependent effect of Karanjin. As at higher concentration, Karanjin can have anti-cancer effects, whereas at lower concentration, it can have proliferative effect. Second, when we look into the genomic correlate of Karanjin, it regulates around 736 genes. There are a host of genes which have been regulated by Karanjin and also we found out that estrogen response early genes, there is a involvement of estrogen receptor pathway. And if we look into the mechanism of action, the ER alpha mediated effect was present. So in the end, what we can conclude is the Karanjin is, for the first time, we hypothesized that Karanjin is a novel phytoestrogen, unlike its anti-cancer property. Also, it can be used in hormone replacement therapy since it mimics estrogen. Second, we also predict it to be a selective estrogen receptor modulator, and it can be used as a potential therapeutic target, which targets ER alpha. since most of the breast cancers, 70% of the breast cancers are ER alpha positive, hence a molecule like Caranzine, which targets ER alpha, can be used as a therapeutic target for combination therapy. It cannot be used as an anti-cancer agent, but it can be used in combination therapy. Next, we also wanted to do some of the in vivo experiments, but because there is no facility for in vivo experiments in IIT, so we are looking forward for some of the collaborations that we can do. So thank you, and I'll be open for questions.
Thank you, Gaurav.
Yes.
Only one humble question.
Yes.
The reversibility of the cancer cell, can it be addressed? All of you are doing the modern way of analyzing.
Yes, sir.
But the Ayurvedic and the yogic way of looking this cancer is, can you make the cancer cell function the backward way?
Yes, sir. A lot of studies are going on for that. So it is basically done through cancer stem cells. So there is a theory that cancer stem cells.
So, so how good is your work towards the cancer stem cells?
So we haven't looked into the cancer stem cell aspect of it.
Also.
So I think my work basically is Caranzine can act as a targeting molecule for breast cancers specifically because it targets ER alpha specifically.
Okay.
So since it targets ER alpha, it can go to if we like derive
Can it also function the same for the breast cancer cells according to your hypothesis?
Yes, sir. Basically, it can go to the breast cancer cells, bind to ER alpha.
No, stem cells, I mean.
So maybe, sir. We haven't worked in stem cells, but we have worked in breast cancer cells. So if we work, so that can be a proposition where people can work on breast cancer stem cells and then see the effect of this compound.
What challenges are there for your, for your Caranzine, according to you?
So the challenges, what I feel is
Okay, all your science, everything is done. I mean, the application perspective.
Yes, sir. So application is, it can be, so first we have to do some in vivo experiments where we have to prove that it binds to ER alpha. And we also wanted to check in mice experiments where we wanted to know that since ER alpha is also involved in bone mineral density and it can lead to various positive effects. So basically targeting this ER alpha is our main focus, sir.
Thank you.
Regarding this Caranzine, can you put some light on how it was traditionally being used? Was it administered orally?
Yes, sir. So basically there are various studies in vivo where they have shown that oral administration of Caranzine has been done.
But what is the traditional?
So traditional use of Caranzine, so basically it is derived from Pongamia pinnata oil, Pongamia oil. And so Pongamia oil is used in traditional medicine system and in Ayurveda for various ailments, such as keloid tumors, for example. If you look into our study, keloid tumors.
Any study related with the history in our GI tract, that particular compound.
Yes, sir.
Where does it get actually absorbed? Has there been any study related to that?
No, sir.
No, sir.
Not for Caranzine itself, sir, but for Pongamia, I think there are. Yes, sir.
The digestive metabolism.
Yes, sir.
No study have been done.
Not really, sir.
Professor Ramgopal sir has, maybe coincidentally or whatever, he has brought over stem cell with courage. It is currently in Ayurveda.
Yes, sir.
And sir was asking about oral. So as per my knowledge in Ayurveda practice, current is mainly used in local application.
Yes, sir. Oils, yes, sir.
Okay, so and see, there are many herbs, even in when you are describing about currants, you say it is anti-diabetic and all these things. because Ayurveda is based on six days, and then 3 dosanas and the combination of that. And see, there are specific plants like in fruit, which can be used as a herbal stem cell. So even Karanj has that property. Okay, and there is one product. There is somebody in Ranchi. Sir, it is very interesting. Like you raised that question of stem cell. So there is some doctor in Ranchi. He is using Karanj oil for all the diseases. And you will not believe, I think maybe 10 ml ampoules, 10 small bottles of 10 ml, maybe 20 or something is selling me for 30 or 40,000 rupees. No local application. Like suppose somebody has got a knee pain, they just apply locally. See, and then according to Ayurveda also, Karanj oil is a very good carrier. So that's the reason like it was all skin disorders, because you know that dryness is the or lack of moisture is the main cause for dry eczema or psoriasis and all those things. And it has got property of it produces new cells. So whichever area, like wherever it is required, stem cell means if some now this herbal stem cell is the new trend. There are especially there are 12 plants like we can use grape skin is one of that. Resveratrol is a molecule which is derived from grapes. It is derived from many other things, but grape skin one is the good one, the very good one. So wherever there is system failure or organ failure, it will start producing the required even I have a product, I have a formulation in my company and I use it with my product patients. So that I think I use it for whether it is kidney failure cases or degenerative disorders, or maybe people have low testosterone levels or RA positive cases, thyroid cases. So stem cells it is equivalent to whatever benefits this stem cell, modern stem cell therapy is promising or offering. We get the similar results with even this herbal stem cell formulations. So nowadays, in Malaysia also some company has come out with stem cell product, and in Switzerland and so many places. Now the herbal stem cell therapy is also becoming very famous. This Karanj has got that property. So that doctor, if somebody has got the spinal problem, just apply on the spine area, and he will say that just apply, don't massage it, because if you massage, the stem cells will die. and people are lining up for that. So any problem, only Karanj oil is there, and it will work up to some extent because it has got that stem cell producing property.
Yes, the Karanj oil is used for dandruff also. So recently there are many formulations for Karanj oil, which is used for dandruff.
As I said, there are in Ayurveda we say, suppose you are talking about Karanj. So we say that Karanj will work in this because according to the test, out of 6 tests, how many tests are present there? So it will work on this disease. But then we call Rogadika. Okay, you are in IIT, suppose you are a mechanical engineer. So mechanical engineering is your forte. But you know IT also, you have learned other things also. In same way, the herb will work on so many other things, but it has got mastery, that is Rogadika. So Karanj has got Rogadika on skin disorders. I have never used, or I have never heard that somebody has, we use Karanj if somebody has got worms infestation.
Yes, sir.
It has some poisonous extensions, so internal use is not promoted of Karanj in Ayurveda.
Karanj oil, yes, sir.
Karanj oil, it is locally
internal utilization.
So Karanjin, sir, not oil. Sir. Oil has many components. Oil has more than, sir. If we see Karanj.
Karanj, not oil.
See, if you are using that, then the question comes, the stability and toxicity study, and then if you are doing the talk, you are doing on mice, but what on humans? So there is a lot of question. And then, you end up with a zero result because you will not be able to prove that it is safe to use on human. So we have to, like, if you are putting your efforts, put your effort, whether, like, in the end, it is like outside, like it is marketable and it gives a proper return on that. So, हम लोग तो देखो भाई वैद्य लोग हैं, देसी लोग हैं, बाप दादी ने जो बोला है, हम आंख बंद करके विश्वास करते हैं और उसी को फॉलो करते हैं। साइंस बिलीव जो देखते हो उस पे आप बिलीव करते हो। In Ayurveda, we say, like for proving anything, one is pratyaksh, like what we see, we believe. Anuman, logic also. Yes, and then yukti, analysis or logic, we karte. And the fourth is aptopadesh. If my dad is saying, like, कोई चीज जो मैंने देखी नहीं है, ताजमहल ऐसा था तो I believe because my dad is saying, my grandpa is saying, or my mom is saying. My guru is saying, or my ancestors have said, like Shastras. Okay. And like people say that Ayurveda is, it's a takya kalam for all modern science people and modern people, that it is anecdotal. Kahani in Ayurveda. Ayurveda is not kahani. All of us, Shastra, Charak, Susud, and everything. Like, see how we are sitting here. So, Punarasu Agni is like the guru wrote Charak. So he has 8 disciples. So suppose today's subject is cancer, Agro. So all the disciples, they are giving their opinion, and in the end, like the guru is giving his opinion. So all Ayurvedic textbooks, whether it is Charak Samhita, Susuru Samhita, Bhagavad, Kasya, Mahaprakas, everything is synopsis of conferences or seminars. Everything is well discussed. It has got all scientific background, and like only to to satisfy the modern world. Like अंग्रेज को हम हिंदी अंग्रेजी में सिखाना चाहने की कोशिश कर रहे हैं. So that is all confusing. Like that is going on. But we should have faith in our own sciences, our own things, and like if we promote it in its natural thing, it will be like cheaper and better for the masses. Thomas ठीक है society के लिए जो आप क्योंकि यहाँ the gathering here is like you people are attached with the rural cause. So that's why I'm saying that we have to promote. Don't go to the doctor. You take this. You promote all that, and then you have to come out with you have to find out something from their traditional practices also, okay, like what they were using, and and then from there, take something from the traditional practices, and then we will commercialize it, and that in that form only we'll take it to the market. And it has started happening, like that Bhim Vita example of Assam, the guy who is making that Bhim Vita from Bhim gold or Bhim banana. So same way, like wherever you are there, like whichever area you are working, just try to see what are the ample produce there and what are the health practices there, what were the traditional values were using things. So from there only, like you'll come to know about a lot of things, and that we can commercialize, and there will be zero investment in that. Thank you very much.
Thank you, sir. Thank you, sir.
Just sharing my opinion, okay? And this is all in a positive note.
Yes, sir.
Okay? We have a very rich culture, very rich science, so we must be proud of it. There is no need to prove that we are scientific. We were scientific and will remain scientific. Any principle of Ayurveda or any principle of Veda, no line have changed. We don't say that this is good for bacteria, and then we say after some time we say that this is not good for bacteria. Now this antibiotic is there. We don't keep on changing our words. हम लोग पक्के लोग हैं, पक्के ही रहेंगे. तुम्हारे लिए बदलते नहीं. Thank you.
Thank you, sir.
After listening to the interesting presentations, we are ready for Q&A sessions. Are there further questions and suggestions from anyone? Also, we'll begin with the felicitation of chairperson and co-chairperson. I would request Laura to please come forward. Thank you so much. Now we are ready for a quick lunch break, and for further sessions we'll meet sharply at 2:45. Thank you.
Hello. Yes, for me it is. Yeah, hello. Yeah, can you hear me? Hello.
So we'll be starting the presentation in a few minutes.
Sure. Okay. Do I need to send you my PPT, or is it okay that I present from here? What do you suggest?
So in whatever way you are comfortable. So if you can share the screen from your side, that would be
That's
better.
what I was saying. So I will share the screen from my side, right?
Yes, sir. Yes, sir.
Okay. Just let me know when you guys are ready.
Yes, sir. So you can share the screen now.
Now? Okay, fine.
Yes, sir.
Can you see? No, sorry, wait, just a moment. I need
We have discussion. It increase the
We'll be having few online and offline sessions. And to begin with, we have Professor Abhishek Datta sir, Dr. Prabodh Halde sir, Dr. Himjyoti Datta sir, Shri Pranjal Deka sir, Ranjit Barwa sir, Jeru Islam sir, Dr. Vivek Srivastav sir. I hope they are connected in online and offline modes.
I have connected, ma'am.
Well connected from all the way from Turkey to us, all the way to you, sir.
Opportunity here in Turkey, which is basically working on the area of perfume and flavor, but on the perspective of chemical engineering. So as a chemical engineer myself, I'm from the Department of Chemical Engineering, as you can see, is basically trying to give some maths, some numbers to this topic. I'm basically a modeling expert myself, so it's quite natural for me to try to understand this area in terms of numbers and modeling. Just a brief overview I will only give to you in this session, because this is a very new work that I'm starting with her. So that's the reason I will not have much results from myself to show, but in the next meetings probably I will be having some interesting things to show from my research as well. So to start with, the brief contextual things that we are going to discuss here very briefly, it's about perfumery engineering. I call it as engineering, not science, because trying to give some numbers into all these things. The points that we are going to discuss is what do we smell, the perception of odors, and of course trying to get some thermodynamics into it, which is basically perfumery ternary diagram, and trying to understand what it is all about in the context of perfumery engineering. I won't touch much about flavor engineering because of the simple reason that flavor engineering is kind of like an extrapolation of perfumery engineering, and there is not much new in this subject rather than what has been already described or discussed in perfume engineering. And also due to the paucity of time, I won't go much into the flavor engineering. Then a brief idea on the statistical data from Turkey, trying to understand why things are interesting here in this part of the world. The same is the case for India as well. Of course, we have an abundance of natural resources to go into this area, and it's $1 billion market for sure. Which is something very important to catch on, understanding the recent economic situations in our countries. And finally, some conclusions and challenges for our work. Okay, so what do we smell? This is a very important point for me, because when I say what do we smell, we basically try to understand things in terms of very qualitative way. We try to explain things very qualitatively, but it is not like that. Ideally, the smell should also be quantified. We should be able to understand what are we trying to smell, and how do we represent the smell. This is basically the thing that I'm going to try in the next few slides for you, and I hope that I'll be successful enough to be able to explain this in a very simple manner. So in the last three decades, scientific methods and for the prediction of the design, performance, and classification of the fragrance mixtures. I'm just gonna take a laser pointer here, it's easier. Fragrance mixtures have been developed. So there has been no doubt that there has been a lot of development in the past few decades on all these things, but things have not been much quantified enough. Unfortunately, that is the problem that we have been facing. So we all start with this basic question, first question that we have. So from this, we try to go into this concept of what you call as a perfume engineering, trying to bring this idea of perfume engineering. So trying to get some idea on what exactly it is all about. So for me, perfume engineering is a branch of product engineering. I repeat, product engineering, which caters to consumer needs for a specific application, and of course to provide a new product which is valuable, which is by following the paths of needs, ideas, selection, and manufacturing. So these are the four principal pointers on which I think that it is important to quantify the perfume engineering concept in terms of product engineering. So
Product engineering, which is a part of the perfume engineering, should normally, for me as a chemical engineer, should involve the thermodynamics, should involve transport phenomena, and should involve psychophysics. This is very interesting that this is where the qualitative things have been all these years, the concept of psychophysics, but this is not enough. So to predict the order of mixtures and of fragrances, etc., we need to have a clear idea about the thermodynamics, the transport phenomena as well, which is a very, very strong chemical engineering or a biochemical engineering phenomenon. So this is again trying to understand that we are trying to predict what is called as the perfumery radar. We try to understand what is all about in terms of predicting odor detection thresholds, classifying the perfumes into olfactory families, and all these things come under what we call as a perfumery, the bigger family of perfumery radar. So when we say about perfumery radar, we now try to understand from this concept of perfume engineering what exactly is in terms of what we call as the perception of odors. Perception. So perception of odors is not a new thing, to be honest. It's not a new thing. It's not a new idea. The earliest mention of perception of odors was basically done by the Nobel laureates, Richard Axel and Linda Buck, which I've just mentioned here as well, in their Physiology of Medicine, in 2004, for the discoveries of odorant receptors and the organization of the olfactory system. So this whole concept of odor perception, it is not a new concept as such. It has been already been there in the field of medicine. And of course, Richard Axel and Linda Buck, they got Nobel Prize for that. So this is not a new concept as you can see already, but the whole idea of bringing this to perfume engineering is rather new. So we can say that this idea of perception of odors is actually kind of like a division of 4 steps. According to me, it's a division of 4 steps. The first 2 steps is evaporation of the perfume, followed by the diffusion in the air. So that's basically where the chemical engineers come into play. Evaporation of the perfume and the diffusion of the perfume in the air until the olfactory system is reached. Of whom? Of us, of course. Until the olfactory system is reached. And then there is the perception of odor intensity and odor character. Odor intensity and odor character are very much strongly qualitative terminologies which rely on human aspects. So this is what I say that it belongs to the psychophysics. And having divided this whole perception of odors into 2 main categories of chemical engineering and psychophysics. When I say chemical engineering, I also indirectly mean biochemical engineering as well, because it's a part of what we are trying to say here. It's all about biology in most circumstances or most situations, sorry. Dividing this into chemical engineering and psychophysics, we also need what we call as the perfumery ternary diagram. This is something that we learned from thermodynamics. Of the guys, those who are here attending this lecture, you might already be aware of thermodynamics and of course the ternary diagrams, something that you probably didn't like in your courses. So this perfumery ternary diagram is something very interesting. There is also perfumery quaternary diagrams, PQDs, which are all kind of helping to understand the dominant order for each perfume composition. This is something very new. This is something very interesting. To understand which order is dominant, why are we even trying to do this? We are trying to do this because we are trying to give numbers. We are trying to give some quantitativeness to the whole concept of perfume. Perfume till now is very psychophysical topic, or had been a very psychophysical topic. Now we are trying to give some engineering aspects to it. So this perfumery ternary diagram, perfumery quaternary diagram, these are the thermodynamical things that we took from thermodynamics to be able to explain all these interesting things for understanding what is the dominant order for any perfume composition. I'll give some examples in the later slides to be able to show you to be able to explain you a little bit more clearly. Sorry. So evaporation and 1D diffusion model is typically what is analyzed on vapor liquid equilibrium. That's basically the first thing to do. You have the VLE, and then, as you know, the diffusion model is nothing but Fick's law. So Fick's law for diffusion gives us the more clear idea about what kind of perfumery performance parameters are needed for developing a kind of like a steady model that can be used for determining the dominant order or orders for different perfume composition. So when we talk about perfume compositions, I repeat that I'm talking about various types of perfume compositions. For example, from shampoos, then the bar soaps, for example, personal wash products such as bar soaps, and of course the home dishwashers, for example. This can look very uninteresting or very insignificant as I discussed, but this actually has a tremendous impact on what kind of products are to be purchased by the buyer. So a buyer will not purchase any home wash products that will have not a very good odor, for example, as compared to others that have a better odor. So this is the whole area that I'm trying to focus on, if you can understand me quite well here. So chemical engineering is nothing but, in this case, it's nothing but as I call it, M square P square E, which is basically the combination of molecular engineering, materials engineering, and of course the two P's which come from the process engineering and the product engineering. So this constitute the whole concept of chemical engineering. And from this, when we look at the product engineering is just what I just mentioned about on the concept of perfumes, is nothing but a combination of thermodynamics, like a repetition in terms of visualization of what I just said in my previous slide. So product engineering, it appeared with a shift in the evolution of the chemical industry from bulk chemicals to very value-added chemicals. These are very expensive chemicals, to be honest, and also these are not bulk chemicals. So these are very limited amount of. There is sound at the background. Okay, so there is these are not bulk chemicals, but very specifically very high added value products such as used in electronics, flavors, coatings, fragrances, and so on. So the perception of orders, when we come to the main idea of this perception of orders, we start with what is called as a Karl's pyramid. Not Charles, Karl's pyramid. So Karl's pyramid is having the as you can see here, it's a pyramid. It consists of what I call as a top node. A top node, it's a pyramid that was devised by Jean Carl. It's a French, been already very advanced in this field. It has been divided into three nodes. One is called as the top nodes, middle nodes, and the base nodes. As you can see, it's in the base, so it's the base nodes. Now, what are these actually? So, for example, the top node is what is giving us the first impact, the fresh impact. For example, the initial impact of the fragrance, typically the citrus fruits, for example, with some possibility that it lasts 15 to 30 minutes. This has been well documented as well. So it lasts like 15 to 30 minutes on skin, for example. This is can be given as the initial, as a first impact or the initial impact, the top note. Then comes the middle note, which is basically the main perfume character, which could be spicy, which could be leather type, it could be floral. So these are lasting 3 to 4 hours on the human skin. And then what is the base note? The base note is basically what is the amber or the musk. There was one presentation yesterday. I think that guy didn't come, and Sid, you presented on his behalf, I think. So what he's looking at, he has a company with agarwood, I understood from Assam, and he is trying to develop what is called as the base notes. So his composition is on the perfumery index, is on the base notes, whereby he is trying to last this fragrance around 4 to 5 hours on the human skin. So again, try or which area you focus on, whether you want to focus on 15 to 30 minutes residence time, whether you want to focus on 3 to 4 hours, or you want to focus on 4 to 6 hours, not 5 hours, 6 hours, you make this Karl's pyramid. So Karl's pyramid is basically a structure of a perfume. It's very interesting. These are very interesting concepts. These are not so well clear in most perfumery industries, as I know, but in France and other advanced places, even in Turkey now, they have started to work on this area just to be able to get into the let's say to the domain of very high level perfumery business or perfumery domain. Anyways, so as I was saying to you that this is very important that we understand that the mathematics behind the perfumes. So here what we basically do is we go to the vapor liquid equilibrium. You may know this formula as well. This is Raoult's law, nothing else. And what is done typically here is that it takes into account the mole fraction and of course the saturation pressure. I'm not going to go into details of this, but what we are doing is you try to model the vapor liquid equilibrium, and using this concentration, which is this C of I over the G phase, this concentration helps us to, this is of course the ideal gas law, as you know. And then from this, we try to develop what is called as the OV, which is the order index. Okay, so this is the order index, or rather we can call it as the order value. This is the first mathematical term that you can use to express your terminologies, you know. So these are very important. AMI is the molecular weight and so on. So this whole formula is actually what you are trying to put as a quantification form in terms of your perfume, whereby you're looking at the evaporation of the perfume, the one that I just mentioned to you, the base notes, the middle notes, and the higher notes. So which is basically the initial impact ones. So the middle and the top, middle, and the base notes. So having this equilibrium, having this VLE, the vapor liquid equilibrium, and contribution methods, I'm not going into the details of that. These are all well documented in chemical engineering, and if you're interested, you can also look into all these things and find out what exactly these group contribution methods are. So based on the concept of, or based on the idea that we have derived from the vapor liquid equilibrium, we are now going into, or we are now trying to design the most important part of the perfumery engineering, is basically what I call as a PTD, the perfume parameter. In this, it's not mentioned here, of course. It's an important parameter here because it is the solvent, in this case the ethanol, that's try to dilute one determine which is the most dominant order in the whole combination of the three order zones that you are. Here comes from chemical engineering ternary diagrams principle using mixture compositions. I guess all those well using mixture compositions. I guess all those of you have done your bachelor's very well. You are very for each liquid composition, the order values are calculated using what is called as a strongest component model. There's a formula for that. I'm not showing you here, but there's a formula for calculating the strongest component model, the SCM we call it. And then based on that, you can map the triangle in regions with different dominant orders. Okay, you can do this mapping in the different dominant orders. This basically clears up your PTD, the perfumery ternary diagram. So this means that a little bit of a homework here, that if you want to calculate the distance of any point from the triangle to the AB line, you have this line, you have this line, this is the line, the AB, AC, and the BC, which means that that your basically your composition lines basically, and then any of the point that you can draw here will be a combination of both A, B, and C, which is also mentioned here in terms of the x prime of C, as you can see here. So XA, XB, and XC, X of A, X of B, and X of C. This is basically the A, B, and the C line. So X of A, X of B, and X of C can be represented directly, but be aware this is done in a solvent basis form, a solvent-free basis form. I'm sorry. So this means that there is no ethanol involved in this theory. Okay, so there is just combination of A, B, and C. We are not involving any of the solvent in this picture yet. So similarly, the distance from a point to AC gives a composition. Okay, this is already I've shown. There's nothing to explain here. So this is basically what I call as a solvent-free and quinary mixtures, because it's not always ternary, as you know, it can be more than that. So just to allow some visualization, the only thing that you need is to have a database of the vapor pressure of the perfumery raw materials, which is very important. We call it as a PRM. Database of the ODT, the order detection threshold, that's also a database is needed. And of course, a tool to calculate the Henry's coefficients or the activity coefficients for the PRM. These are the three basic things that you need in order to develop your own quaternary or the quinary mixture diagrams. So the PTT tool allows us to analyze the effect of the non-ideality of the perfume mixtures on the order zones, as well as the description of smell with order value with intensity from the Stevens power law. Okay, so having said this, a small example can be seen here. as you can see here, there's a small example whereby you can see that by changing, this is the same composition, or rather the same base notes, which is basically in the left you can see limonene in square form, the B is the geraniol, which is in the triangle form, and the C is the vanillin, or the vanilla, the vanilla flavor is from there, vanillin. And this at this point they are all at a combination of this in terms of the diagram, but when you add a sufficient amount of the solvent, so it basically what happens is that if you change the base note, if you look here, if you change the base note a little bit towards from the vanillin section, and if you go towards what I call as the tonality, so basically if you move towards the C portion, which is nothing but if you move from the geraniol to the vanillin section, you will see that you're actually depleting in terms of the vanillin, and entirely a new note which is basically comes, and the ethanol you get a more flavor in that. So this is a kind of like a tool to predict or to mask some flavors, which is done in a very scientific manner. You can mask some flavors, or you can unmask some flavors using a certain amount of solvent. This whole ternary diagram is not from me, it is from this paper, ASAG Journal 2009, Texera and et al. Okay, so having said or having explained you guys, let's a little bit about the whole idea of how to get the ternary diagrams and the quaternary diagrams and their importance in flavor. These are all done very scientifically these days, and for this we typically use we start with the default, probably have known by now. This is nothing but the Fick's law of diffusion, and again based on the subsequent maths that you do, you can actually draw this kind of diagrams, the OV diagrams, and the distance up till where the order evolution can reach from to a certain section, depending on what kind of perfume mixtures you are using. So every perfume mixture, including the base note, the middle note, and the top note, will have a different diffusion model. Very important. Okay. So yeah, and again, based on the perfume diffusion model, again, you can come back to your diagrams. As you can see here, the evaporation lines are actually play with your composition. You can play with the solvent rather. In this case, they have played with the vanillin. As you can see, the vanillin flavor changes a little bit between limonene, geraniol, and ethanol, and you can actually play around and you can create your own, try to create your own flavor, your own fragrance, I'm sorry, or you can also unmask or demask the fragrances. So perfume diffusion model, the 1D model can also be used as a 3D model. There's no need to go for a one-dimensional flow. You can also go for a radial flow, which is a 3D model. I'm developing currently one-dimensional model for this. And basically, you can try to understand that this is all these informations are needed to finally go back to this ternary diagram or the PQDs in order to develop the ultimate model, which is basically a combination of thermodynamics, as I've just mentioned, transport phenomena, and psychophysics. Okay, so not like take much time. Now a little bit of storytelling, let's put it like that. Turkey, as you know, I'm right now in Turkey and I'm teaching here. So obviously I'm looking into the Turkish industry for this. There is very high demand of Turkish natural resources to make perfumery and flavors and this kind of things, you know. So basically the cosmetic products, I call it as a cosmetic products. This is going at a severe high rate. I mean, people are these are in huge demand. All the things that I've written down are actually based on the Turkish export market report, which is 2021 report. And as you can see here, that everything, there's a huge increase starting from this is the last 2 years, rather 3 years. Everything is on a high demand because of the fact that Turkey is investigating much into not only into the quality of the product, but also in the science of the products. So they're trying to get into the technology, so learning as well as for India as well. And of course, lessons from India that can be learned for the Turkish manufacturers. The market here is growing at an average of 10%, according to, which is very important. I think yesterday I was listening to some of the sessions, and one of your presenter was looking at or talking about this this regulations, and I have to tell you the most stringent time to explain all these things here, and is also not the purpose of my presentation. So exports are into 199 countries, I'm sorry. The biggest one is going to Iraq, USA and Iran. These are the main customers, around 1.1 billion US dollar, which is I think a pretty good market, pretty good amount. Within the scope of this Turkish export strategy 2023 reports, they have made a kind of like a clear vision of what is the target, which countries are they going to target to, and what would be the ultimate revenue that we're going to generate. And this is something around 3.3 billion US dollar, and I'm pretty sure that they're going to reach this target. So things are, as you can see here, they make very, very short-term targets. Targets are not 5 years, 10 years. Here, this target was made in 2021 on 2023. So this is a very serious issue that they looked into, and they're now reaching probably around this margin already. I mean, I will get the report anyway in 2023. We can find out what's happening. Anyways, so some of the things I wanted to mention here as well, that why these Turkish products are very popular in Europe. One of the main reason is that they have not only that they have understood the market very well, they have also gone into the science, or they are going to the science and technology, which is very important. There is a large young population and rapid urbanization. They are one of the principal growth factors. You need to be aware that there is a big market of Turkish perfumery industry right inside Turkey as well. During the COVID, this product, Turkish cologne, was extremely sought after in whole of Europe. The Turkish cologne made a huge market because it gave something beyond alcohol. There was not just 80% alcohol in it. There are also some citrus extracts, and that gave a whole new dimension to the industry. You know, this disinfection industry in Europe, and the Turkish cologne was. I mean, this was hot cake. I know that it was extremely well sold, and this has been a very, very nice niche product for the Turkish perfumery industry. As I told you that I will not tell much about flavor engineering, because the ideas are basically the same. We derived the same concepts of transport phenomena, chemical engineering, back from the perfume industry to the flavor industry. So idea is to basically define what you call as flavor detection threshold, or FDT, which is the lowest liquid concentration of component I mean, as a component in a multiphasic mixture, which is detected by the retronasal route. So that's basically following the same ideas. I'm not much working on this, so that's also the reason I cannot say much more on this. My main focus is actually on perfume engineering, and basically the idea is the same. You bring up the same concepts, and at the end you try to work on the same principal parameters in order to get the same ideas, bring the same ideas, and basically extrapolate it for flavor engineering. There are some conclusions for this talk, and also importantly some challenges ahead. So there has been some progresses that has been made already right now, which was not explained or not told. Some of the challenges that are ahead are prediction of the order of chiral molecules and the prediction of the order quality of molecule, which may change with this concentration. This is a very challenging topic to deal with. Here, one of the possibility is to use artificial intelligence, and you guys probably know now that European Union, as well as the big section of the European Union, as principally they are focusing on what we call as the digitalization 4.0. So with the advent of this fast computing facilities, digitalization, and the cloud data storage systems, there is a high possibility of integrating artificial intelligence with perfume design. I'm going to try that myself in our group, trying to understand the relationship between a structure of a molecule and its order, which we call it as a QSOR, the QSOR relationship. It's very interesting. At the end, it's also important not only this, but also to go to the technology for the extraction process. There's a whole section I can talk on this, but it's not for today. I'm working also on simulated moving bed technology, which is a little bit deviant from the fixed bed moving technology, which is basically chromatography. So it's an improved chromatographical technique. Since some of the things are patent pending, I cannot disclose all of the things here, but the idea is to borrow some ideas from the chromatographic science and try to use this, what is called as a simulated moving bed, and improve this for the extraction process related to our perfumery industry. Okay, that is all. If you have any questions, I'm open to answer them. Thank you very much.
Abhishek Datta Saab, fantastic presentation.
Thank you. Thank you, sir.
Very good presentation.
I'm happy that you liked it. I hope everybody says the same. Thank you, sir. Well, if there are any questions, I'm happy to answer.
Anyone have any questions?
Okay, if there are no questions, guys, I know that it might be intimidating to ask here. Please feel free to ask me questions. Please feel free to email me. If you just Google Abhishek Datta and Turkey, you will probably find me very easily. And just ask me questions. Of course, with Professor Sinha, I'm starting a collaboration very soon. So we know each other since a long time, since our Jadavpur University days. So Sid, and I call him Sid. So Professor Sinha and me, we are going to collaborate. So I mean, you guys can definitely contact him or me for any kind of discussions. We are also planning an Indo-Turkey. Maybe Sid, I should take this opportunity to say that in the next few months, we are going to plan an Indo-Turkey kind of like a one-day, get together. Meet, know each other, greet each other kind of a meet, whereby we'll try to bring some of the industry experts from the perfumery industry and some professors who are working on this from my side, and Sid is going to do also from his side, and we're going to combine all these things to basically benefit both the countries. That's the idea, not just to benefit Turkey or just to benefit India, just to benefit both the countries, because there are huge potential for our two countries in this perspective.
Dr. Abhishek, can you hear me? Hello.
Yes, I can hear.
Yeah, good afternoon. This is Sanjukta from the School of Agro and Rural Technology. A very good presentation, first of all. And I have a very small question. Where does the perfume industry based on natural products in India stand as of now?
Could you repeat? I didn't hear you clearly. Did you mean that what is the present standard of the perfumery industry in Turkey as compared to India? Is that what you're saying?
Not compared to India, only in India. Perfume industry based on natural products. Where does it stand in India?
You're talking about in India. Where does it stand from the Indian point of view?
Yeah, I'm talking about where does it stand in Indian market as of now, so that we can understand the gap, and this will help us, plan many things.
That's a very good question, actually. That's actually what Professor Sinha and me had been trying to understand. I understood one thing here, that the countries that are blessed with natural resources is, for example, India is one of the countries which is huge biodiversity. There has been almost probably, in my opinion, and probably it is not wrong if I say that I think less than half has been explored so far. It's a huge market, a huge possibility. The market is huge. But again, Madam, I want to reiterate that why I'm focusing on the engineering aspects, because until and unless the European, because eventually our market will be Europe. For the Turkish manufacturers, for the Indian manufacturers, whoever, doesn't matter. But the European manufacturers, I am an European citizen now. I have a Belgian citizenship. I have been there for 15 years. I know that European customers or European clients, until and unless they are very, very clear and detailed about the perfume, they are not going to buy it. As simple as that. And this is where I think in India, the manufacturers need to be aware of that. We need to make this extra jump. It is not just good smell anymore. If you understand what I'm trying to say, it's not just good smell. How good is the smell? What is the quantification? This is where we need to make this. And in my opinion, most of the private companies probably do this, but they don't do it in a large scale. To bring out the entrepreneurs from our country, I think it is very important that we indulge into this kind of scientific knowledge building. The IITs are there perfectly for this, and a lot of other institutes also are there. Center of Excellence, as Professor Sinha mentioned yesterday. You have to go for a scientific knowledge building in this area. There is not just psychophysics anymore. There's much more than that. And this is what I'm trying to say as the knowledge gap. The knowledge gap is also the market gap, is also the revenue gap, is the same gap. So this knowledge gap needs to be fulfilled. Right now, Turkey has understood this gap, probably because being so proximally close to Europe. I mean, they are sharing borders with Europe. Probably the reason for this that they have understood this and they are now taking this leap inside. So they are forcing the scientists or the engineers like us to help the local industry. As you know, the Sparta, which is very famous from the Roman times. Sparta is a little city, not very far from here, actually, from Izmir, where I live. And this is famous for lavender. The picture that I showed you in the beginning is for lavender. This picture is you can see that the market of rose and lavender. I mean, this is amazing. The tulips, the original tulips that Netherlands is that Europe has, especially the Keukenhof in Netherlands. It's originally came from the Ottoman times, from the Turkey. So all these things they had been giving to Europe without understanding many of the things. However, right now they have understood that there is a lot of maths and there's a lot of technology behind that, and they are going into that step. So I think this is, if I understood your question, and what you understood, the knowledge gap or the revenue gap, which is basically the same, they go hand in hand. I think this is where we need to plug in. This is my opinion.
Thank you, Dr. Abhishek. Thank you.
So, Dr. Abhishek,
Thank you.
I would like to ask you one more question. Compared to when you are talking about biodiversity in India, we have a lot of good fragrant flowers. And when you are saying about your top note, middle note, and base note, so if you know the active ingredient or the fragrance kind of a compound, is it possible for adopting your model or the concept? Of course, it
Absolutely.
is.
Absolutely.
So can you quantify
Absolutely.
the
Yeah.
things which is the which could be the probable distribution of that fragrance into top note, middle note, and base note, so that a normal manufacturer can actually plan accordingly. Is it possible?
Absolutely, madam. Absolutely possible. You can, as you correctly said, and indeed, I missed to say this point in my slide, and very nicely that you mentioned. If we can find out these active ingredients very correctly, you can design your perfume, thereby retrace it back and design your flower in order to get that. So you see, it's a step back, not a step forward. And this is very important and very interesting. So you go forward to understand the knowledge, and then you come back to the field, and then you explain to our farmers how are you going to design. It will not take an overnight. It is not going to be so easy to say as it is. As you know, I mean, it takes time to do all these things. But this knowledge building is needed.
Can we use AI very efficiently? Create a model. The fragrant flowers of different countries will have a different kind of a notes, depending on our India is a very tropical country which has naturally the strong flowers, which is whatever it is a nature's gift. So we can have your AI or that kind of a simulatory models. I do not know whether they are existing, and if it is so, then why don't we start applying and see how it is? So that is my other.
Very good question, madam. Very good question. I have a team also. I have a team with one of my colleagues, postdoctoral researcher. She is also working on AI. But as you know, I always go for a humble approach. I'm not there yet, to be very honest. I'm not there yet. We are developing, trying to do it as good as possible. Once we are ready, we'll definitely bring it on. And of course, being connected with Professor Sina, it makes things much easier. But definitely, AI is one of the answer to go into this. Definitely, there is no other. I don't think there is any other solution yet. Otherwise, we have to go to the molecular biology of everything, and things get, it's like basically developing a new pharmaceutical product, and it costs a lot. So I think AI is definitely the way to go on to make things a little bit quicker. But as of now, honestly, madam, I have not the model. I do not have the model, and we are working on it. We need a lot of data. We can definitely collaborate, and we'll try to, if you can give us some data, we'll definitely try to help you. Also learn ourselves, not just helping you. We'll also learn ourselves in order to make very strong, robust AI models, so that this interesting perfumery indexes and these diagrams can be eventually built and designed.
Thank you. Thank you, Dr.
Thank you. You're welcome.
Thank you so much, sir, for your valuable insights. We have another session, online session. Now I welcome Dr. Prabodh Maldi, sir, who is head of regulatory affairs at Marico Industry. His area of interest includes food science and has 16 years of experience in the domain. Over to you, sir.
Can you make me admin? I want to show my presentation.
So you can share the screen. So you can.
Hello.
Yes, sir. So in your screen on the towards the top right corner, there's a share option. You can just click on that and you can share your presentation.
Am I audible?
Yes, sir.
Yes.
1 minute. There is a blackout in my computer. I don't know what has happened.
Dr. Hadi, in case if you have difficulty sharing from your side, you can send us the PPT. We can do it from our end. It can also.
I have been logged out. Okay. Good evening, everyone. Sorry Actually, pranam sabko. It was a fantastic presentation by Dr. Datta. Amazing. And happy to be here after a long time. Professor Mishra ji is our guide and dear friend. And thank you, Siddharth ji, for inviting. If you are ready. Abhishek ji, there are so many things to learn from your presentation, sir. I would request if you can share the copy of presentation, if you don't mind. It is all about science. And
thank you. We'll do, sir. Definitely, no problem. I'll just get your email ID, sir, from Siddhartha.
Yes,
We'll be very happy to keep in touch with you. Of course.
Yeah, definitely.
I mean, the whole idea is to, I mean, the whole idea matters. I realized. I mean, that's so important.
Absolutely. And I like about your engineering formula for the top note, middle note, and the dilution aspect of the perfume.
Yeah. Actually, I mean, I will tell you, sir, when I was in India, I did meet a very elderly man, a guy from Bangalore. You know, he was telling me कि मेरे पास तो सिर्फ knowledge है और मैं सिर्फ अंदाज से करता रहता हूँ. I do it with an andaz, you know, and then I realized that, of course, he will not be able to transmit his knowledge, or even if he does, only to his sons or daughters. So if this knowledge needs to be quantified and, you know, extrapolated to the greater community, which is the most important. Anyways, I'm looking forward to your presentation, sir.
Yeah. Is my presentation screen is visible now? Can somebody respond to this? Sir, is visible?
Yes, Dr. Prabodh, it is visible.
Thank you for invitation. And this is International Seminar on Medicinal Plant Production and Advancement in Herbal Medicine, Nutraceutical, Cosmetic and other herbal products, Formulation Technologies. It's an apt and timely efforts. I congratulate the organizer. I will take 20 minutes. And I am working as a head of regulatory in research and development R&D of Marico Limited, having 28 years of experience, food and cosmetic and nutraceutical. Today, nutraceutical is a coin term for the food, but before FSSAI, there was so much of confusion where this nutraceutical belonged to, whether it is a food, whether it is a medicine, whether it is Ayurveda medicine, whether a drug. But now FSSAI has given the solution. It is being placed under food definition. Since a lot of students have joined this conference seminar, I would like to know how many have done in your life, though you are a teacher, professor, watching the movie one day before the exam, traveling in a general compartment of the train without reservation, eating the fruits without permission from the nearby tree, and going to the unknown places alone without friends and mobile. If you have done in your life, kindly ping me in the chat box saying that if you are online, if you are offline, I just want to hear the voice you have done. So Siddharth sir, if offline, can just confirm that anybody has done in the offline? Can you raise the hand?
Sorry, I missed the last few minutes.
So what I'm asking, the picture which are displayed on the presentation, I want to know how many of you have done this in your school time? Watching the movie one day before exam, yeah, eating the food.
All of them from my side.
Almost all of us.
Great. Okay. Friends, I just want to tell you, though you are in IIT Kharagpur and most of you are intelligent, average than the intelligent, because IIT is the dream college for many. And life is not about only study. Life is about study and application. Ex-president of I was a president. I always wonder, whenever we study any subject, always there are the references of Western books. And I wonder, why only Western book? Why not Indian? We have a scientist, we have a brain. We have 1.4 billion population, and our culture is there from last, if not less than minimum 30,000 year, 50,000 year. And we have progressed in many areas: space research, engineering, chemical. Then in the food science, Ayurveda also, we are very well, medicinal science also. So particularly food, many of us are not aware. King Nala Damayanti Nala was one of the best cook and one of the best food technologists, probably the first food technologist of the India, minimum 10,000 years back. His book called Nala Paka Darpan have given a lot of recipes, veg and non-veg. Have talked about hygienic requirement of the people working on the food area. And I was amazed to read those books. Ma Sita was one of the best chef. Her tribal recipes are very famous. Ballav Bhima in Mahabharata was a chef in Virat Kingdom, and he is the inventor of the avial dish, which is being consumed by South Indian. Shah Jahan was one of the very famous cook, and he has given a lot of recipes, more than 350 recipes, including gulab jam and lot of biryanis. You may get surprised to know that sambar, which we eat with idli and dosa, was invented by mistake when Chhatrapati Sambhaji Maharaj visited his cousin's place at Tanjavur, at the south in Tamil Nadu. Tanjavur is the place where IIFT Tanjavur is there, called as the Niftam Tanjavur. The king of that Tanjavur was a Shahuji Maharaj. He wanted to offer Sambhaji Maharaj with the amti, which is a Marathi dish. But the chef could not prepare amti properly, and you need to add dahi into it. He added tamarind water, and the dish by mistake has been prepared, and that was named as Sambhaji's Ahar, called Sambar. It's a serendipity. What I'm trying to convey, that so many good things happen in India. Unfortunately, they are not part of syllabus, or our interest in the study is also not less, and we only been copying the West. I want the brain of the India to work on Indian culture, and if we do not respect the Indian culture, who will respect? We are, every person on the earth, 1/6 is Indian, and we need to respect our culture. I'm not particular a religion. I'm saying whatever the religion, whatever the culture of India, we should respect and we should study. The whole food processing industry has been changed until the invention of the particularly pasteurization, 1864 by Louis Pasteur. The application of the principle of the scientific term was coined by Charles Olin Ball in 1928 when he started working on the thermal processing of the food. Till then, canning technology was not accepted. First World War is the area when the canning technology has been used, and that was a game changer. As you are aware, bigger the crisis, bigger the innovation. Both the World War, first and second, you will see maximum innovation has happened. And most of the patent has been filed during these two crisis condition. And COVID was the third crisis condition, where a lot of innovations have happened. And we do not work unless there is a kind of crisis. So the whole aspect is that in the food processing, food technology, agro processing, Ayurveda technology, when such innovation can take place, the way Ola has changed the entire taxi business, Make My Trip, Facebook, Paytm, these are the smart innovations. Internet itself, can such innovation happen into the food processing area? The people sitting in this conference, online, offline, I really appeal to you. Can we do this kind of innovation and change the face of the industry? It is very important. If we do not innovate, we will evaporate. What has happened to the Ambassador car? What has happened to the Kodak film? What has happened to the pager, the HMT watches, Ravalgaon chocolate, Duke's Cola? They were number one of their time. They were doing very well. In spite of that, where are they today? Food processing is the most important thing for the country like India and populated country where the population is high, because you are directly connected to the farmer, well-being of the farmer, value addition of the farmer, and giving money back to the farmer. It is not the mere business, it is a social enterprise. Food processing is so important. As you say, nutraceutical is something which is in between food and pharmaceutical. And India is going to be the diabetic population and heart disease population by 2030. Prevention is the most important thing than the cure. Good health. After COVID, everybody want to look good. Everybody want to feel good. And for that, food is the most important aspect. Medicine is only being given to the curative for the primary health care, but preventive health is the food and nutrition and nutraceutical. It is very important to see the food rather than going into the medicinal view. During COVID time, we have seen that the nutritional upliftment and the quality of the food which we ate, because of that, our actually consumption of the medicine was as low as possible. 6 months we were under the lockdown strictly. Hardly we visited any doctor, but everybody was fairly healthy. That shows that if you take care of your health by doing a regular exercise, yoga, and herbal medicine or home cooked food, things will be better. So this nutraceutical word has been coined by a pharmacist called Stephen Day in 1989, and then it has been marketed all across globe. Today, if you see entire globe, there are many drivers why the nutraceutical will do well and why the herbaceutical will do well, why the Ayurveda food will do well, because people want to take care by prevention rather than the after happening of some disease. So prevention is better than cure. Singular family, rising the income, urbanization, these are the things which actually are the driver for increasing the growth of the nutraceutical business in coming days. Research and development in any of the food or pharmaceutical company are based on two aspects: consumer backward, technology forward. When I see what is consumer backward, means there are some unspoken needs of consumer. Consumer are looking for some solution, and you understand those nuances. Do the market survey, do the consumer insighting, and give the solution through your R&D. It is called as a consumer backward. This is the best way of launching the product. Technology forward means you develop the technology and then try to fit that technology, giving solution to the market. But technology forward R&D is very expensive. But there are some examples. Technology forward ideas means Apple phone. Created it and successfully able to sell the concept of touchscreen phone. In case of food, the concept of Tetra Pak, having milk at room temperature 6 months. Consumer never demanded to have the shelf life of the milk for 6 months. But Tetra Pak has created this technology and successfully implemented. These are the breakthrough consumer technology forward ideas. Consumer backward means there are many products which has been given through the consumer interest and consumer mapping, like Maggi. After the lunch and before the dinner, in between the snacking time, we have to give something. So that particular snacking time, Kellogg's. So all these examples are the consumer ask for this requirement and companies have responded to that. After the COVID, what I have seen, and this is not my copypaste.com slide, this is very important slide. What I have understood that these are the future trends which would be coming to the food industry in next 5 years. Personalized nutrition, organic, natural, traditional, vegan food, e-platform to increase the change, the phase of the industry. Exports. China is exporting worth of 96 billion, 96,000 crore of total export value of the China for the food processing. India is 16,000 crore. And I see that there is a China, because of the COVID, many people don't want to go to China. And there is an opportunity for India where there is an export potential subject to quality and quantity. Home cooked food, plant-based meat, intermediate product. Because of the Google and YouTube, many male members are preparing the dishes. They don't want to prepare from the scratch. So they just want to bring some intermediate product and make the product final product. So ready to cook kind of intermediary product intervention have increased. Ayurveda food is a very great example. Packaged versus loose, cloud kitchen, artificial intelligence in the food processing, big data, machine learning in the food processing, post-COVID food, functional food, immunity boosting food, mental health food, sleep disorder. Sleep disorder is one of the area. A small country like Denmark and South Korea, they are the two countries where sleeping pill business have increased because of many people are suffering heavily for the sleep disorder, and better than medicine, the food can work. And there are the actives, as my earlier speaker talked about, bioactives. There are many bioactives which can work beautifully on your neurological properties for getting a good sleep. Ashwagandha is one. Food, ready to eat, ready to cook, on the go food, high protein, multigrain, cereal science. So next year is a millet year, and multigrain is the area which really can grow. Functional food, nutraceutical, cut vegetable. These are the areas which I see the huge future. In India, if you see minutely this slide, drug at the bottom and the food on the top, the driver of the food is a taste, and for drug, there is no taste, but it is the result. Cure, diagnosis, mitigation or prevention of the disease or disorder. But the space in between is called as a nutraceutical or health supplement or food for special dietary purposes. The distance between the food and drug is narrowing down. Food is basically for the taste, no claim. Now, the moment you enter into the nutrition food, functional food or risk reduction, nutraceuticals, there are many claims. And that is a space which is growing. In Japan is the only country where nutraceutical penetration is as high as 360 pills per year to the manufacturer and the farmer. So I see there is a good scope for Ayurveda, herbal nutrient food, Ayurveda food, nutraceutical or health supplement. I will decode the entire section now. Health supplements now finally are part of the FSSAI under section 22. What is the basic difference between nutraceutical and conventional food? Conventional foods are the mass consumption food. Nutraceuticals are targeted. Conventional for the taste and satiety. Nutraceutical are for disease and disorder. In conventional food, there is a serving size. In nutraceutical, there is a dose. Conventional food, they are in conventional format. Nutraceutical would be in the capsule, granules, tablets, and syrup. So this is the difference between conventional and non-conventional food. These are the total area of the food which falls under broader category called nutraceutical. Starting with the health supplement. Health supplement example means protein supplement, vitamin supplement, multivitamins, all will fall under health supplement. Then the nutraceutical. These are the isolate extracts of the or concentrate of the nutrients. FSDU is food for special dietary uses. Like if somebody has a lactose intolerance and you are making a food without lactose, and that is for satisfying the particular area because there is a deficiency of particular thing, and then it is a special dietary purposes. Food for special medical purposes, Means under doctor's recommendation, if somebody has some disease and medical condition and doctor has prescribed particular a food, it is a food for special medical purposes. When the food can be injected through the tubes and can be put it through the tubes, not only oral. Probiotic, prebiotic. Probiotic is a very important aspect. Growing prebiotic, having particular fibers which are good for your growth of the microbes in the gut health. Let me tell you friends, gut health area is one of the very ignored area, but gut flora is a second brain. If you just want to know how is your health, just see that how is your gut. A reflection of the gut is on your stomach. If your stomach is good, then your gut is good. If your gut flora is good, your stomach will be good. If your stomach is good, your health will be good. If your health is good, your mind is good and you are happy. That's why under Ayurveda, if you go to the traditional Ayurvedic doctor, they first will cleanse your gut before giving any medication. 3 days, they will ensure that your gut flora is flushed out completely from the foreign bodies and the natural flora has been regained, and then they will give the any medicine. That is the theory under Ayurveda. So just wanted to give you a good gut is a good health in short. Specialty food and novel food. These are the nutraceutical aspect. Then there are various schedules. Schedule 1 is a vitamin, Schedule 2 is amino acid, Schedule 3 is vitamins and minerals for FSDU, Schedule 4 is a plant and botanical. There are 392 plants and botanicals which can be used in the food thanks to the FSSAI. So you can make any product using the botanicals or the plant ingredients. Friends, everybody has a turmeric at your home through the masala box, and Indians are actually liking this masala. But let me tell you, our ancestors were very smart. They incorporated this masala consumption purposefully through our diet. And this masala box is nothing but the factory of the nutraceutical at home, because it contains antioxidant. Antioxidants are good for fighting the free radical. Fighting the free radical helps in immunity increasement and also helping to control the disease. Mustard seed, cumin seed, turmeric powder, red chili powder, coriander, coconut, home grade garam masala, amchur, all these are depending on the seasonality, depending on the geographical condition. Your masala content may change, but finally all masala together gives you the taste, gives you the volatiles, and gives you the flavonoids, gives you the various aromatic actually characteristics and antioxidant, which is very important for your health. This is one study which actually have just found out that in US or in UK, they do not eat the masala the way we eat. They do not eat the turmeric the way we eat. But look at the dementia problem, Alzheimer problem in the US versus Indian population. Closely 2% of the US population is suffering from Alzheimer. Alone Alzheimer. And India penetration of Alzheimer is just 0.3%. Many of you get surprised, like I am only talking about Alzheimer, but there are many other dementia and the neurological problems. If you put together, the percent population who is suffering from dementia is 7.2% in US, vis-a-vis in India, close to 1.3%. And these neurological are directly linked with the function of some kind of antioxidant lacking, and spices are the one who are responsible for arresting these issues in India. So friends, spice consumption, if you infuse in the spices in the proper doses, it works like a medicine. These are the nutraceutical, nothing but nutraceutical. So we should thankful to our ancestors who have told us how to eat the spices. Look at the technology. You have to boil the oil and then you put your spices. So oil soluble ingredient in the spices get infused in the oil, and through that oil it goes into your kind of vegetable. What kind of infusion is this? It is all about science. So this is an amazing thing. Friends, post-COVID, if we do not innovate, we will evaporate. So whether you are a technologist, whether you are an academician, whether you are an industry, whether you are a scientist, whether you are a student, future is today uncertain. There is a VUCA word. There is a terminology called VUCA because volatility is everywhere. We don't know what will happen to the share market. We don't know what Russia and Ukraine will continue. So everything is uncertainty today. But do not worry about the future. Focus on the present. Research and development jobs will increase because of the post-COVID. Everybody is working on the new product. Control on your expenses. Do not wait for a good package for the joining. Wherever you get opportunity, start working, increase your strength, help others. Help others is the most important thing. After COVID, money will not take you, but the relations will definitely take you to the heights. Sharpen your skills. Startup is a good option. Innovate, innovate, innovate. Ayurveda Ahara is recently has been launched by FSSAI. I am a part of their committee on Ahara committee by FSSAI. And this is something can increase the export. Ayurveda otherwise is banned in outside India, but Ayurveda Ahara under FSSAI is allowed to export. So I see the big future for Ayurveda Ahara food. India can become an export house. Make in India can increase contribution in creating Ayurvedic Ahara brand in India and internationally. This has been launched by 6th May 2022. As I said, innovate, otherwise you evaporate. Friends, there is a great experiment by Dr. Kurt Richter in 1950. He placed the rat in the water in the beaker. After 58 seconds, the rat was supposed to get killed in the water by sinking. He removed the rat from the water and kept it outside. Rat understood that somebody is taking me outside and giving me a life. Again, he puts the rat back into the beaker. Now this time he found that rat continued swimming for next 30 hours in want of somebody to take the rat out. This is called as a belief. So the mind works beautifully. There is a hope, there is a belief. You can do, you can bring the mountain back, or you can do the uncertain things which are impossible. So the message to the student, have a positive hopes. The life is really good and do not worry about future. You can do the miracles subject you believe that you can do the miracle. This is very, very important. I always tell all the students, by the way, I'm doing my second PhD from MIT Pune, and I have published 2 patents. I'm the now publisher of 2 patents on my PhD, and it is all possible. So I just want to tell you, the students and the professors, it is very important to have a belief. Unless we come together as the industry and the academia, we're not able to do innovation. Whether it's a Turkey, whether it is a US, whether it is a UK, we have to bring academicians and industry together, particularly after COVID, because we are not perfect. This is for the students. Always there is a question during my PhD, should I go for research paper or patent or only research paper? I personally believe, and IIT Kharagpur is one of the very pioneered actually institute. I request you guys, you are an intellectual hub. Please, please, please go for the patent. As I said, I am an inventor of primary inventor. I'm the first inventor for the 3 patent. And Mr. Datta, I will share with you. I have 1 patent on the aroma also, the particular diffusion technology. So which is not in the food, but in the area of aroma. So I personally believe that if your idea is a real invention, and if you believe that people have not worked on your idea, go for a patenting. If you believe your research work is not innovative, you can go for research paper. But importantly, I suggest that amongst the 10 PhDs or 10 scholars, at least 1 patent should be there. Whether you go for patenting or publication, your research should have some commercial utilization. If your invention doesn't have commercialization, then it is a value of death. No point of working on innovation or invention or that in a particular research. So I have prepared a 2x2 matrix. Most of the professors who are sitting here, you can apply this. On the x-axis, research. On y-axis, commercialization. It could be societal use, commercialization, farmer use, whatever. So if your research, high on research, low on commercialization, then it is a long-term research. It is in yellow window. If your research is high on commercialization, but low on research, then you are on the, again, a yellow zone. But your research is low on commercialization, low on research, then you should avoid doing that. It is a copypaste.com. Ideally, 2x2 matrix, high on research, high on commercialization is something what we are looking at. But every research cannot be on the green matrix, green color. There will be some yellow, but avoid the reds. This is for the student. One of the study in London, which they found that the successful CEOs comes out of the bed just in 30 seconds. And Dr. Abdul Kalam has said, dream is not what you see in the sleep, but dream is something what doesn't allow you to sleep. I have a question for the audience. How much time you take to come out of your bed when your eyes are open in the morning time? Can you put into your chat box, or I request Siddharth sir, can any volunteer say that how much time they take to come out of the bed? Can you hear some voice?
About a minute. And that's why I'm not sleeping.
Hello?
Anybody who wants to answer the question? I think there are some entrepreneurs sitting here. So you must ask them as well.
Okay.
Okay.
Also, the student, I always appeal that I have started recently when Prabodhwani, it is on my LinkedIn. I completed 20 such episodes, 4 to 7 minutes of my talk. Book reading is the most useful technique, and one should read the book. And for the student, you should read at least 12 new books per year. And I am not categorizing which book to read, whatever the book reading. Book reading enhances your capability, intellect, and wisdom and vision. More you are linked to your Facebook, Instagram, and phone, your brilliancy reduces because everything comes ready-made. In the book, you have to do the imagination. So my appeal to the student, I know that you must be reading the books, but please continue this good habit of reading the book. Very, very important. What happened? We asked God, give me a good job. What is the requirement? I want a AC cabin and a lot of money around. I asked the God, I want to control hundreds of vehicles on my fingertips and thousands of drivers will listen to me. God says, Tathastu. God says, yes, boy, I will fulfill your dream. But I do not give a good command to the God. I just required some aspect. And what is the reality? I become the security guard at the ATM where it is a AC room and thousands of dollars around me, but it is not belong to me. Same way I become a policeman controlling the entire traffic on my fingertip, but none of the car belongs to me. So the message from this is, if you really want to do something, hard work, there is no shortcut. Respect your teacher, respect your mother, father, and put your efforts with sincerity, and there is no shortcut to the success. It will take its own time. Thank you for inviting me. These are my credentials. Anybody is interested, I will be available. I am mentoring over 300 startup actually businesses. I'm also the chairman of Small and Medium Chamber of Commerce and ex-president of AFSTI, chairman of All India Food Processing Association, Western region. I mentor a lot of students. I can be contacted on this number. Very active on the LinkedIn, or Insta or the Facebook, you can connect with me on the Gmail ID. Thank you, Siddharth sir. Thank you, entire professor. I respect you, and God bless everyone.
Wonderful talk.
I can take 1 or 2 questions if there are questions.
Yeah. I think separate. So let all the talks be over, and then we will open it for Q&A.
Okay. Thank you, sir.
Thank you. Thank you so much.
Thank you so much, sir, for sharing traditional knowledge and history of India and world about food technologies and nutrition. Now we have our next speaker, Dr. Himjyoti Datta, sir, who is assistant professor at Mizoram University, and his area of interest include food, health, research, and nutraceuticals.
This one. Kombucha. Okay. Yes, sir. So 220. Good afternoon. Thank you very much, the organizers, for inviting me for this beautiful event. So, I am Dr. Himjyoti Datta. I'm assistant professor and currently heading the Department of Food Technology at Mizoram University. I shall be speaking about kombucha, and I have given a subtitle to the name of the movie, A Sacred Drink to a Nutraceutical. There is an objective behind. Kombucha was supposed to be my PhD area, but because of some lack of facility and some understanding, I changed it to something else. Now, most of my researches, if you see, are on carbohydrate polymers, but this remained inside me. I'm still trying to continue with it. It's very interesting. I shall be sharing my experience with kombucha. In 2020, I published one chapter on kombucha. After a long journey of my keeping my interest with me on this particular drink. This has a very beautiful history in my life. The lady sitting, so I hope there are people who are basically from Assam, maybe knowing of the district Borpeta. I have highlighted it over there. So in Borpeta, this lady, she did one thing in the place of her prayer. We called it Guwahati. She used to brew some secret drink. It was not, obviously it was not beer. It was a place of worship. But she secretly brewed some drinks and she did not tell about it. She did not talk about it. And she did not share any information about it with her children and grandchildren. So it was secretly brewed and talking about it was a taboo. Selling was not permissible. She could not sell it. And later on, we came to know that donating the material to others
was considered noble by the community, but it remained a secret. I came to know about this from her granddaughter, who happens to be my mom. So she is my great grandma from Barpeta. The magazine Prantik, which is an Assamese bi-monthly, in late 1990s, a set of articles were published in which I was a kid. I was maybe in class 6, 7 like that. So some reports on one drink called kombucha was published, which in Assam they call it sahpitha. Somebody told it Manchurian tea. By different names it was known. And after discussion with my mom, I came to know that this particular drink is that drink which my great grandmom used to brew in the Guwahati. Then from those discussions, I also read that it was used as a cure for cancer and was also an anti-aging remedy. Later on, I also prescribed this to my friend whose father suffered from cancer. He continued for a few years. After that, he collapsed, he expired of cancer only. Then this thing remained in my young mind, in young brain. I kept reading about it, and from that, those information gathered in those days, I came to know that the distribution of kombucha, it was somewhere here in this region, especially in China, Korea, Japan, also in Russia, and in our Northeast India. There was no report from the other parts of India, or perhaps I haven't gone through, I haven't received any. Then there was a huge gap, maybe of 10, 15 years. I could not work on kombucha. Then I again, for this book chapter, I started reading. Then I came to know now this particular drink is popular in the USA, Canada, as well as Brazil. So this particular drink is commercially being manufactured in these areas and predominant in these areas and also in Australia. Looking at the history, the earliest available report on kombucha dates back to 2020 BC in China. So it must have originated in China, and it has a relation with tea because it is brewed from tea. So sugar tea, you have to add sugar to the tea and then brew it with some microorganisms. So basically it started in China, which was the richest in tea production at that time. And it was called as divine lingche or che. So, and in Czech Republic, the Moravian monasteries kept their fermented tea beverage Olinka, they call it Olinka, as a closely guarded secret. You can just find out the maps with Czech Republic, those monasteries and my grandmom's prayer area. It was the same, it was something like a cult. So this practice gradually made kombucha practice divine and the myth on it was developed. So later on, a doctor named Kombu was summoned to Japan to treat the king of Japan. And this became popular from his name Kombu and the term che. So it was started to be called as kombuche and later on as kombucha. In Russia, it was found that kombucha consumption reduces the aging, the human aging. It reduces the rate of human aging. And more recently in 1986, it was found that after the Chernobyl disaster, the kombucha consuming areas, the people residing in the kombucha consuming areas were less affected by the radiation damages. Then with the British company route, the kombucha, it migrated to US and other places. And during the World War II, there was a scarcity of tea as well as sugar, the raw materials required for kombucha production. And hence, there was a drop in the popularity of the kombucha beverage consumption. And further, in 1950s, a rumor on its toxicity, probable toxicity, was spread, which affected its popularity further. So necessarily from the 1960s, research on kombucha started. Scientific research on kombucha started by Swiss scientists, and they reported that drinking kombucha was similar in benefits as consumption of yogurt. So to define it, kombucha is a beverage produced by traditionally practiced fermentation technique involving sugar dissolved in black tea by a symbiotic culture of bacteria and yeast, which we call as a SCOBY. And this bell-shaped or cake-shaped SCOBY culture is also known as the tea fungus. It's a symbiotic colony of some bacteria and some fungus. The fermented beverage, when freshly prepared, it tastes like sweet sparkling apple cider. So in the market, in Guwahati market also, nowadays you will see kombucha being sold, one bottle, one small bottle like of the thumbs up bottle, it will cost you around 100 rupees or 120 rupees. And it has a vinegar-like acidic flavor to it. The predominant species of bacteria and yeast that have been found in different kombucha cultures are presented in the slide. You can read. And the SCOBY, it looks like this particular bell-shaped structure. This is the fermenting inoculum used for the kombucha preparation. And scientists from different parts of the world have found out that the SCOBY, the composition of the SCOBY, the bacterial and yeast species in the SCOBY, it differs from country to country, from altitude to altitude, from temperature regions to temperature regions. And because of this difference, the final product quality of the kombucha, that liquid, is also different. It will taste different. So the targeted functionality, since it is being sold as a functional drink, so its targeted functionality will vary considerably with difference in the microbial population. So our review, it found out that 85% of the total bacterial population is gluconobacter, and Zygosaccharomyces is the predominant yeast in different kombucha cultures. If you look under the scanning electron microscope, you will see that there is a development of nanocrystalline or nanostructured cellulose fibers, which forms the SCOBY, and it is basically produced by the Acetobacter species. And upon that layer of microcrystalline cellulose, the microbial cells will get clustered and finally form the SCOBY. And it has a relation with the traditional vinegar formation. So vinegar is a byproduct of wine fermentation. So in the wine, what happens? The sugar is converted to alcohol by the yeast, and that alcohol, that converted alcohol, is again turned to acetic acid by the acetic acid bacteria. So it is a two-way process. It is both anaerobic alcohol production and aerobic acetic acid production. It is somewhat similar to that, but it is more complicated. That's what. So unlike simpler vinegar production, kombucha production forms alcohol, different kinds of organic acids, and some other metabolites. These other metabolites are the things which are considered to be the health promoting factors or nutraceutical. But the problem is that many of the research papers still are naming them as other metabolites. They have not been identified. So hence it is more complex and more critical. Formation of a daughter SCOBY is a unique and peculiar phenomenon. If you produce kombucha at your lab, then you will find that from one layer of the SCOBY, one daughter SCOBY emerges. It is like cell division, but at a large scale. So once the daughter SCOBY is formed, it is considered that fermentation is over. Now you can drink the kombucha. And the daughter SCOBY you can carry out to the next batch of fermentation. This is a schematic diagram that we have prepared. So after fermentation, the pH, because of the acid production, it drops from around 4.6 to it drops down to around 2.0. Gradually, the daughter SCOBY, it develops, and it makes the method semi-aerobic. It is neither totally aerobic fermentation nor anaerobic fermentation, but it's a semi-aerobic fermentation. So this is the flow chart for if anyone wants to produce kombucha drink at his home, you can take it from me. And the product quality, it can be modified by altering the process. The selection of the raw material, you can take black tea, you can take green tea, you can take the waste of the tea industry also. The color of the final product is generally decided by the color of the tea that you are using. If you're taking green tea, the color will be lighter. If you're taking black tea, the color will be obviously darker. However, significant color changes during fermentation can also occur. The quality of the water that you are taking, it plays a major role because of the dissolved minerals in it. And the double stage fermentation can sometimes be carried out to enhance the fizziness. And if you increase the fizziness, obviously, if you are increasing the carbon dioxide, obviously the alcohol production will also increase. That you have to consider. So green tea has been considered as a better candidate for kombucha manufacturing. And one important fact is the black tea waste from the industry can also be used as a raw material for kombucha manufacturing. A 12-day of fermentation is considered ideal for the ideal product formulation. After 12 days, what happens? The formation of acetic acid, it increases. That can cause an acidic toxicity also. Non-tea kombucha, without the tea, if you can use some other extract, other plant extracts, kombucha can be formed, but you will be missing the tea metabolized from there, and hence it may not be that healthy as it is made from the tea. Those are called kombucha adjuncts. So these are the different metabolites that have been identified in kombucha, and their health promoting factors are also mentioned in the slide. It may be looking very small. So if you need, then I can share it with you later. So other minor metabolites are also produced depending on the SCOBY constituent, the tea composition, the sugar content, and the rate of fermentation. Health benefits, I have put a question mark because there are people who puts a question mark on any real health benefit of brewing the material, because tea itself is healthy. And upon that you are fermenting it and you are telling that it is healthier. It has been questioned because there have been insufficient scientific evidences, although it's an age-old. Added benefits of kombucha beverage have been often doubted as it's an exaggerated hype and is considered to be a result of tea constituents only. And all the studies that have been carried out since morning, I have been sitting here, I have been listening to different talks. One major problem with these nutraceutical products is lack of trials on humans. In kombucha also till date, there have been no human trial. But established properties in vivo in animal models have been on anti-inflammatory and anti-cancer, anti-hypertensive, anti-diabetic, so all the lifestyle diseases. And they also have antimicrobial properties, which may have a positive effect as well as a negative effect on the gut microflora. So we have to be responsibly drinking it. The safety aspects, the prolonged fermentation forms high acid, lactic acid, acetic acid, citric acid, and other organic acids. So at such low pH, chances of microbial contamination is minimal. So other microorganisms cannot grow over there, but cannot be nullified. There may be some toxic microorganisms growing. So you have to be careful in that also. Then unacceptable mold growth on the surface of the kombucha, as well as sometimes the SCOBY, it gets affected by some toxic molds. So that has to be also considered. And there may be some inorganic contaminants also. There have been 5 cases of toxicity and 1 case of death after consumption of kombucha till date. But so the US FDA in a survey in 1995 has said that no pathogenic bacteria or hygiene violation was found in the USA after consumption of kombucha drink by the population. So discussing the safety aspect, it has to be considered that it is a traditional drink and there has been tradition of its production and use by people every day people have been drinking for generations without any adverse effect on human health. There have been no guide on kombucha. There have been no guidelines on kombucha by the FSSAI till date. And it is very confusing because whether it is alcoholic or not, itself is we put alcohol in the wine shop, we keep the other drinks in the stores in India. So it is very important for putting the guidelines for kombucha, but till now FSSAI have not given any. The Bureau of Food Safety and Laboratory Services of the Pennsylvania Department of Agriculture, they have set some guidelines. So based on that, the pH of the kombucha, it should be the pH less than 4.6 for pasteurized kombucha. If you are pasteurizing it, then it is pH can go to below 4.6 and pH of less than 4.2 for non-pasteurized kombucha to attain within 7 days of fermentation. If you are selling it as a non-alcoholic product, the final product should have lower than 0.5% of alcohol content in it, which is very difficult to control because even after bottling, sometimes if you are not pasteurizing it, the fermentation it continues. pH below 2.5 should not be taken without diluting the kombucha. And you can add 0.1% of sodium benzoate and potassium sorbate as preservative. If you want to pasteurize it, you pasteurize at 82 degrees Celsius for 15 seconds. So if you go for any product formulation as nutraceutical, so this is the process flow chart. So you have to abide by all these guidelines, and human trials and clinical trials remain to be the issue with kombucha even after so much of its availability in the current market. FSSAI being the leading in the world, because I have been teaching nutraceutical to my students of MSc, third sem. This is a slide from my class. FSSAI has been a leading organization in setting standards for nutraceutical and nutraceutical-like products. So there have been distinct categories of health promoting products that have been recently in 2014 released by the FSSAI. Under this, the FSSAI has categorized those products into these particular categories. Already Prabodh sir has detailed about this. So what are the major gaps that I would like to discuss? As SCOBYs are different, the microbial species are. So our friends who have been working on TLC, HPTLC, they may be helpful in that. And the different components and their bioactive pathways need to be validated. And the ideal product regulations, including FSSAI, including Codex Alimentarius, including European Union for kombucha, have to be given as soon as possible. The waste utilization of the kombucha industry, which is the SCOBY itself, has not been taken into account. As I have told you, it is a hub of nano to microcrystalline bacterial cellulose. So that aspect has never been, that has been considered, but not to the extent that it deserves. It has metal removal properties. It has been found that some of the metals after the production, after the brewing, the metal level, it goes down. That means something is absorbing the metal or eating up the metal. So that has to be found out. I think what I personally think the cellulose, the microcrystalline cellulose should be responsible for this. That same cellulose, it can be a bioabsorbent material. It can be used as a filtration device. It can be a biodegradable substrate, or it can be used in composite as biodegradable composite material synthesis, and can also be used in some cosmetic products. Here are different kombucha manufacturers in the world. I have given a list. One local, our friend from Aizawl, I am from Mizoram University in Aizawl. There is a success case. This particular guy, Mr. Lal Biagzwala Sakchuak, he is an enthusiast, is a kombucha enthusiast. After MSc Psychology, he has come up for kombucha manufacturing. He has been doing it since last 3 years, and he has been in constant touch with me. He has been developing many different variants of kombucha and also kombucha adjuncts from non-tea sources, not tea, but some other sources. He keeps it a secret. I have asked him not to even tell me, so that his business secret remains with him. This is a success story. I asked him, I'm going to IIT Guwahati to present on kombucha. Please give me a photo of yours and your kombucha. He sent me this. So I'm sharing it with you. So kombucha or any food science or food research, it involves many aspects, several aspects. What I feel, it encompasses aspects of anthropology, of folklore, where I started this presentation, the basic sciences, physics, chemistry, mathematics, biology, nutrition, engineering, studies of the environment, psychology of people who are consuming and manufacturing it, and of course the commercial aspects of the manufactured product. So it actually involves the whole of food research, what I feel. So thank you. Where I started, this is our Namghar or our Namghar, where actually all this my lecture started, where it was originally brewed by my great grandma. And there are lots of different aspects related to our food, related to our dress that we have been wearing, related to our culture, which are dear, which are being practiced in dear. We have to open our eyes and spread our hands, go into it and find out the scientific aspects of it. And these are not just places of worship, but these are places and resources to be pondered about. Thank you very much. If anyone has any question, you can please raise. Okay. Okay.
Wonderful presentation, sir. Wonderful presentation.
Thank you, sir.
Thank you, sir, for the amazing presentation. Now we have Honorable Chief Guest of the valedictory session, Shri Pramod Bodo, sir. I request Arunimadi to felicitate the Chief Guest and his associates.
Now heading towards the next speaker, we have Mr. Pranjal Deka, who is Technical Officer at Food Safety and Standard Authority of India. Over to you, sir.
A very good afternoon to all the respected dignitaries present today in this seminar. Myself, Pranjal Deka, I am a technical officer working in Food Safety and Standard Authority of India, Northeastern Region, branch office Guwahati. So somebody else is operating the system? Okay. Yes, okay, fine. Thank you. So today, actually, there's a very interesting discussion going on regarding the nutraceutical and health supplement products. So I'm thankful to Mr. Siddharth, Professor Siddharth, Dr. Siddharth, for inviting us to put some light on the regulatory aspect of this nutraceutical and health supplement products. So I'm from FSSAI, and all of you might be aware, FSSAI is the organization who is looking after the entire food system in India. This is the FSSAI is the regulatory body to provide standards and norms for all the food products that are available in the market. Being the nutraceutical, one of the products, food products consumed by the consumer, it is also governed by FSSAI norms. FSSAI came into effect from in the year 2011 through the Act Food Safety and Standard Act 2006. To implement the Act, Food Safety and Standard Act 2006, a few regulations were made. These regulations are like licensing and registration regulation, through which it provide the provisions how to issue the license to the food business operators. Similarly, we have got the food product standards and food additive regulation, which provides the standards and standards that are required for a product to be available, to be eligible to get into the market, and the additives what are allowed for a certain group of products. Similarly, we have got this food safety and standard health supplement, nutraceutical food for special dietary use, food for special medical purpose, and prebiotic probiotic food regulation 2022, which we are now calling it as a Nutra Regulation 2022. Initially, there was a regulation in the year 2016. However, since this topic is very sensitive and a lot of research are being going on continuously, a new draft has been prepared, and this has been operationalized since April 2022. And an attempt has been made to make it more easier for the food business operators. I'm not going through the operations of FSSAI, where the manufacturers are producing less amount, have the capacity of producing less amount of product. They are either given license by the state or they need to have registration. That means their production capacity is less, so there is a specific category for registration. However, since 2021, there has been an advisory through which all the nutraceutical manufacturers are made compulsory to have license from Central Licensing Authority only. So all the nutraceutical and supplement manufacturers need to have central license, not the registration, not the state license. Already a lot of talk has been made regarding this, that what are the products should be there in a license. Actually, under this regulation, these kind of food products have been included. They are specially processed and formulated and specific nutritional and dietary purpose. There should be a special nutritional and dietary purpose, and they should be specially formulated. They should not be, and it should not be like parenteral use. There should not be parenteral use. That means it should be only oral use only. And this regulation regulates the food products for people above 2 years of age only. Below 2 years of age, there is another regulation for that. Standardized food products and standardized food ingredients, which are already standardized, they cannot be considered as a special food under this regulation. That means they cannot be considered as a nutraceutical or health supplement. They have already been standardized, like biscuit or any beverage. They have already been standardized. We can't consider, say, by simply those products as a nutraceutical. For being a nutraceutical and health supplement, it should be specifically designed. It should, as per this regulation, the products coming under the drugs will not be considered under this regulation. Similarly, products should not contain hormones, steroids, and narcotic and psychotropic substances are not coming under this regulation. Products claiming cure, prevention of mitigation of any specific disorder are also not considered under this regulation. Mere food forms, vegetables, cereals are also not considered as under this regulation. So basically, there are 5 categories divided under this regulation: the health supplement, nutraceutical, food for special dietary use, food for medical purpose, and prebiotic food and probiotic foods. This has already been explained very well by Prabodh Halde sir, so I will skip it. Basically, health supplements are the products which have concentrated source of vitamins and minerals. Then nutraceuticals, they will be having the extracts and the extracts of the some extracts which have nutritional benefit to the health. And food for special dietary use, they are like for sports person. There are some products for weight reduction. There are some products. These are FSDU products. Then FSMP products. FSMP products are given to medical practitioners. There are some people who are unable to digest some component of the food, so they need to provide a special food. In that case, this FSMP food products are provided. So there are some general requirements. Whenever you are going to manufacture this nutraceutical and health supplement, there are some general requirements you need to follow. So like the delivery system, you can see that the delivery system can be either powder, granules, tablets, capsules, or any conventional form way. But if it is a conventional way, like biscuit and like other candy or something like that, it should be properly distinguishable from other regular products. The label should distinguish it from other products. Similarly, if we are developing some new technology, nano-derived ingredients or modified drug delivery formats, then we need to have a special permission from FSSAI before implementing it in your product. Ingredients. This regulation has provided the list of ingredients for different kind of product. I have provided you 5 different categories of product. For all these different categories, there are some lists are available. It is called under schedules. Different schedules have been provided, and we can use the ingredients from these schedules only. Like schedule 1 has been mentioned as it contains vitamins and minerals and other nutrients. It contains around 16 vitamins, 15 minerals and their sources, 52 amino acids and 7 nucleotides. So you can use this vitamins and minerals for the product development of your product from this list only. And the source is also very important. You can't use so sometimes what happens, some people are using some plant, they are mentioning the botanical scientific name and the plant name, but they are not providing the part, which part they are using, whether they are using the leaf, the seed, they are not specifying. That has been specified here. So before when you are producing something, you need to document all the details, and these details have been provided here only. There is also another schedule. This is schedule 3. This is specifically molecules, isolates, and extracts for health supplement, which is I have already explained that it is a concentrated source of vitamins and minerals. These health supplements are not allowed to have any ingredient from this schedule 3. Other than that, this is specifically meant for nutraceuticals, FSDU, and FSM in your product. Other than that, there are some other conditions. If you want to use the additive from anything else from this other than those tables, at some point they are allowed. This all has been given in the regulation. Due to lack of time, I am not able to provide you of cereals and legumes which can be used. If non-specified foods, including novel foods and non-specified ingredients, you want to implement, then you need to have a special permission for approval of non-specified food before using it in your nutraceutical health supplement product. If an ingredient has a safe history of use for in India for 15 years or all over the world for 30 years, excuse me, then that can be allowed upon prior approval from FSSAI. If the completely new ingredient is being used, then again you'll have to take as non-specified food non-specified food and food ingredient permission from FSSAI. Additives and processing aids, I have already told you additives. This schedule 3 has been provided for additives. Processing aids are allowed. The flavors as per 3.3.1 of food safety and standard food product standard and food additive regulations, they are allowed. You will have to also maintain the purity criteria. There are other terms as well. This needs to be followed. And let us come to the labeling part. This is the labeling part for normal food. It contains the name of the food product, net quantity, date of manufacturing, license number, logo, etc. This is for regular products. However, for nutraceutical and health supplement products, you need to follow additional kind of informations, like you have to provide in front of the packet whether it is a health supplement, nutraceutical food for special dietary use, or medical prebiotic food. You have to mention it. Then again, some other duration of use, not in not to use exceed recommended by daily use. Then warning statement, product is required to be stored out of reach of the children. This kind of statement you have to make for this special products. Claims and approval. No claim that cure, mitigate, or prevent any specific disease, disorder, and condition or refer to such properties unless specifically permitted. Excuse me. Permitted by food authority. So in your food product on the level, you can't claim any cure or prevent disease, something like that. When you apply for this license under food safety for your product, you need to provide the ingredient and additive list of your product, like this format. This is a format where you have to provide the RDA value, percent RDA available in per serving of your product. Then ICMR, which the reference ICMR which you are following 2010 or 2020, your product target group, serving size, etc., etc. Anyway, thank you. I know I had to go through very fast and do most of the slides were words only, very less pictorial slides because I had to depend on the facts and figures only, couldn't depend on pictorial parts. So if you have any doubt, you can contact us in our email ID and phone number. This is our address. Thank you. Thank you very much.
Thank you, sir, for sharing your profound knowledge with us. Heading towards the next speaker of the day, I would like to invite Mr. Ranjit Barwa, who is director at Aromika team, to say a few words, sir, please.
Should I send one second? Yeah, okay. Okay. Put it in the bigger. Yeah. As it is a PDF, so I have you have the right. No, we can control it from the media Adobe Drive. Yeah. Open your Adobe system. Will it move? This is system file. This is the file. Just dedicate his picture. Take one of it. Okay. Ready? Yes, sir. Ready?
Good evening, respected guests, distinguished professors, experts in the industry, student friends, and all esteemed members, guests present today on this seminar. It's a pleasure to be invited and be given an opportunity to be a part of this prestigious program. My name is Ranjit Borua, and I am the founder of a tea startup called Aromika Tea. A little of my background, I've been working in the tea gardens for about 20 years, having worked with McLeod Russell, one of the largest tea company, having worked in different places across Assam, Upper Assam, then in BTD, then in Lower Assam, in many places. Today, this seminar where a lot of discussions are going on about the health benefits of of medicinal plants. I think this is one of the very interesting discussions going on, especially for startups like us. We have a startup, Aromika Tea, where we create value-added products based on tea, where we add different flowers, fruits, herbs, and spices to different types of teas that are produced in Assam. Assam is a gold mine of tea. How best we can value add that tea to get an additional market, to get better price realization, and to expand even in the remotest corners of the world. I basically work with small tea farmers. My farmers are from Upper Assam up to Kokrajhar, from Dumduma to Kokrajhar. They are small farmers, have small lands, but they make fantastic teas. There's a shortfall, there's a lacune for market, there's a lacune for better price realization. So I have used my knowledge in tea blending, tea testing, and adding different spices, flowers, fruits that are more prominent in this part of the region, that has nutritional value and that has been time tested. Basically, something like tea is using age-old wisdom and blending it with modern knowledge of tea. So what problems? Why we have done this? There are so many. Our daily lifestyle has become very, I say, fanatical. Like we are stressed. There loss of immunity. Tea can be a very good solution for this. It has got antioxidants, and adding different values, different ingredients to it adds the value. Some of our products that we have used, we have made is the blue tea made of butterfly pea flower, rosella tea, which in Assamese we call tengamora, bamboo shoot tea. We know bamboo shoot is an integral part of the diet of the Northeast region. It is very rich in antibacterial, antiviral properties. Moringa tulsi, spice turmeric, and Bhujolokia tea, karha tea, lemongrass, rooibos chamomile that improves quality of sleep. So these kind of different blends we make, and I've got two samples here with me today. This is a blue tea and this is a Bhujolokia tea. In fact, this is one of the most popular teas now people have. In fact, this is one of the most popular teas now people have really accepted it. It is blended, Dheki tea blended with Bhujolokia, ginger and lemon. It's one of the highest selling teas during the Corona times. Does not cure Corona, but it really gives you relief from the problem associated with Corona. So something like this. So basic idea is to add different improve the nutrition and the taste of the teas. There's a huge market across the world, and we can add more value to the teas produced, and by improving the nutritional quotient. Challenge is how do we validate this? Challenge is how can we say that this tea is really helpful for the particular illness? We know that ginger is good for many health, small illnesses that has been traditionally being used in the house. Presently, we have to bank on all those data, all those research papers. We know tulsi is good when we have small cough and cold, our grandmother, it has been there. And when you have a cough, you have tulsi with madhu. So these are the common knowledges. This age-old wisdom we are trying to use into the teas to take out a product. In this forum, I am really looking forward to experts in this field where we can get this validated, where there will be, my previous speaker has mentioned about the human trials. Now this is a big challenge, human trials of the products. And there's a lot of challenges in this. This needs to be overcome so that we can get into more overseas market, more confidence of the people. Like when I say that blue tea is anti-aging, it has got butterfly pea flower, it is good for sugar and pressure, it makes you feel young, it takes care of, reduces the wrinkles, and gives a glow to your skin. Now, these certain things we cannot claim, but it has got health benefits. Now we need solutions for this. How can this be done? Is it actually helping that? Presently, we can say that these products are known to have these qualities based on the health benefits research done on the butterfly pea flower. So certain challenges are there. So we have tried out a new tea called Rudraksha tea, which is made up from Rudraksha. Now Rudraksha, many people use it for mystical powers. We know Rudraksha can have 105 Rudraksha beads. It has got a lot of mystical powers. So I've tried to use the Rudraksha powder into the tea and get in the system. We can only say, but then to validate, to get the market is different. We can base our products based on the research done on the Rudraksha. It is being used in Ayurveda. So we can take a reference from there. So certain that Rudraksha powder. Ultimately, the health benefits and the taste matters. There are, of course, much more better economics for the thing. I just tried to show it. So any entrepreneur who can look into this, like adding lemongrass or adding different spices to the normal tea can give a lot of revenue, increase in revenue. So this in very short, what we are doing, we are trying to get into more and more bigger markets through product consistency, through product validation, through more taste, and also trying to meet up to the customers' requirements such as organic certification. Now there are so many export inquiries where they want to know the traceability, they want to know the organic, whether the particular teas or ingredients are organic. We have many growers. I have got 2 dedicated growers, one in Kokrajhar, one in Boyagaon, one in Gopur, who are producing this organically. But then they are certified organic, but the transition, the blending unit, everything has to be organized. The ingredients that we use, maybe a lemongrass, maybe a ginger, maybe the bhojolokia, maybe the butterfly pea flower, maybe the tulsi, we need to certify that these are also organically produced. So that ultimately the end product is fully organized, is organic, and we can claim a premium price on the products. There's so many challenges. I think this is a very good forum for me to even speak out and the requirement that we have, so that from this esteemed guest, experts in the field, we can definitely take in ideas, we can take in more collaboration, we can bring in more growers into the fold, so that the entire industry, the agriculture, the producers, every producers benefits by collaborating with each other. Thank you very much for this opportunity. And we have put a small exhibition outside the thing. I would request everyone to just come and have a look at our teas, and we look to our support in all fronts in the times to come. And thank you, Dr. Siddharth sir, for giving me this invitation, for inviting me here, to give me an opportunity to speak about us in front of such a distinguished crowd. Thank you so much. Thank you.
Thank you so much, sir, and thank you all the presenters who are present online and offline for the interesting information. Now the house is open for a Q&A session. All the questions and suggestions are most welcome.
Hello. I think probably a couple of questions I have from my side, particularly for Dr. Vivek. Dr. Vivek, are you there?
Yes, I'm very much here. I was enjoying the sessions, listening the expert comments from speakers there, so I'm well present here.
Yeah, so in last 2 days, we have deliberated on many kinds of herbal products, whether it is herbal medicine or herbal food or even herbal cosmetics. One common issue we have seen in all these presentations or representations, when we talk about entering into a market which is regulated by FDA, for a small organization or even for an academic lab, when they start working on discovery phase of a cosmetics or a food or medicine product, how do you see that they can go about for preclinical and clinical trials particularly? Because unless and until they have clinical trial, they cannot go into the market. So can you suggest a model? That is one, and probably that will help many of us here. And second thing is there any institutional framework or even in your organization as a entity who is into the business can help them in this regard?
Thank you for your question. So am I audible clearly?
Yes.
Right. So answering your first question on the clinical study and why this is required, what is the best model to do this? So normally in case of the nutraceutical products or any other product category where you talk about the cosmetic products or any other food product, when you claim something clinical, anyways, you have to go for the clinical study, right? For example, these days when the FSI has put forward the regulation and they have given a very concrete framework for this regulation of the nutraceutical product, health supplement, and other kind of the product categories. When you're going to market a product, say for example, we designed a product and that is for the cardiac care or anything, something to manage the disease or to manage any physiological condition. It is very important to validate that based on the clinical study. Whether it's probably like FSI regulation says something that which has been in use in the country of origin since last 15 years. Of course, it has ample data to show, ample data to present that supports that this ingredient is found to be safe for human consumption, because clinical studies, something which talks about how safe is that particular product, what kind of the interaction is happening inside the body, or how long the product should be consumed for the desirable benefit. A lot of other aspects is very much important in the clinical study. So I would not deny the clinical trial should perform, but that should be based on the kind of the claim which you are mentioning on the product. Because if you go for the clinical study, I don't think that every product you can invest lakhs of the rupees and then to find some revenue out of it. But yes, a same ingredient, say for example, if I take an example of botanical ingredient like Boswellia serrata, this is one of the anti-inflammatory ingredient, or curcumin for curcuma longa, and we know that these are the anti-inflammatory component. And like, for example, you say now these ingredients, apart from its anti-inflammatory activity, you find, okay, this has the new therapeutic property. Of course, I would stress that clinical study should be performed in that case, and that too, that with the proper decision, what should be the primary objective and the secondary objective of the clinical study. Because in case of the drug and the pharmaceutical salt, the standards has already been developed. The pharmacokinetics and pharmacology is well defined. So supporting data is already available. But in case of the nutraceutical ingredient, or if you're going for any cosmeceutical ingredient, which is to be used on topical application or and then external application, I think to establish the safety study, it's very important to do the clinical study. But that too, that what you are claiming for. Here, the other thing, the bent of thought can be like, sometimes like in international market, the investigator-initiated trial, which we call IIT, it's an investigator-initiated trial where the few doctors or a single doctor involve few patients for the observation on that how the product is performing, what is the outcome is coming out of this product. That is a less, expenditure thing where the investment is not that high, while it validates the claim of the product and exactly the, the result of the product or the action point of the product. So that can be also
it's again a very big support to substantiate the claim or to validate the claim. But I mean, somehow the clinical study becomes very important subject in case of the nutraceutical, cosmeceutical, or any kind of these product where you are claiming something to alter the physiology. Because if the product is therapeutic, if the product can bring some physiological change or can improve the condition or body condition, I think that requires clinical study to validate and substantiate the proper safety and the biological activity overall. That's my answer for your first question. And can you please repeat your question number 2?
The second question was, as an organization, do you support this kind of micro industries? Because there are many micro industries here who are looking for, validating their product.
Yeah, I think undeniably, I think there is no point on this. Like academia and industry collaboration has always been an important, and these days it becomes more important. So profoundly, I can say yes. We look forward for such kind of the vast studies, exclusive clinical study or the preclinical studies, wherever or any kind of the project where we can collaborate with the institutes to validate our claim or to make it more, substantiate our product research, product designing, ideation, and everything.
Sorry, I think I didn't make my question clear. I was asking about the micro industries. For example, somebody is working on bamboo tea and the person wants to validate. They have started their own organization. So whether they can collaborate with your kind of organization and go ahead.
Yes, sure. We wholeheartedly welcome such projects, and we would definitely welcome such concept if any industry want to collaborate with us and to work together. I think it's very affirmative. I would like to express my intentions here on it.
Thank you so much. So we can move on to the next question.
I just want to add, there are a lot of small industry or independents here. So I'm Dr. Vijay Swaha from Mumbai. So what we can suggest, that still after induction of the central FSI rules, scenario is quite confusing, and for small independents it becomes very difficult. So I think jointly, they can have some senior FSI consultant who, even you can recommend somebody with whom you have some interaction or something. And I think that will be great help for these people, because we find a lot of people they have the product, but they don't know how to go about it. And still, in industry now, many people in Maharashtra, Rajasthan, still they are working with the state FSI. So that, I think we need to educate them, because they are confused and they need some guidance, whether they need to go for a central FSI or how, but and I think some authority is also here. So I think they can also have some sort of knowledge session where you can invite. We have met a lot of independents here, and they don't have any guiding real guiding source or window where they can go and ask. So I think how now even for making socially there are a lot of camps going on for making a Aadhaar card or brain card or like that. Same way I think authorities in FSI or in Ayush, they should try to whenever there is a chance, they should organize some camp sort of things and try to educate and help as much as they can for this micro industry or small enterprise. Just a suggestion. Thank you.
No, I take it very positively. I think, as I said earlier, the collaboration is always very helpful. So we look forward for such invitations on it, like if we have to collaborate and to do something, or those concepts which is being there at the micro level, and we can make it commercialized, we can buy those concepts and to make it happen. I think we can definitely discuss offline to this, and if any proposal is there from the institute or the collaborators, we would love to understand it. We would love to explore it properly. That's how I think. So it's a very affirmative that we should go in this direction, because a lot of the innovation at different strata is happening. People, they have the good concept, but
to advance it to the commercial level, something stops them or the impediments are there. But we as a part of the industry, we at that, we can definitely fulfill those, result oriented on that part. Thank you.
Thank you so much, sir. Now I request Siddharth sir to come forward for the valedictory session and closing remarks.
Thank you, Rashi. Thank you. Thank you so much for everyone's cooperation. We have come to the end of today's proceedings, and I'm extremely delighted that to inspire us and support us, we have our chief guest of this valedictory session, Shri Pramod Boroji, the Chief Executive Member of Bodoland Territorial Council. In fact, there is a reason, sir, why we wanted to be here, because when we talk about herbal products, medicinal plants, we have very, very, important connection with Bodoland. Being a, land of indigenous people, and it is a storehouse of ethnobotanical knowledge. So people use this herbal product for their medicine, for their food, for their veterinary needs, and whatnot. So we invited you for also initiating, why don't we do something as a herbal manufacturing hub for Bodoland, where if we get your support, then probably all the stakeholders who are present here can work together, and probably we can contribute something for the land. So I would request you, sir, to kindly come on stage and take your seat. In this valedictory session, we also have Madam Manjula Shaikia Bhuya, Madam, the ex-director of Department of Industries and Commerce, Assam State. She has long experience of, administration, and she was always kind enough to come forward and support us. She is also one of the advisor of our School of Agro-Rural Technology, and we get, her guidance for all occasions. We would also like to call Dr. Mrs. Ketaki Bapat, advisor and scientist G from Principal Scientific Advisor to Prime Minister's Office, to please come and take your seat in the dais. I'd also like to call Dr. Debajyoti Bose, who is kind enough to join us. He is representing Yobe State University, Nigeria, because we have some plans of collaboration, and we would like to take this platform to announce such collaboration. And last but not the least, our guest of honor, Dr. J.L.N. Sastry, the ex-CEO of NMPB, and also former head of healthcare of Dabur Research and Foundation, Dabur Research and Development Center. He is on the way. Probably he will be joining in the later part of this evening. So before we start, the program is not for us. It is for the participants. So for the 2 days, whatever we tried to do, we probably did a lot of mistakes. We have some ups and downs here and there, but we would like to take a feedback from all of you. What is your feedback? I would like to start from Dr. Jahirul Islam. Yesterday, probably some of you might have heard his initiative on Aga Road. So probably he himself could not come yesterday, but today probably he can add something, and real value addition to this program he can do.
Sure. I'm Dr. Zahirul Islam. I'm working with Agro I think you people are very familiar with the agar. It's the world costliest product, and Assam agar is a very valuable and costliest product in the market, in the world market. Since 75 years, our agar product is capturing in the entire world market some other ways. First time in the history of agar, this government, Dr. Himanta Biswa Sarma, leading government is taking first initiative for legalizing the agar. Actually, agar, after our tea and oil, the agar is most important and costliest product of Assam. Since 75 years, our product is going abroad by other ways, not officially, unofficially. And first time we are actually legalizing this one, and we are making the organized sector, this agar. Every year, 35,000 to 50,000 crore illegal trade was done from Assam, Northeast. The agar jo hai, Hindi is okay for you all. Yes, and Assam is also. Assamia আছে তো বহুত নহয় জানো আপোনালোকে মই অসমীয়াতে কয় এইকাৰণে আমাৰ বড়োলেণ্ডৰ চিকু আছে ইয়াত আৰু বাইদেউ মঞ্জুলা বাইদেউ আছে মঞ্জুলা বাইদেউ আমাৰ লগতে বহুত দিন একেলগে কাম কৰিছো মঞ্জুলা বাইদেউৰ কন্ট্ৰিবিউশন আছে তো আপোনালোকে জানি সুখী হ'ব অসমৰ যিটো আগৰ সেইটো এটা একচুয়ালি টপ মেডিচিনেল প্ৰডাক্ট হয় কিন্তু আমি অতদিনে ইয়াৰ বিষয়ে একচুয়ালি এৱাৰনেছ আছিলে কিছুমান মানুহে অবৈধভাৱে এই গোটেই বিজনেছটো নিজৰ হাতত ৰাখি এই বিজনেছৰ পৰা আগৰ বিষয়ে মানে কোনো কামেই কৰিব নিদিলে তেওঁলোকে অপৰাধ কৰিলে মনোপলি কৰিলে কৰাৰ কাৰণে আমি অসমীয়া থলুৱা খিলঞ্জীয়া মানুহখিনিয়ে ইয়াৰ পৰা কোনো লাভ বা আমি হেৰি নলওঁ হয় নে মঞ্জুলা বাইদেউ হয় নে নহয় তো এই বস্তুটো আমাৰ এছাম মানুহে অকলে কৰিলে এই আমি প্ৰথমবাৰ আমাৰ এখন চৰকাৰ অহাৰ পিছত আমি লিগেলাইজ কৰিলো আৰু ইয়াত অৰ্গেনাইজ চেক্টৰ কৰিলো আগৰ যিটো বস্তু আগৰ 1 লিটাৰ আগৰ তেলৰ দাম পৃথিৱীৰ বজাৰত 15 লাখ টকাৰ পৰা ষ্টাৰ্ট হয় 1 কোটি টকালৈকে 1 কেজি তেল বিক্ৰী হয় যিটো আমাৰ অসমৰ গোলাঘাট যোৰহাট শিৱসাগৰ 78% আগত আমাৰ আগৰ অইলৰ বিষয়ে একো নজনা নাছিলে আমাৰ মানুহে অতদিনে মই আমাৰ বিটিসি চিফ সাহেবকো কৈ আছো আমাৰ মানুহে অতদিনে আগৰ বুলি ক'লে আৰবিয়ান মানুহে তেলটো লগায় আৰবিয়ান মানুহে লগায় বা তেওঁলোকে আতৰ বুলি ইউজ কৰে হে মেন্টালিটি আছিলে কিন্তু আমি প্ৰথমবাৰৰ বাবে ইয়াক ৰিসাৰ্চ কৰি ডেভেলপ কৰি এই আইআইটি গুৱাহাটীৰ লগত আমি সহযোগ কৰি ইয়াক ডেভেলপ কৰি নাইপৰো মোৰ লগত কাম কৰি আছে গতিকে আমি 78% কম্পাউণ্ড পাইছো যিটো আমাৰ এই ইয়াত যিটো মেডিচিনেল প্লান্টৰ প্ৰডাকশন এণ্ড এডভান্টেজ ইন হাৰ্বেল মেডিচিন বুলি কৈছে ইয়াৰ মেক্সিমাম টপ যিটো কম্পাউণ্ড আমি পাইছো সেইটো আগৰ অইলৰ পৰা পাইছো আগৰ অইলত আপোনাৰ ক্যান্সাৰ কিউৰৰ কিছুমান কম্পাউণ্ড পাইছো এইডছ ৰিলিভাৰ কম্পাউণ্ড পাইছো পেইন ৰিলিভাৰৰ কম্পাউণ্ড পাইছো এতিয়াও শেষ হোৱা নাই কিন্তু অত বছৰে আমি এইবোৰৰ পৰা বঞ্চিত হৈ আছো এতিয়া আপোনাকো মই আপিল কৰিম আমাৰ চিফ মিনিষ্টাৰে বিৰাট ডাঙৰ ইনিচিয়েটিভ লৈছে আমি গোলাঘাটত ইণ্টাৰনেশ্যনেল আগৰো ট্ৰেড চেণ্টাৰ আনিছো যিটোৰ পৰা আমি এক্সপেক্ট কৰি আছো 50,000 crore টাৰ্ণঅভাৰ কৰিম আমি ইয়াৰলি ৱৰ্ল্ডত হাইয়েষ্ট টাৰ্ণঅভাৰ আগৰৰ পৰা হ'ব গতিকে আপোনাকো কৈছো যে আমি কোকৰাঝাৰ হওক তাতে হওক আমি আগৰৰ ওপৰত বহুত কাম কৰিব লাগে আইআইটিৰ সিদ্ধাৰ্থ সাহেবকো মই কৈছোৱেই কালি মোৰ একচুৱেলি মাৰ গা বেয়া আছিলে মোৰ মাৰ গা অসুখ থকাৰ কাৰণে তেখেত জিএমচিত এডমিট হৈ আছিল সেইকাৰণে মই কালি আহিব পৰা নাছিলো আজি আৰু উপায় নাপায় বোলো আহিলো অলপ আগৰৰ কথা কওঁ কাৰণ আগৰৰ বিজনেছটো যদি আমি কৰোঁ বা আমাৰ ইয়াত যিমান প্ৰফেচৰ বা ৰিচাৰ্চাৰ সাইণ্টিষ্ট আছে আমি সাধাৰণ মানুহ জাষ্ট এইটো আমি ডেভেলপ কৰিছো সাহসেৰে অসমৰ হাসিটোক সাহসেৰে এছাম মানুহৰ পৰা কাঢ়ি আনি আমি অসমীয়া থলুৱা খিলঞ্জীয়াৰ মনত আনি দিছো সেইটোৱে কাম আমি কৰিছো বাকি আপোনালোকৰ বহুতৰ মানে সিনিয়ৰ সাইণ্টিষ্ট সকলৰ বা প্ৰফেসৰ সকলৰ এটা দায়িত্ব থাকিব তাৰ পৰা আমি বহুত কাম কৰিব পাৰো বহুত এনালাইসিস কৰিব পাৰো বহুত ৰিসাৰ্চ কৰিব পাৰো আৰু এটা সিস্টেম অফ প্ৰডিউসিং আগৰ 30 ম্যাক্সিমাম 38 to 40% হেলদি কম্পাউণ্ড আমি এভেইলেবিলিটি পাইছিলো আৰু এই জ্বলা আৰু ধোঁৱাৰ গোন্ধ আছিলে ডাইৰেক্ট হিট কৰি প্লাই প্ৰডাকশন কৰিছিলো হেটোত আমাৰ বহুত মানে কম্পাউণ্ডবোৰ মৰি যায় এতিয়া মই
যিটো জুইচ জগাত মই পানীৰ ষ্টিমেৰে মানে এবষ্ট্ৰেকশ্যন কৰাৰ এটা পদ্ধতি মই আৱিষ্কাৰ মানে তৈয়াৰ কৰিছোঁ। IIT গুৱাহাটীও লগত আছে, দিল্লী IITও লগত আছে, তো আৰু বহুত মানুহ মোৰ লগত আছে। তো সেইটোত মই প্ৰথম পেটেণ্ট পাইছোঁ। আৰু সেই ছিষ্টেমত যেতিয়া আমি আগৰ অইল প্ৰডাকশ্যন কৰোঁ, টপ কোৱালিটিৰ মানে হাইজেনিক আগৰ আমি অইল প্ৰডাকশ্যন কৰিব পাৰোঁ। যিটো এতিয়া মোৰ দাঁতৰ বিষ আছিল 2 দিনমান আগত, মই আপোনালোকৰ নিজে এক্সাম্পল, মই কেতিয়াও পেইন কিলাৰ নাখাওঁ। মোৰ মাইগ্ৰেনত আজিলৈকে পেইন কিলাৰ খোৱা নাই। মই আগৰ অইল ভালকৈ ইউজ কৰোঁ, ভালকৈ একদম যিটো মোৰ পিয়'ৰ যিটো আগৰ অইল সেইটো মই ইউজ কৰোঁ, মোৰ পেইন মাইগ্ৰেন ভাল হৈ যায়। এতিয়া মোৰ মিছেছক কৈছোঁ, আজি এটা এক্সাম্পল লোৱা, মোৰ দাঁতত বিষাই আছে, মই কালি দাঁতটো পেলাই আহিছোঁ তাৰমানে। তো মই ক'লোঁ আগৰ অইলটো লগাই দিওঁ, চাবা মই IIT পাওঁতে শেষ হৈ যাব। ৰিয়েলি মই আহি পাইছোঁ, মোৰ বিষটো ভাল হৈ গৈছে, মই কথা ক'ব পাৰিছোঁ। তাৰমানে ইমান কম্পাউণ্ড আছে, ইমান প্ৰপাৰ্টি আছে, গতিকে মই সাহসেৰে বস্তুটো উলিয়াই আনিলোঁ। গতিকে আপোনালোকে খালি এতিয়া সহযোগ কৰি ইয়াক এক্সপ্ল'ৰ কৰিব লাগে। ইয়াত আৰু বহুত কিবা কিবি আছে। তো মই পলিচিও গভৰ্ণমেণ্টৰ পলিচি বনোৱাটো মই লাগি আছোঁ। আই এম দ্য ব্ৰেইন, মঞ্জুলা বাইদেউ জানিব, আই এম দ্য ব্ৰেইন বিহাইণ্ড অফ আগাৰুড পলিচি ইন আছাম এণ্ড ত্ৰিপুৰা। মই অসম চৰকাৰৰ আৰু ত্ৰিপুৰা চৰকাৰৰ আগৰুড পলিচি বনাইছোঁ। যি হওক, শুদ্ধ হওক, ভুল হওক, বনাই কেবিনেটত অনাবৰ পৰা ব্যৱস্থা কৰিছোঁ, লিগেল কৰাইছোঁ। 500 কোটি টকা দিছে ইলিগেল, মানে লিগেল কৰিব নালাগে বুলি। সেইসকলে 500 কোটি টকা দিছে ইলিগেল, মানে লিগেল কৰিব নালাগে, ইলিগেল পদ্ধতিতে থাকক। কিন্তু আমি শক্তি প্ৰয়োগ কৰি পেলাই আমি লিগেল কৰিলোঁ। যিয়ে হওক লিগেল হ'ল। আজি ট্ৰেড চেণ্টাৰ হৈছে গোলাঘাটত, তাৰ পৰা অসমীয়া থলুৱা খিলঞ্জীয়াৰ ল'ৰাখিনিয়ে, আমাৰ থলুৱা ল'ৰাখিনিয়ে বহু উপকৃত হ'ব। আৰু 50,000 কোটি টকাৰ আমি টাৰ্গেট কৰিছোঁ আগৰৰ বিজনেছৰ পৰা। তাৰ পৰা নানান ধৰণৰ মেডিচিন, এতিয়া নাইপাৰে আমাৰ নেশ্যনেল ইনষ্টিটিউট অফ ফাৰ্মাচিউটিকেল ৰিচাৰ্চ এডুকেশ্যনে তেওঁলোকে এতিয়া ইনিচিয়েটিভ লৈ আছে, মোৰ লগত লাগি আছে। তেওঁলোকে এই যে যিখিনি কম্পাউণ্ড পাইছে, কাৰণ আমি জাষ্ট উলিয়াই দিছোঁ বস্তুটো কি কি আছে, বাকী ফাৰ্ডাৰ টেষ্টিং তেওঁলোকে কৰিব, ছিনিয়ৰ চাইণ্টিষ্টে কৰিব। যে ইয়াৰ পৰা তেওঁলোকে এনিমেলছৰ ওপৰত ৱৰ্কিং ষ্টাৰ্ট কৰিছে, যে কেঞ্চাৰ কেনেকৈ ভাল হয় বা পেইন কেনেকৈ ৰিলিভ হয় বা অন্যান্য ধৰণৰ তেওঁলোকে অলৰেডি ষ্টাৰ্ট কৰিছে। নাইপাৰে ভাল সহযোগ কৰি আছে, ভাল লাগিছে। গতিকে আপোনালোককো কৈছোঁ, ইয়াত যিখিনি আমাৰ ছিনিয়ৰ চাইণ্টিষ্ট বা সকলো আছে, আপোনালোকে ইয়াৰ ওপৰত কাম কৰক। আৰু মোৰ যিমান সহায় সহযোগ লাগে মই কৰিম ডেফিনেটলি। মই আৰু এটা বস্তু ডেভেলপ কৰিছোঁ আমাৰ গোলাঘাট, যোৰহাট, শিৱসাগৰত। চিফ ছাৰ, আপুনিও এইটো শুনিব চিৰিয়াছলি। আমি গোলাঘাট, যোৰহাট, শিৱসাগৰত আমাৰ নেচাৰেল ইনফেকশ্যন হৈ আগৰুড হয়, যিটো জিঅ'জেৰা কনফাৰ্টা এটা পখিলা আছে। এই পখিলাই গ্ৰ' কৰাৰ পিছত তাৰ যিটো মলমূত্ৰ বা চেলাউৰি এৰে, অৰাৰ পিছত এটা ফাংগাছ ডেভেলপ হয় ইনচাইড। তো এতিয়া আমি এই IIT গুৱাহাটীৰে মোক হেৰি কৰিছে, ছাপৰ্ট কৰিছে। মই এটা ফাংগাছ নহয়, ডেট ইজ চলিউশ্যন। এই চলিউশ্যন এটা ডেভেলপ কৰিছোঁ। সেইটো আমি আগৰ গছত ইনজেক্ট কৰাৰ লগে লগে তাত কিছুমান মানে ফাংগাছ ডেভেলপ হৈ আগাৰ মেটাবলাইজ হয়। তো সেইটো এটা ভাল ৱৰ্ক, মানে খুব ছাকচেছফুলি হৈ আছে। আমি উইদিন 3-4 ইয়াৰ্ছত আগাৰ মেটাবলাইজ কৰি আছোঁ ইনচাইড দ্য আগাৰ ট্ৰী। আৰু আমি যিটো ডেভেলপ কৰিছোঁ তাত তিনিটা ফাংগাছ পাইছোঁ। এইটো IIT গুৱাহাটীৰ পৰা ৰিপৰ্ট আহিছে, তেওঁলোকে বাকী এনালাইচিছখিনি টেষ্টখিনি IITয়ে কৰে। মই ইমান নাজানো, মই জাষ্ট বস্তুবোৰ ডেভেলপ কৰোঁ। ডেভেলপ কৰি পেলাই ৰিচাৰ্চ কৰি আছোঁ কি ক'ৰ পৰা হ'ব পাৰে। গতিকে এতিয়া কোকৰাঝাৰতো আপুনি গছ লগাব পাৰে। কোকৰাঝাৰতো আমি ইনোকুলেশ্যন কৰি পেলাই আপোনাৰ লেটেষ্ট টেকনলজি প্ৰয়োগ কৰি পেলাই আমি আগৰ ফৰ্ম কৰিব পাৰোঁ। আৰু 1 হেক্টৰ মাটিৰ পৰা আপোনাক কওঁ, আজি 1 হেক্টৰ মাটিৰ পৰা ইকনমিক্সটো জাষ্ট আই এম টেলিং দ্য ইকনমিক্স, the people can understand.
I think if you tell it in English, it will have broader impact because
yes
from
yes yes. So actually I'm telling now economic. From 1 hectare land, if we cultivate agar 4,320 nos from 1 hectare, if it infected after 10 to 12 years, we can earn 4 crore 50 lakhs rupees from 1 hectare only. Only infected wood. And if we produce oil from this infected wood, another 3 to 4 crore we can earn. We can develop our region by this agar only, not other any crops. And if NIPER proof or IIT proof, it has been a medicinal property inside the oil, then it will go high. Because now 15 lakhs kg per kg agar oil. After that, if we found that there is a medicinal value high value compound inside the oil, definitely it will go crores rupees per kg. So we are working on it. If you want to know anything about agar, definitely call me. I will definitely help you. And I need your support and blessing in this regard in Agarut. We want to actually develop, and we want to develop in our nations and as well as our state. Thank you. Jai Hind.
So anybody else from the participants would like to give feedback for these 2 days proceedings and something you want to tell about way forward?
Thank you, Dr. Siddharth, for giving us this good opportunity. And I like to compliment your personality, you as a person, as you like you as a teacher. Okay, because students learn from the teacher. So a person, a teacher, a professor like you, who is full of positive energy, that see, I tell you, I'll just give, these are all mostly students. So see, I am from Ayurvedic college, like Ayurvedic school from Bombay, which is supposed to be the most, it is the most ancient Ayurvedic college. And still today, in our Ayurvedic college and in Ayurvedic hospital, even a patient is 103-degree temperature, we don't use even a single allopathy medicine for that, in emergency also. So we have got education from there, and that's the reason we have got confidence in Ayurveda. And when I was a student in 84, 85, I dreamed that I'll go abroad, and because our Guruji, Vaidya Sri Ram Sarma, used to travel all over the world, and then because he was sharing his experience and seeing his positivity and his confidence, we got that confidence. So it is connection of a professor, a teacher, and a student. So the students who are with you in the project or their student, I praise them that you people are lucky. And see, I know that whether the students here or entrepreneurs here, whoever is here, you people want to walk your way. But some are walking, maybe the speed is slow. Some are thinking whether I should start or not. But see, you have to initiate. You have to start walking. And once once you start walking and you have a goal, like suppose I want to go to the airport, first I have to get prepared mentally that I have to reach airport. Then only I'll stand up. I'll try to get a rickshaw or a taxi or whatever, and then only I can reach. So there is a very good saying by Tulsidas in Ramcharitmanas, "Jaki rahi bhavana jaisi, प्रभु मुहूर्त देखी दिन तैसी." है ना, मतलब हम जैसा सोचते हैं भगवान को आप उसी रूप में देख सकते हैं, है ना। हमारे अपने सनातन में कोई शिव को भगवान मानता है तो उसमें भगवान दिखते हैं, कोई राम को मानता है, कोई कृष्ण को मानता है. So see, it is mind power, and you know that all the motivators only on this saying, you go anywhere in the world, there are a lot of motivational speakers, motivational teachers. It is all your mind. It's all your power of your visualization, power of your imagination. So first of all, be positive. This herbal industry, Ayurveda industry, it is a sunrise industry. Nutraceuticals, Ayurveda, everything, it's a sunrise industry. Always because we are doing some activity, Dr. Sastry is joining us soon. We have made a very nice platform and we are open to that. See, because I'm from a village of UP, I came to Bombay, I did my struggle and only with the mind, I travel internationally. So to sum up with this, I'll not take much of time.
we all are gathered here. I will suggest Dr. Siddharth that you should make a WhatsApp group with whoever active people and we keep in touch, okay? And because I'm sitting in the commercial capital of the country, they're like, let it be FSI related things or any other things. There are a lot of people who wants to share their knowledge, okay? And let it be like how Dr. Sastry is in medicinal plants. You might be heard about Mr. Ramakant Harlalka. Harlalka's company Sugandhim is #2 in aromatic products in all over the world. And like now this year, we are now #1 in geranium production. So there are a lot of happenings in aroma industry. There is wood board different, Dr. who was talking about agar. So like for sandalwood and agar, there is different board now. That is called as wood board. For sandalwood and agar, this new ministry has created a new board and new targets and everything. Okay, so there is a lot of scope, but you have to decide. The power of sankalpa. Like when we perform puja at home, Panditji is giving a sankalp or he's taking sankalp. Means making decision. Once you decide, first you have to decide, and the success will depend how strong your sankalp or your decision to achieve your goal is. If it is strong enough, you will achieve the goal. And there are ingredients like hard work, discipline, patience. You have to wait. Success doesn't come overnight. Hurdles will come, but you have to find out the solution. You have to approach people. You have to meet people. You have to respect your elders. You have to respect your teacher, and you have to you keep learning. And now with this mobile phone and computer and everything with Google, everything has become so easy. It has become more easy to learn. So thank you very much once again, and thank you, Pramod sir, for being here. I visited your area, and I loved the area and people also. And the new generation, I met few people in your area. Like their dedication, the younger generation, dedication to develop their area or their Matrubhumi or Janmabhumi. The dedication to that, I really appreciate that. Thank you.
Now we would like to announce the prize winner for the research presentation. Today morning we had a session on research presentation. So I would request our Honorable Chief Guest, Shri Pramod Goroji, to present the certificate and announce the name.
Thank you, Dr. Siddharth. Dr. Vishwajyoti Bora. So, yeah, please come. Congratulations.
Congratulations, Dr. Bora. It was a wonderful presentation. I was all through your presentation. Thank you so much. Thank you, sir, for presentation. Now we will move to the other dignitaries. Maybe we can start with Manjula Madam. A few words on the 2 days proceedings and something for the way forward.
Good evening. Good evening to all of you. Honorable Chief Guest, Shri Pramod Goroji, Chief Executive Member of Boreland Territorial Region, Sir. Dr. Madam Ketaki Bapat, I hope I'm correctly pronouncing your name. Dr. Mr. Ranjit Barua, all dignitaries, press, media, all present here, and my dear students and scholars, scientists, the dignitaries present here. I don't want to take much time. We are eagerly actually waiting to listen to the chief guest because yesterday I was present But once again, I would like to repeat few things. Once again, what I have seen, or my little experience, I expertise this particular field. Even then, I was talking about the regulations and regulatory measures, the perspective and that perspective. It is very important to commercialize whatever product we develop. I feel since there is a herbal medicine global market is so high, so big, and which is expected to be $5 trillion by 2050. I think most of the things are not even now explored or evolved. I once again urge upon all our research personnel, the scientific community, including IIT Guwahati, to come forward for the larger applications of medicines, which is utmost important. I also feel that in the way forward, there should be an organized, sustainable system of plantation, the raw material availability. The sustainability is most important. Unless and until we have a certain volume, as in case of agar, some kind of a 1 hectare of land or something we can say are economics, but we don't have such for other herbal or medicinal plants. I think that really needs to be looked into. Most importantly, I find is the clinical part. Yes, whether these constituents are okay or not. Is old, definitely. We have been using since so many years. It's a 6000 BC when Ayurveda came, and we are used to it. Even then, now today's world is moving very fast, and we must have that kind of scientific background and analysis. I request this community to come forward and work together. The standardization is very important. IPR. I always feel that the patenting is so important. If we do not do the patenting, whatever raw material or whatever plants or herbs or medicinal aromatic plants are available, I'm talking about especially in Northeast. These are unique, very unique in nature. If these can be preserved, and this should not be region. I'm talking in the sense because importantly, this is standardization has to be done. The organized plantation will have to be done. Then patenting will have to be done. IPR is utmost necessary. I would like to look into that perspective because I heard some of the regulatory institutions or organizations or ministries have spoken length. Ayush and also Ministry of Ayush, Ministry of, I've seen this NMPB, they also have said, you see, FASI is already there, but so many regulations are there. Whether our entrepreneurs will be able to understand that language, whether consultants or the mediator will be required. So regarding standardization, I'm talking about because if a center of excellence is really is thought about in an premier institute like IIT Guwahati is an excellent idea, and we welcome that. I feel that this should be worked out unless and until the things are getting proper standard validation is done, clinical all analysis is done, and it is ready and okay, then only the commercial aspect can be thought about. The volume is very important in case of any industry. Otherwise, we cannot go and have a production. There is no commercial viability in that case. I heard the Turkish, the expert, Doctor. It was very nice regarding perfume industries, how Turkey has been doing. You see, they are not just saying that this is age old. No, what they have been doing is quite different. They are focusing the market. And completely Europe and USA market, they have captured Iran, Iraq and all. And we should also understand where the market is mostly we can penetrate. Mostly export market we have to look into, because nowadays the developed countries are really opting for that. They're switching over now to the instead of normal our pharmacy. So this few things I feel. Last but not the least, I would like to bring it to the notice of Madam Sanjukta and Dr. Professor Siddharthji, especially. There should be collaboration, a platform for all the stakeholders. Unless and until we come in to discuss and understand what is the need of the industry, what the rural people will do. They only will keep on doing their plantation in their home garden, home yard, or somewhere else. What will it their economics? We should think they should be paid for what they have been doing. So these are the few aspects. Maybe I'm talking about only commercial side. I'm not talking about the scientific side. But kindly look into that, and then the institutional, all the institutions, wherever are the ministries, various ministries, agriculture, maybe Ayush, maybe health, maybe all should come together on same board. And I think your school should take care of that. And, time is a major factor. If we keep on waiting, it's a very proper time. I congratulate the organizer that this subject has been brought. This is a very proper time. We are already late. We should not be late more. I will only again name that in Assam, there are some industries already existing. Few of them I told that there's a 1967 only the Ayurvedic, Assam Ayurvedic, one of the, which even today people, they buy the small things from there. Any rheumatic pain, you go there. The oil is there, ointment is there, honey is there. I'm just citing some few things. So this is under state government of Assam only. Now, pharma industries are very famous. Pharma industries are also doing a lot of researches. So we need to collaborate. We need to collaborate with them. They have very good research wings. I have seen Sun Pharma, Ajanta Pharma, Ojan Pharma, Hitaro Pharma, Pure and Cure, Hindustan Lever Limited, Godrej Consumer, Patanjali, and recently I think there is a Herbal Park. There's a Herbal Park that is in coming up in Borderland also. So we hope that we will listen from our honorable minister regarding this herbal farm and their estate. I once again would like to congratulate and thank the organizer for giving me this opportunity, and it's a privilege to be here. Thank you very much and wish you all the best for the Center of Excellence. Thank you.
Thank you so much, madam. As you rightly said, that we alone cannot do anything. So we need collaboration, and probably is the right time to talk about an Indo-Nigeria collaboration, for which I would like to call Dr. Debojyoti Bose from Yobe State University, Nigeria. Say a few words and say us about the way forward.
Thank you. Hello, a very good evening to IIT Guwahati and our respected dignitaries, delegates, colleagues, friends and students. I am Professor Debojyoti Bose, Head of the Department of Microbiology, Yobe State University, Damaturu, Yobe State, Nigeria, is here to present an initiation of international collaboration with Indian Institute of Technology, Guwahati. This is a presentation. Furthermore, after this conference and seminar, then we will be discussing more about this. And our chief guest is also here, so we'll discuss more about this matter and we'll try to finalize this process. So let me begin with the slides. So the basic objective is collection of traditional medicinal plants with antimicrobial activities and extraction of bioactive components, because it is very important to identify the bioactive components so that we can compete with other medicinal aspects like allopathy and all others. Then we can go for preclinical phases, especially for new antimicrobials, and clinical phases also especially for new antimicrobials, because for the available one, we can go for toxicity testing and all those, but for the new one, we have to do these things. Then further, we can go for commercialization of products. So these are the basic objectives. I will not take much time. I will go very fast. So some selected antimicrobial medicinal plants in Nigeria that we are using are Argemone mexicana, Ficus exseparatus, and so many are there which are showing antibacterial activities. Then if you go for antibacterial, antiparasitic, and antifungal, there are so many more that are used commonly in Nigeria. Enantia, Chlorantha, Azadirachta indica, even though Azadirachta indica from the name you can understand that it originated from India, but still they are using it in an enormous way. Next, we can go for antiparasitic only. So we can go here, Magnifera indica. So you can see many plants, many medicinal plants are common, but some are different. So many plants are there. Then we can go for some antiviral, which are very important. And we can name them as Mondia white, then Agerantum conizoides, and so on. So we have this. I think I updated the DOI from where we can get the detail. So maybe later on it can be shared. So basic reason for development of international collaboration. First of all is synergy. Because we need to collaborate, we need to develop, we need to grow further. Next is approach to the sustainable. Approach to the sustainable is very important nowadays and it has to be there. Next, we can go for effective processing of the products. Then technology transfer can take place. And finally, socio-economic benefit, which is very important. Not only commercialization, along with that, also local employment and all those can be covered up. Next is strategy for collaboration with Indian Institute of Technology, Guwahati. What are the strategy we'll be going for? That is exchange of raw or processed plant materials of significant value, because sometimes, we cannot directly bring the plants from which are indigenous from one country to another. Sometimes we have to go for bringing some extract by means, and so many clearance we have to go for. So definitely we do that. Then isolation and identification of bioactive components. Then we'll go for preclinical phases, like so many are there, toxicity testing and various testing processes are there, and so many are there. We'll go for that because we need to establish properly. Whatever we should do, we should go up to the end and up to the depth so that it can be commercialized or it can be given to the society in a proper manner, utilizable manner. So next we'll go for clinical phases. So clinical phases, definitely we have to go for human subjects after going for animal or cell cultures. And that clearance, ethical clearance and all those things we have to go for. And definitely it will be not so problem in our country. And here our collaborators will be, they are taking care of this one, Dr. Siddharth Singh. Then we will go for commercialization and socio-economic development. It is very important because wherever we are doing, wherever we are, suppose we are using herbal plants or medicinal plants, our society should also get the benefit. They should also go for employment generation and so on. So we can go for a conclusion that international collaboration with Indian Institute of Technology, Guwahati, with respect to research, exchange programs and industrial consortia will open an effective exploration towards traditional medicines extracted from plant origin, leading to betterment towards sustainability. And in fact, this collaboration will also strengthen the strategic and commercial ties between the Federal Republic of Nigeria and the Republic of India. So by this, I want to end my presentation. So thank you, all of you. Thank you, organizers. And thank you, IIT Guwahati.
Thank you. Thank you so much, Dr. Bose, for announcing the formal collaboration that we are going to have. Because it's not just the 2 days conference and we forget about everything. We should also carry forward something from here. So on the same line, I would like to call Dr. Ketaki Bapat Madam for sharing here her views.
Respected chief guest, Shri Pramodji Boro, and other respected dignitaries and participants and other dignitaries and professors and students. So let me first congratulate Dr. Sanyukta Patra and Dr. Siddharth, because the conference which was aimed at and successfully you have arranged, that is so complex. Right from the subjects, like from nutraceuticals. Okay. Yeah, I think from nutraceutical, herbal products, Ayurveda, and cosmeceuticals, many things were discussed. And actually, we were so delighted that all these stakeholders were actually called for, and we could get their wisdom and interesting presentation. So congratulations for that. There are 2-3 things which actually gaps were highlighted, and we really need to act proactively, because right now, going back to the history and saying not do now. Other startups also they have presented, like tea varieties they have presented. So many good things are happening, and we need to take it forward, but in a scientific way and in a very progressive way, without making anything. Supposing if anybody is using as an innovative idea for Ayurveda concepts also, like for tea or any other thing, So my request would be, those concepts should be validated the way you are making the use. Suppose any flower you are making use in such certain way, whether really your Ayurveda or the Shastra is actually approving or not. That is also very important. And I am sure the producers would take care, or if it is not, then please do it. Because here the idea of this conference and having these interactions, very lively interactions, what I felt that, okay, the charm is there. Everybody is geared up. A complete conducive atmosphere is there by our government policies and our young generation's enthusiasm, even techno incubators, startups, you name it, and it is there. And pertaining to NER, really I am very touched upon, because all these all these ecologies, what we are saying, and now we are ripping off the fruits. This is somebody else's and years together tapasya of this region. Because तभी तो ये इतना हो गया है. Otherwise, for any agar wood or something or any plant, it takes time to grow. And we need to look at the sustainability. So these aspects, I think we need to take it. Similarly, for labeling of the product, when I was looking at this startup, some product I got in, though the efforts are really noteworthy, but the labeling and this was not proper. Let me tell you, I will not name, but it needs to be very careful. And then FSSI has made a very beautiful framework. I think we should use all these things. The things are there, but I think it is not reachable to the people in the way they want. And for that, I would again request the Ministry of Ayush or FSSI or any requested, the involved ministry to come forward, and how we will enable this ecosystem, innovative ecosystem, a strengthening one. Can we have a seamless kind of a transfer, or which forms to be filled? The startup person was telling, oh, I am taking so much time going here, there, I don't know what to do. So these are all, see, we cannot blame them. They are creative, they are innovative, but we need the guidance. And how best we can do, we need to have one kind of a framework for that also. Similarly, here can we make this India as a hub, if you are thinking, like today when we are listening to Dr. Datta, if they have very innovative concept, And now our Indian flowers, Indian herbal medicine, so whatever you name it, we have some peculiar fragrances. And if we are using it, any kind of a indicative molecule or so, and if we can identify with any AI related model or any chemometric kind of essays, that how different it is. The ideal one, how it is, and now how it is. So that, climate change indicator also we can think about it. Similarly, when we are thinking about this NER kind of a, or making a hub here, so we need to see that it is completely local, local material, local packaging, and locally done, so that we are able to, take care of completely. Otherwise, again, carbon footprinting, you are making something X thing here, Y thing somewhere else, and see again coming back to you. It is happening in wool industry in Himachal Pradesh or Uttarakhand. Wool, they are producing it, goes to Panipat for processing, comes back there, they are making something, goes back there. So this is what is the thing. And for that, we can work on cluster or whichever appropriately we should do it. So this is what, and when we are having the collaboration, let me say here that to facilitate the ease of technology transfer, our principal scientific advisor's office has taken initiative to make a technology transfer form, which where the concerned ministry representatives are there, and we are working on that so that we can any bilateral kind of a collaboration or this kind of a collaboration as it is proposed now, very interestingly. So we can help in a much better and in a scientific way. So that's all I could say, and I would urge students also to make use of it, and let us have this. Can we have these two products as a cosmetic products or something like that? Can be made in 6 months' time from NER? Can be? Because Dr. Kushwaha has also indicated, today I visited Naikat, so marigold kind of a things, they have isolated some compound and it will be useful for cosmetic. So again, I would request Dr. Siddharth, can we aim? Because if we high aim means we will be able to. And their school of agriculture and rural technology is there. Dr. Sayukta is there. Dr. Siddharth is there. Can we make? Because unless we have the motto or the focus, I think we won't be able to. So I with that hope, and let us make, and any thing from principal scientific advisor's office is required in a scientific way or any which ways, we are always there, and wishing you all the very best. Thank you.
Thank you so much, madam. Now, for the very specific guidance which we were looking for, I would like to call our chief guest, Shri Pramod Boroji, to say a few words so that we get encouraged and we get involved in such activities in future as well.
Thank you very much, Dr. Siddharth and other organizers of this today's wonderful seminar. And I'm very sorry that I should come yesterday, and I missed very important sessions that you have discussed so many new things which actually I wanted to learn and listen from you. Being here, I'm thinking something and I'm feeling something that since for long years I was in student movement and social movement, and my long experience taught me something, that how things happen in any part of the world and any part of the country, and how things not happening, though we want that it should be happen. So today I'm glad that most important, the scholars and the thinkers are all together here. Maybe I am only from government, and Madam also retired, so I don't expect any new thing from any kind of invention, any kind of innovation from government side. Most of the time we wait for government policy, but policy comes from institutions, because the institution do the research, and the research are done by the scholars, and you are doing the research, you are doing the experiment, so you know the thing, but we don't know. I don't know. So ideas generate in the institution, not in the government office. And in our country and in some part of, maybe in other part of the globe also, it's happening that we're waiting for someone that government will do this, government will do that. But real things, what we expect to be done by the government, that should be started by the, the institutions. And today, Guwahati, IIT Guwahati is doing the best thing. I also congratulate and thank Siddharth and his colleagues for organizing this wonderful seminar, and also collaboration with Nigeria and other institutions. So actually, we all the year we eat so many herbs. So we are herbs eater, medicinal plant eaters. Traditionally, at the time of Bihu, first day of Bihu, we eat hundreds of different, the plants together, and we take it like the medicine. And there is a belief that the tribal people, we don't have much, the health problems in village because of that. But that's not there. It's already over. Now, chemical coming everywhere. No more organic food, no more herbal food, no more natural foods are there. I think yes, yeah. Please, yeah. Okay, please, yeah. So knowledge exchange and idea exchange is very important at this point of time, and good things should start as early as possible, because we have already done so many wrong things in past few decades. So to stop this, the wrong things we did, we should start some good things as early as possible. So most of the people now becoming very conscious of their health and their life and everything. So we have still those things are there in the art, which can save our life, which can sustain our livelihood. I mean survival. So ये सारी चीजों को करने के लिए हम लोग ज्यादा वेट नहीं करना चाहिए. आप लोग के पास नॉलेज है और हम लोग के पास एनर्जी है, रिसोर्सेस है करने के लिए. तो इसको इन कोलैबोरेशन, जैसे कि आप लोग ने बोला है, उसको जल्दी करना चाहिए. और हम लोग का जैसे जो लोग हैं, तो उसको फैसिलिटेट करने के लिए हम लोग बैठे हैं, क्योंकि हमारे एज में हम लोग ना रिसर्च कर पाएंगे, ना अभी कोई इन्वेंशन कर पाएंगे. जो आप लोग कर रहे हैं उसको हम लोग फैसिलिटेट करने के लिए हम लोग तैयार हैं. तो इसलिए आई वेलकम ऑल ऑफ यू टू बोडोलैंड. बोडोलैंड इज मतलब कहीं भी नहीं है, आसाम में ही है. तो ये छोटा सा कोना है जहां हम लोग भूटान के साथ साथ रहते हैं. और आप लोग जानते हैं भूटान में जो हैप्पीनेस इंडेक्स जो है दुनिया में, शायद वो लोग फास्ट शुरू किया है. अभी हम लोग को भी एक हैप्पीली जिंदगी जीना है. तो अगर हैप्पीली जीना है तो हम लोग का प्रॉब्लम को हम लोग को कम करना पड़ेगा. और प्रॉब्लम कम करने के लिए तो बेसिकली जो हेल्थ प्रॉब्लम है, अभी कैंसर इतना बढ़ रहे हैं. मैं 2 साल हो गया पॉलिटिक्स में आना, अभी तक थाउजेंड्स ऑफ मतलब लोग को मिल चुका हूँ जो कैंसर पेशेंट्स है. और कोकराझार में अभी कैंसर हॉस्पिटल टाटा ने स्टार्ट किया. उसमें 2 महीने में 300 से ज्यादा वहाँ कैंसर पेशेंट आ गया है. तो और भी बहुत आया नहीं है डायरेक्ट वो लोग वहाँ चेन्नई में जाते हैं, बाहर जाते हैं. वो सारा यही हो रहा है कि जो अभी फूड हमारे पास आ गया है, तो हम लोग अपने फूड को छोड़ के कोई मतलब बाहर से जो फूड खा रहे हैं तो उसमें जो केमिकल आते हैं उसके वजह से, और गरीब लोग जो है वो सस्ता में चीज को चाहते हैं। पहले हम लोग का गांव में वो सब नहीं जाते थे, हम लोग कोई भी उठा के, कोई हर्बल हो कोई भी हो, उसको उठा के हम लोग खाते थे, तब इतना बीमार हम लोग का नहीं होता था। हम लोग गांव में चुनता ही नहीं था किसी को प्रेशर हुआ है, डायबिटिक हुआ है, कुछ प्रॉब्लम हुआ है जब बचपन में। अभी तो गांव में हर लोगों का भी प्रॉब्लम हो गया है। तो इसीलिए ये जो काम है इसको जल्दी से आगे बढ़ाना है। तो फ्रॉम माय साइड ऑल्सो मैं आप लोग जो भी किया है इसको मैं भी पढ़ूंगा, देखूंगा डॉक्टर सीडर से कलेक्ट करके, और उम्मीद रखूंगा कि आने वाले समय में आप लोग जरूर कुछ करेंगे। और हमारा डॉक्टर साहब ने जो बोला है गुरु के बारे में, हम लोग प्लानिंग कर रहा है कि अभी 2000 हेक्टेयर जो लैंड है हमारा, हम लोग निकला है वो काम करने के लिए। तो आप लोग से बात करूंगा। हम लोग का शुरू थोड़ा लेट हुआ है, हर विषय पे थोड़ा लेट हुआ है हम लोग का शुरू, इसीलिए थोड़ा हो नहीं रहा है चीजें, बिकॉज यू ऑल्सो नो that our region was disturbed for last many decades. Now peace prevailing in the region, so we are also starting to think some good things that should happen, and we need your intervention, your support. आप लोग का इंटरवेंशन, आप लोग का सपोर्ट चाहिए, बाकी सपोर्ट नहीं, नॉलेज सपोर्ट चाहिए। अगर ज्ञान मिल गया तो सब कुछ कर सकता है, ज्ञान ही हम लोग का कमी है। तो बहुत बहुत धन्यवाद एंड कॉन्ग्रेचुलेशन वंस अगेन फॉर दिस द वेरी मीनिंगफुल एंड वंडरफुल टुडेज सेमिनार। मैं आपको धन्यवाद देता हूँ। धन्यवाद।
Thank you so much, sir, for your inspiring words, and I'm sure you will keep inspiring us and supporting us in our future endeavor also. Really, we will come soon again to you with some concrete proposal. Then in today's session, the concluding talk we would like to hear from none other than Dr. J.L.N. Sastry. He doesn't need any introduction. He was the chairman of NMPB, and the kind of work and vision he is having for this herbal product sector, that is unmatchable. And without much delay, I would like to call Sir. Sir has come all the way from Bombay just now, and he has directly gone into the stage. So in one side, we are sorry that we couldn't inform you before, but on this other side is something a matter of delight and honor for us that you could make it here. Thank you, sir. Over to you.
Thank you, Dr. Siddharth. Respected dignitaries on the dais, and all the students. Indeed, it's my pleasure, Siddharth, to be part of this gathering. Doesn't mind how much you traveled, and when you really are committed to something, you should not look at how many hours you are working. You should deliver the things, and then you have to look at your watch. That's how, at least I believe. At the outset, I congratulate the entire organizing team as well as the students. When I came to know, just I requested Dr. Vijay to flow from Bombay and to attend, because we have a small startup right now. As you rightly said, I was the former CEO for National Medicinal Plants Board, full-time non-IFRS CEO. But of course, because of my own perceptions that came out, I'm really thankful to Government of India for giving me the lateral entry. I did enjoy, and we formulated a concept under Atmanirbhar Bharat called VIBGYOR. And I still remember 22nd July 2000, when in the South Block we had the final meeting at PFC. Ms. Somanath, after listening to have a problem? Okay. To the objections of the entire team as well as the other departments, he said somebody thought about the future of this country and given this project, and he allocated about 4,000 crores for the VIBGYOR.
And at that time, also my focus was Northeast. Initially, I was thinking it is Seven Sisters, but today I call it as Ashtalakshmi. So that's the only difference. Right from 2015, I'm coming to Northeast. And on my first visit to Imphal in August, it must be 13th August 2015, if I am right, when Dr. Balaprasad was the PCCF. I realized as an Indian, we did a mistake for not getting focused on Northeast. And since then, I'm relentlessly working. Even through Medicinal Plant Board also, we tried to resolve, but my tenure was very short. And I quickly realized that I have certain goals where I need to get focused myself than blaming any system. I came out and we started a small startup. There are multiple things like previous speaker was mentioning about Podoland and then the livelihoods related and so many things. Each of these regions have inherent problems. So as responsible citizens of this country, we need to find solutions. We enumerate the problems and we really understood the problems. Do we have the solutions? Like when I went to Mizoram a month back, I realized Mejoafera is their state tree. And do you know we are importing Mejoafera from Sri Lanka? It's the national tree. Okay, just by collection of the flowers, which are abundant in Assam, Manipur, Meghalaya, Arunachal, and Mizoram, you can make money. These flowers are coming in thousands of metric tons from Sri Lanka and being sold at around 200 rupees per kilo. And if you try to cultivate it in any other part of the country, it's the incubation period is large. It will take 15 to 20 years. But in this area, the weather is conducive. That is polyphila. We talk about the cultivation, we don't do that. Ashtavarg, as a matter of fact, Chyawanprash consists of Ashtavarg. No brand of Chyawanprash in this country has Ashtavarg. We have one excuse that since it is not available, you can use the substitutes. Instead, industry is using, I was part of it, I'm shameless to mention that, I was part of it, using all the excuses and they don't try to cultivate this Ashtavarg and still the formulation is called Chyawanprash. So why don't we do that? It will be gold mine. Sikkim, Arunachal, they have done a lot of work and even some places of Assam also you can cultivate this. We can come together with a plan. I have blueprint for anybody who is interested. I can share. There is no IPR on that. IPR is for the nation, not for me. Select 10 plant species and 5 tree species and focus on this. Many times I met 3 of your CMs in this region and I was enlightening them not to go for any industrialization. You are fortunate for not having industries in this place. Please feel proud for that. Today, the entire country is just getting organic certification. It is certified organic, not real organic. Friends, please realize, if at all you want NPOP FDA, you will get it from Northeast, nowhere else in the country. Why don't you preserve it? 86 to 92% of Mizoram is forest. More than 75% of Arunachal is forest. And almost 50-60% of each of these states are having rich forest cover. Consider this as the boon, and most of these areas are with the local communities, oblique tribes, and you have the diversity of the tribes in the entire Himalayan belt. And why don't you use that as your SOP and make your own fate? Since you have not realized your strength, you are thinking it's your weakness, it's your strength. Just I was mentioning while coming to Rahul, Suppose if you have a crop, you have an organic certification, immediately be 2x. Price will be 2x. So 100, suppose it's 300%. Now the price is 100 rupees becomes 300 rupees. Put 100 rupees on your freight when by airlifting. Still you get another 100 bucks. Why are you not looking at it? Why do you want to establish industry here, then create pollution here, lose the organic certification, which is otherwise not possible in any other part? And for industrialization, my friends, again, competitiveness in the market. If you make a product here, the freight will kill. Even if you talk about the Northeast freight subsidy and all, it doesn't work after GST. So the best thing is to use it in its virgin way. Don't spoil it. Your strength is organic certification. In other places in the country, it will take 3 years. And it may take only 3 months for you. Even for organic collection, APADA can give you organic collection. And many times we don't realize that the NPOP of India and NPOP of USA have only 5 steps access. That's it. So if we can just realize and train, we are here to help. Of course, I'm not expert. I'm jack of all. Since I worked as the Dabur's CEO head for R&D global. So I can help you out, and where I can tell you about what are the territories. How many of us know that your Giloi, Tinospora cardifolia cannot be exported to Uzbekistan? Any idea? Okay. So yesterday we said, let us send them Tinospora malabarica. What is bad about it? You have all the species. And especially, I'm glad the CEO and State Medicinal Plant Board Assam, he had written a detailed note to me about the mixture of these 3 species, Crispa, malabarica, and Tinospora cardifolia during the COVID time. Because the noise made by some of the hepatologists was on Crispa. Crispa has side effects, it's documented. So if you separate, probably Uzbekistan will tomorrow take Tinospora cardifolia. And Withania somnifera is now under the clinical trials at the London School of Tropical Medicine, Hygiene and Tropical Medicine. Tinospora cardifolia monograph is there in British Pharmacopoeia. There are more than 96 herbs in the US Herbal Medicine Compendium. A lot of things. So are we going to explore this? Why I'm giving this enumeration? They talk about traceability. And in India, this trade, according to the document I generated, or the data I generated, the total business opportunity of raw material is somewhere between 40,000 to 70,000 crores Indian rupees. And more than 90% is unorganized. And if you want to make sincere efforts to organize, like we have one Muthu Veladam, actually these kind of people are real heroes. He got 164 acres of private forest near Madurai, where he trains the local tribal healers. Every year he conducts workshops for them, training them how to manage themselves. In the Northeast, Arunachal, Assam, other places, you have a lot of local healers. Develop complete knowledge through them, whatever folklore claims they have. The good news is the new regulations are going to allow you to introduce herbs which are not there in the Ayush Pharmacopoeia. This is another good development. Are we geared up? We are not. We talk about these hardly 350 herbs which are there in the market. Time and again, we are increasing the so-called bioburden. We don't talk about bioburden. I put more weight on Karanam Malleswari, who got the gold medal in the Olympics, and she can't lift in the next Olympics. So why don't we create 10 Karanam Malleswaris? Therefore, we get 10 gold medals. So bring in more plants. Don't add burden to the existing species. So for that, where is the diversity? Where is the plan? So I have a lot of ideas in my mind, but this time very short. And of course, I'm sorry I am an uninvited guest for you, but I'm thankful to you. You have allowed me to be part of you, but I promise you I'll be with you. I created your regional facilitation center when I was in the Medicinal Plant Board at the NIST, Northeast Institute of Science and Technology. Nara Shastri and his team, Mohanlal, they are really doing well. So, and you have in Impal, you have ISBD, and at Shillong, you have National Institute for Ayurveda Homeopathy. Can we bring them as single consortium, Northeast Consortium of Medicinal Plants? Okay. And don't call people like me who talk many things and don't do anything. Okay. So I can definitely help you with my ideas, but I need this young generation who can really make things out of it. Okay. And the opportunity like a Mizoram, I met the CM and I said, your state is about 92% forest, don't spoil it. You have an opportunity of 1,000 crores if you really believe yourself. Homeopathy. I'm an Ayurvedic physician, but I'm glad I could throw some light on homeopathy also. About 2,000 crores of homeopathy tinctures are being imported into this country. You have any idea? 2,000 crores. Can't you make 50% of these tinctures in India and save foreign exchange for this country in euro dollars? Feel proud. You are contributing in multiple ways. You can, and Northeast is only going to play a major role. Like I got a call from the former MD of Natrion, New Jersey, and if everything goes well, he said he'll give me about $4 to $5 million for my project. And I surprised why. डॉक्टर साहब अब इंडिया से जो भी जड़ी बूटी आएंगे यूरोप में तो रिजेक्ट करेंगे क्योंकि हम ना पेस्टिसाइड देखते हैं ना कुछ और देखते हैं तो बेहतर यह है कि आप जो कॉन्सेप्ट लिए नॉर्थ ईस्ट में करने का वही सही लग रहा है आई मेट दिस जेंटलमैन ऑन 21 अप्रैल इन्वेस्टर समिट एट गांधीनगर मोदी जी जो उद्घाटन किए थे and later it faded away. We had a lot of discussions, but once its samples were tested in Europe, we realized the importance of organic cultivation. Friends, last 15 days, again, Vijay is there, is the witness. He could not succeed in getting me 10 metric tons of ginger from this region. When I decided to give him a blank check, ask him, blank check, you put your price. If I don't pay, you ask me. People are ready to sell it. Last time when I came here, about a month and a half back, I really got surprised. You get only 50-60 rupees per your ginger in the local market. And when I'm giving a blank check, you are not in a position to give me for one reason. You don't know what is the definition of organic. You don't know how to trade. So finally, he said he got some FPO which has an organic certification that is sufficient. Let them procure and supply. Na? When I test in my lab, it should abide to certain what you call requirement or the norms. And then it is a big opportunity. And likewise, you have plenty. You have black turmeric. You have ginkgo biloba coming from here. What not? You are sitting on a gold mine, and a person coming from what do you call that? Extreme south. I should be really mad, na? Traveling so many times to this place. So I suspect that this guy got some personal interest, and he's going to make some commercial benefit out of it. And you try to exploit your resources before I exploit. Okay. And rest of the things we can discuss. Thank you very much. I don't want to bore you at the last part of this wonderful session. And you can take whatever. Yeah, I can see. Very, very simple. The same concept under Vrukshaish, what I wanted to do. Let us knit the entire herbal trade onto one platform. At that time, I called it as an IT platform, IT corridor. Itanagar to Trivandrum. Itanagar to Trivandrum. There are 16 states through which our southwest monsoon starts its journey and takes a curve, leaves Arunachal Pradesh, comes back now next month as northeast monsoon and leaves this country via the same route. By this blueprint, you will be reducing the dependency on the groundwater. That is the first thing. If at all you want to effectively use the natural monsoon rain, this is the best way. The second thing, understand germplasm. Most of the Himalayan species, why they are not surviving in other regions? Because there is no genetic diversity in these plants. How to bring in diversity without changing the chemical constituents? So your conventional 15 agroclimatic zones doesn't work if you are working on the medicinal plants. I vehemently oppose the scientists. I'll be opposing. You have to divide this country into 8 geoclimatic zones. For one reason, paddy will be paddy whether you cultivate it in your Srinagar Valley or Kanyakumari. But Sasaria Lappa or Ginkgo biloba or your Gucci fungus or Ganoderma lucidium, you can't cultivate it wherever you want. They have lesser genetic diversity. So you have to find a solution. Can you make an alpine species as a temperate species? Can you make a temperate species as a subtropical species? What are the obligations you have? So you have a huge, humongous task in front of you. And you talk about the AI or machine learning or what you call algorithms. Track those previous happenings through your algorithms, generate algorithms and find out what is the best solution. Taking a small satellite picture, looking at the soil typing from Palampur Institute, I introduced this saffron cultivation in Sapor. Sapor is the apple city of this country in Baramulla. Because tourism, it will take time to reach this place. So I thought the better way, and in 2 months you will get whether you have worked for saffron or not. There are scientists who have worked on saffron to get what you call awards. But I don't know how many of them really seen saffron calms and how do they work. In 2 months you get more price than what you sell gold. So Pampur is the traditional area of saffron. Today, in 4 different localities of Sapor, saffron is available. This is like grade A and B. There are ABCD grades according to BAS. A, very rarely you get even in Pampur. Now, this particular sample, which we cultivated, just you require 2 months, August and September. You need not work the entire year. Initially, when you lay the bunds and keep the calms, that's the only effort you have to put in. And lifetime gold mine. And one of the best varieties you have in Baramulla. And now rest is history. I'm glad on 21st April, I received a sample of 2 grams of this saffron. When we tested it, the crocin and precrocin contents are almost equal to the variety which comes from Iran. So you have to work. When I have taken Inula racemosa to Leh Ladakh, I was not knowing whether it will grow or not. Today, rest is history. 6 villages we adopted about 7 years back. And today, Inula racemosa is back into almost near to its original flora. For one reason, right from Hindu Kush ranges, it is migrated, and today only under cultivation you see it in the Kelang area. Now from next week onwards, Kelang will be closed now because of the new tunnel. Maybe the transportation is open, otherwise it's completely closed. Now this plant, which takes about 6 months to grow to a particular height, it could attain the same height in 2.5 to 3 months when we have taken it to Leh Ladakh. You look at Ratan Jot, which otherwise is available in Lahore Valley. Spiti Valley, not Lahore Valley, Spiti Valley. Lahore Valley is for the inland and social park. So you can migrate these plants with the proper planning. And with the recent request of Dr. Chaurasia, I could not go because of some brother in the family, I could not go. Otherwise, in August, I promised him. We wanted to take hing cultivation to Kargil, where the migrations can be reduced. And you can help the Indian Army indirectly. Because the migrations, if they are down, they cultivate the vegetables at least for 4 or 5 months. Otherwise, continuously from either Hindan, Chandigarh, you have to lift the vegetables for the Indian Army. And Kargil area, you have the natural populations of 2 varieties of hing. Instead, we are breaking our head to cultivate it in Palampur. Okay. I don't know how many of you have seen the efforts. I really appreciate the efforts of IHBT and Dr. Suresh for this. But it doesn't grow. If you have ever seen. And one of our friends from Hyderabad, he got a tie-up with Uzbekistan Agriculture Ministry for 10,000 acres cultivation of hing. Hing is a gold mine. And these are the plants you can try for upper parts in this particular region. So my endeavor is to bring in the plants which can be grown in the given climates, which are exportable, no standalone crops, wherever the value and volumes are low, because it will not give livelihood. And our project is to give salaried farmers for this country, the farmers who will have a PF and ESA facility. That's my design. And since my concept is seed to shelf, I want to be everything. And then on the way I was mentioning about tomato and this pyaj, hardly they get 5 rupees, 10 rupees. But in the market, we purchase for 50, 60, and sometimes 200 rupees. So in the supply chain, where is the value addition? We say because of value addition, there is no value addition. Tomato is tomato, it is being purchased from a farmer for 10 rupees, sold for 100 rupees. Where is the value addition? Have you ever asked? We come across a lot of platforms, we discuss, debate. Is this value addition? Exploitation, blatant exploitation. So try to intervene here. So my concept is that, nothing else. When I present my case to many people, venture capitalists, they say, okay, you have 60% operational margin, Dr. Shastri, why do you want to give 15% incentive back to farmers? I said that this whole project is for the farmers. Okay, so if people are interested, my team, of course, Vijay will be here, and I'll be coming time and again. And Muthuji is there. Tomorrow we are going to Manipur also, and our effort is to get medicinal plants cultivated in an organized manner and create an export market in this region, which ultimately gives the livelihood. And I'm thankful, without mentioning FISS, which is the NGO, our partner here, which has given us the office as well as the working facility in Damasa. It is a wonderful experience the last 3-4 months for me. And I'll be part of any of your activities, if at all you feel I'm a befitting individual for you, or my team is ready. And Vijay will be stationed here most of the time. And few people who are interested, they can join. We can definitely form a team. And we have identified about 20,552 hectares, 25 FPOs, 2 individual entrepreneurs, about 3 SHGs, having about 19,000 odd farmers. And that's the project on which right now we are working for Assam alone. And in that, yes, I need people. Without people, we cannot do anything. And thank you very much, Siddharth and team, for making me a part of this debate. Wonderful. Namaskar. Jai Hind.
It was an absolute pleasure to hear you, sir. I am sure it must be inspiring many of our young friends and fraternity from industry and startup ecosystem. Without much delay, because we are already late, but I won't say people are sleeping, they are still enjoying. So we will hand over some memento to all our esteemed guests. I'll request Neha, and, Arunima, Laura to please present the goodies to our esteemed guests. Start with Dr. Jalen Sastry. Before we depart for IT, I would like to request our head of the department, Professor Sanjukta Patra Madam, to give official vote of thanks so that we can conclude this session.
Thank you, Siddharth. Good evening to all present here. First of all, I would like to, before saying goodbye, again, a final welcome note. Sri Pramod Boroji, Chief Executive Member of Bodoland Territorial Region. Guest of honor, Dr. Shastri. It was very knowledgeful talk you gave us, and we definitely need your hand holding. Dr. Mrs. Ketiki Bapat, Advisor Scientist G, Office of Principal Scientific Advisor of the Prime Minister, and of course our own. And we also have on the dais Dr. Debajit Bose, and finally Manjula Saikia ji, who had been always with us throughout in all our events, and she has been constantly with the School of Agro and Rural Technology. All the guests present on the dais, thank you all for being with us, sparing your valuable time to make this event a final success. To start with, I would like to thank Director IIT Guwahati and Deputy Director Professor Kakotiji, who was with us yesterday. Without their enthusiasm, motivation, and support, we would not have been able to take this event. We had a similar event just 2 months back, and this time it was MedProTech, and it was an aptly organized event to start with the knowledge base and resources, taking forward towards the processing part, the commercialization part, the regulatory aspects, and finally how to achieve all these together. These were all the points which were put together in this particular conference. And we had all the stakeholders who were at these different points. So thank you to the organizer, who was Dr. Siddharth. He almost did it single-handedly, along with all the students. We were definitely with them, but definitely he did it single-handedly. So thank you, Dr. Siddharth and his entire team. Thank you all the dignitaries, the participants who were present here. Thank you to all the media persons who were present here. It was, and all the speakers who delivered their talks during the different technical sessions. It was not just knowledge oriented, but it was also giving us the exact situation, where do we stand and how do we need to take it forward. With all these words, I would not like to take it much further and say thank you to all, one and all again. And for a final say, definitely I will hand it over to Dr. Siddharth, because I feel without him, this cannot be concluded. Thank you.
Thank you so much. I'm sure everybody is hungry and sleepy. It's a long day, so I'm not stretching it. Please enjoy your tea outside. It's a it's a cold evening, and warm tea is essential. That's the first prerequisite. So thank you everybody for coming here and making this event really successful. Thank you so much.
आयुर्वेद का नंबर 1 दवाई है अगर तो आपको खड़ी हम सर्कल लेके आओगे सब कुछ सुना देते ना पता चल ये अभी मेरा रिजॉर्ट कंपाउंड है